Hi everyone, we're just waiting for the YouTube stream to be up. And then we can start. Welcome everyone. Okay, we're starting in a second. We're just having some issues with the YouTube stream. But once that is fixed, we'll jump straight in with our talks.
And then it's possible to see this presentation, no?
Excuse me?
It's possible to see this presentation in YouTube, no?
Yes, exactly. So we're live streaming on YouTube. We're just making sure it's definitely working, which is what I'm doing here. It is loading. I think we may just assume that it's working. And then I think we'll just have to hope for the best. Anyway, it will be recorded on Zoom, so if anyone who's not here, they'll be able to see it just later then. Sorry, everyone, please bear with us. We do have some time to buffer for things like this. All right, welcome everyone. Hello, my name is Ellie. I'm the MC today. We're in the theme D on cognition, motivation, and emotion, and this is a session of traditional talks. That means speakers have up to 12 minutes, and then we have some time for questions and answers. Please ask questions. They will be answered even if we run out of time. The speakers will stick around to answer your questions. You can vote questions up if you want to. We'll give you the opportunity when you ask your questions to also ask them in person by unmuting you. If you have questions that you think may be stupid questions, they're not. We really want to create a level playing field here. So if you have a question and if you've not understood something, you're so welcome to ask these. With that, I'm happy to introduce our first speaker, which is Pablo Maturana from the University of Barcelona. And Pablo's going to be talking about the neuromechanisms of emotion regulation. a study with fMRI and dynamic causal modeling. Pablo, if you want to share your presentation with us.
Okay, I can start now. Okay, good morning. My name is Pablo. I would like to speak about today about the emotion regulation and a study with fMRI and using DCM and dynamic causal modeling. I'm working in the psychiatry service in Belvitge Hospital here in Barcelona and in the University of Barcelona. And I would like to start this presentation with this sentence of Olaf Sporn. Because the brain is a complex system, maybe the most big system complex in the world, maybe in the universe, I don't know. And it's interesting because the brain is a fixed architecture. It's only one architecture characterizing its connectivity. But this architecture have many functions like action, perception, or cognition. For me, it's very interesting using the neuroimaging data to understand what is the dialectic between a structure and function. I think the most important study in my case is to analyze the connectivity in patients with pathology or normal patients. So I would like to make 2 or 3 slides to speak about the connectivity, what's mean the connectivity. We have here 3 kinds of connectivity. Structural connectivity, this is only about the presence of axonal connection. For example, in this case, tractography, DTI, or a tracer study in monkeys. Functional connectivity is a statistical dependency between regional time series. In this case, we can use the information with fMRI or EG or MEG, and we can study the correlation or study using independent component analysis. And finally, the effective connectivity is the influence between neurons or neuronal population. What is the relation in one area with other area? In this case, we can study the direction. What is the technique to study the effectivity connectivity? There are many, but maybe the most important in this moment, because have many components, is the dynamic causal modeling, or the DCM. Other is the Granger causality, other technique is CEM. So I would like to speak in general about the DCM, because maybe it's a little complicated, but I would like to say something in general. Here we have a network, and the network is composed of the node, in this case, our region of the brain, or voice or voice. The connection between this node and one input to modulate the response. So in fMRI, the result we have the signal is the signal volt. Is a contrast. And in this technique, we can to study, or we can to understand what is the neural response. But it's not directly, it's indirectly using the signal volt. So in this technique, in DCM, we need to model the response neural response. So we need an equation to understand or to approximate the response. And in this case, we have this equation. It's not complicated because we have here the function in relation with x in this case is the node, u is the input, and this letter is the parameters. So we have here the response, neural response, And on the other side, we have here defined the signal bond. So we have here two equations to understand or to approximate what is the response neural in activity, in one activity, or in a task in fMRI. So how we can do this approximation? X in this case is the node, is in relation with the time because it's a time series. To understand this equation, we need other equation or other series. This is the Taylor series, and the Taylor series give a response or approximation and this question: what is the neural response? So we have here A, B, and C, three different components in this Taylor series to understand the network. So in this case, A is endogenous connectivity, is the connectivity is the edge between one node and other node. B, in this case, is the modulation of connectivity. In this case, the input modulatory and the drain modulatory is a representation of response of the brain, because the brain in some time respond fast, on other case respond slow. This is the paper to speak about what's mean the driver or the modulator response. And finally, we have C, and C is the driving input, is what is the input to understand my network. I know the explanation is easy, but I would like to give a general mark to understand what's mean the DCM. So, in my case, in my study, I'm studying the emotion regulation and obesity, because what is the relation between this topic? There are many studies speak about the relation in this kind of patient in obesity with emotion regulation. So, for example, I put here some examples: alexithymia, difficulty in identifying feeling and externally oriented thinking style, which in turn my interference with subsequent emotion regulation response. There are many studies speak about what is the role or why is important the emotion regulation in obesity. For example, here is a good approach to take the emotion to understand why is the final cause to obesity. So there are many papers to speaking about this subject, review, etc. So my goal in this case is to study the emotional regulation in this kind of patient and in other group of patient control, especially in one task. This task is reappraisal. What's mean reappraisal is rethinking the meaning of affective charge, stimuli, or events. So I have here a scheme in general to show maybe the most important part in relation with the reappraisal task. In this case, is the amygdala in relation with the prefrontal cortex. In this case, ventromedial, dorsomedial, and the regulation to amygdala, in special to emotion. So my study is part of this study, and the study general to understand the emotion regulation in women with obesity. And I would like to study in special what is the direction or what happened in this kind of patient. So I have here the task. The task is the reappraisal. This is a classical task. You have image, salient emotional picture. during 20 seconds, and then other kind of image in 20 seconds. And the task in this case is maintain the motion, or suppress the motion. In this case, for me, the most important thing in this case is suppress, because when the patient suppress, it's possible to see the regulation of the emotion. So in this case, for me, the input to modulate my network is the suppress or regular. So this is the sample. I have healthy weight control, 18 patient, and obese patient, 70 patient. Here are some the area to study: the amygdala, and two areas in the prefrontal cortex, the right dorsolateral prefrontal cortex and ventromedial prefrontal cortex. Here the coordinating NI. So this is the driving and modulatory input to my network. In this case, the first input is, I put here emotion, but in general is aphotic. It's all the image. When the patient see the image, for me is the input to my network. But the most important thing in this case is this signal, because it's when the patient suppress the emotion. So for me, I would like to analyze in this patient with obesity how can regulate the emotion. So this is the modulate signal to analyze my network. So this is the proposed model. So I have here the amygdala and the ventromedial prefrontal cortex and dorsolateral prefrontal cortex, and I think about the modulation in all the connection. So in this model, I have 4 modulation to 4 connection, and I would like to analyze why is the Wiener model in this case, in patient healthy and patient with obesity.
I'm really sorry, you have to skip ahead a little bit. You're past your time. If you want to jump to your results and conclusions, that'd be awesome.
Okay. And so I have here the result to the healthy patient. The healthy patient is the model proposed in the bibliography. The modulation in the 2 area, the dorsolateral prefrontal cortex and the ventromedial prefrontal cortex, regulate the activity in amygdala. This is correct. But the problem here is the patient with obesity, because this connection or the modulation in this connection is not seen, is not upper in this case. So this is maybe the conclusion. The healthy subject describe the bibliography, the regulation of amygdala by ventromedial prefrontal cortex and dorsolateral prefrontal cortex is correct. It's like a bibliography, but in this patient with obese, only I can see the modulation with amygdala to the dorsolateral prefrontal cortex.
Thank you very much. We do have some time for questions. And the first question is here already by Elena. Elena, I'm going to allow you to talk, so you can unmute yourself to ask your question, please. I can ask it on your behalf as well.
Yeah, okay. So I'd like to ask, there is a cases when obese people loses weight, and I think that it's not always permanent, but do you think that there may be some changes in emotional regulation in these cases, and how do emotional regulation
or with obesity is patient with an EMS is higher, is maybe the 14. So it's not possible to change this EMS in this. It's not 1 or 2 kilograms. is patient with obesity is diagnostic. Is not more or less kilogram.
Thank you. In the interest of time, we will have to move on. Please do ask your questions in the chat or the Q&A. I see one of our panelists has a question. You won't be able to use the Q&A probably, but do use the chat. Pablo, if you stick around to answer questions in writing, that'd be great. And thank you again for this really interesting talk. Our next speaker is Xiaolei Xu on behalf of Kongkong Li. She's from the University of Electronic Science and Technology of China and will be talking about disorder and emotional context-specific neurofunctional alterations during inhibitory control in generalized anxiety disorder. The stage is yours. Thank you very much.
Thank you, Ellie. Hello there. This work was mainly contributed by Congkong Liu. However, she just got a surgery, so I'm here on behalf of her to give this talk. Our research was supervised by Professor Benjamin Baker. The title of our talk is Disorder and Emotional Context-Specific Neurofunctional Alterations During Inhibitory Control in Generalized Anxiety Disorder and Major Depressive Disorder. As we know, GAD and MDD are among the most prevalent and devastating disorders, and comorbidity represents normative clinical course. In fact, more than 58% of GAD patients are also comorbid with depression. And on the symptomatic level, both disorders are characterized by several common symptoms like sleep disturbance and concentration difficulty. Since both disorders are highly comorbid and there are so much overlap in symptoms, how can we distinguish them with the help of neuroimaging technology? One recent study used a PAN-EPSI task found that only MDD patients showed alterations in right dorsal anterior insula and its communication with the amygdala, but GAD patients didn't have these alterations. But however, when experiencing physical pain, both disorders has common alterations in the MPFC. One earlier fMRI study using emotional conflict task also found that GAD and MDD showed common alterations in ventral cingulate and right amygdala. Except the empathy and emotion regulation just mentioned, MDD patients was also found deficits in emotional context-specific control, which may represent potential behavioral and neurobiological markers for MDD. However, previous studies in this domain rarely considered the medication, duration of disease, and relapse. All these influences. And also for GAD patients, research on this domain are still scarce and revealed inconsistent findings. So our present study aims to determine disorder-specific alterations in the domain of emotion cognition interactions, recruiting treatment-naive first episode MDD and GAD patients compared to healthy controls by employing a validated effective go-no-go fMRI task. In this study, we not only considered the medication, duration of disease, and relapse, we also excluded those participants who comorbid with PTSD, mania, OCD like this. So the final, based on our exclusion criteria, the final sample size for analysis included 26 GAD 30 for healthy control and 30 MDD. The effective linguistic go-no-go paradigm was a FMI paradigm. It was a mixed event-related block design, which included 12 blocks per round and 2 rounds in total. There are 6 conditions in this study: neutral go, neutral no-go, negative go, negative no-go, positive go, and positive no-go. At the beginning of each block, participants were instructed to press the 1 button if the upcoming words was normal font, otherwise they don't need to press any button. To explore distinct dysregulations in the interaction between emotion and inhibitory control between different categories, the following contrast of interest was subjected to analysis. For negative valence, we use negative versus neutral times no-go versus go. And for positive valence, we use the positive versus neutral times no-go versus go. We use a whole brain level, cluster level FWE correction for our analysis. Here comes the results. This is our demographic information. There were no significant differences between groups in gender, age, and education. But for anxiety, depression, and early life stress scores, there were significant differences in 3 groups as we expected. On the behavioral level, MDD patients exhibited less accuracy to no-go trials. That means they made more errors to inhibit their, to control their finger to press the button. On the neural level, there were no significant differences in positive context. However, for negative context, there was a mean effects of group. And the post-hoc analysis found that MDD patients showed decreased activation in left and right post-central gyrus and middle cingulate. And post-hoc comparison of the 3 groups confirms that MDD showed decreased activation in these regions compared to healthy control and the GAD groups, which indicated that the interaction effect was driven by altered neural activation in the MDD patients. When directly compare GAD and MDD groups, we additionally find that a significant increased recruitment of the left dorsolateral prefrontal cortex in the GAD relative to MDD patients. And to further distinguish the complex interaction effect and to determine alterations during inhibitory control in negative contexts, an additional voxel-wise ANOVA focused on the corresponding contrast, negative no-go versus neutral no-go was further analyzed. And we found the same results as I described before, that MDD patients showed decreased activations in those regions compared to both healthy control and GAD patients. The behavioral analysis I showed before has already revealed that GAD patients exhibited preserved inhibition control in the context of negative emotion and exhibited increased DLPFC recruitment in a direct comparison with MDD patients. This may suggest a neurocompensatory mechanism which may facilitate intact response inhibition in GAD patients. So to explore this hypothesis, we employed a voxel-wise regression model to examine associations between accuracy rates for negative no-go trials with whole brain activation in the corresponding contrast within GAD groups. Results reveal that within the GAD group, better inhibitory control in negative contexts was associated with increasing activation in the left DLPFC. These findings may, however, no such correlation was found in healthy control and MDD patients. So these findings may reflect that GAD patients can maintain inhibitory control in negative contexts via compensatory recruitment of the DLPFC. In conclusion, on behavioral level, MDD patients, but not GAD, exhibited impaired inhibitory control irrespective of emotional contexts. And on the neural level, no alterations were observed during positive contexts, yet specifically MDD patients demonstrated attenuated recruitment of a broad bilateral network during inhibitory control in the negative context. GAD patients exhibited stronger engagement of the left DMPFC called the DMPFC related to MDD patients. And within the GAD group, better inhibitory control in negative contexts was associated with higher recruitment of this reason. So findings from the present study suggest that disorder and emotional context specific behavioral and neurofunctional deficits in inhibitory control in MDD in negative emotional contexts and may point to a depression-specific neuropathology and diagnostic marker. In contrast, GAD patients may maintain intact inhibitory performance while compensatory recruitment of prefrontal regions. This is our GAD and MDD research team leading by Professor Benjamin Becker and Professor Keith Kendrick. Thank you for your attention, and more details can be found from our preprint. Your comments and questions are welcome. Thank you.
Thank you very much, and thank you for your awesome timekeeping as well. We already have a question here in the Q&A panel. Lucy, I will be allowing you to talk, so you can unmute yourself to answer, to ask your question. But if you want me to ask as well, I can do that. Lucy, no, she's able to unmute herself, but I'll take that as a sign for me to read it. Lucy says, nice study. Could the paradigm be applied to the clinical diagnosis or determination of MDD versus GAD?
Thank you for the question, Lucy. Actually, that is our aim to apply our technology to diagnostic in the future, but now I think it's very difficult to apply this this paradigm to clinical diagnosis because this result is from a group level analysis, and but diagnosis is more individual level. So in the future, I think we need more precious design and study to examine the individual differences in neural mechanisms. And this study is very basic research to explore the neural mechanisms in cognitive control.
Thank you very much. For the next question, Rosaba, you'd be able to unmute yourself if you want to ask a question yourself.
Yes. Can you hear me?
Yes, absolutely.
Okay, so thank you so much for your talk. I don't know if I understood correctly, but it seems like in the different group that you use, all the subjects were male. You didn't decide to include the females, and if yes, why?
Thank you for your question. Actually, we involved females. You can see from the table that in total, there are 26 GAD patients. But here we give the number of males. The number of male is 14, so female it is 12. So in this study, we included both male and female, but there's no gender differences between groups.
Thank you very much. I have a question myself as well, which is, it seems that you don't really find any differences between generalized anxiety disorder and healthy controls. So I wonder how you come to the conclusion that your population with generalized anxiety disorder is using compensatory mechanisms.
Oh, that is because also for the behavioral level, we didn't find the differences between GAD and the healthy control. But for the from the neural level, when we directly compare GAD and MDD, actually we compared the post hoc analysis compared healthy control and MDD, healthy control and GAD and GAD and MDD. And we found that although there's no difference between GAD and healthy control, but when we compare GAD and MDD here, we found an increased activation in the DLPFC. So when we compare these two disorders, we found this DLPFC. So we think that the reason that GAD didn't show alterations in both behavioral and neurobiological, maybe it is because the DLPFC has already worked as a compensatory mechanism.
Yeah, I just wonder why you wouldn't just conclude what's way more obvious, that there may not be an impairment in that particular task in people with GAD.
Actually,
because a compensatory activation, you would assume that they would have more activation than the healthy controls, right? Because that's a relevant comparison.
Yes, actually, we didn't find that. It's just compared to MDD. It's not compared to healthy control, because both behavioral and neurological, there's no difference between GAD and MDD, between GAD and healthy control. But compared to MDD, GAD may benefit from the DLPFC.
Thank you very much. That's really interesting.
Thank you.
Moving on to our last speaker, who is, in this piece of notes, Maria Veldhuizen from Mersin University. Maria is going to be giving us a talk entitled "Too Salty, Your Amygdala Thalamus Gain Network May Need to Loosen Up a Bit." Maria, thank you very much, and we'll look forward to your talk.
Thank you so much. And I would also like to thank, I guess, the NeuroMatch algorithm, because I think the symposium is pretty well composed in terms of overlap between the different talks, as you will see, hopefully. So I'm going to talk a little bit about taste perception. Taste acts, or the function of taste is basically to be a gatekeeper of our internal milieu, and basically the perception of different taste qualities functions to help us identify harmful or neutral substances. For example, bitter and sour tastes are unpleasant and could be potentially signifying spoiled or poisonous foods. And then there's the aspect of intensity perception of tastes, which will indicate the quantity of substances. And so why is this important? Well, we think that these aspects of perception are related to food preferences, a diet, and risk for obesity and alcohol consumption. So I'm going to take this example that I think everybody will relate to. So if you go out to dinner, you will almost always find a salt shaker on the table. And that's because people's levels of salt perception just differ hugely. So for example, I may actually send the plate of food back to the kitchen because I think it's too salty to even eat. And another person may say, you know what, it's pretty strong, but it's all right. And another person may add like a whole bunch of extra salt when you're eating the same dish at the same table. I wanted to tell you, well, this is an interesting observation. I think that there are such huge individual differences, and an indication of why that may be came from taste illusions. So you probably know about all kinds of visual illusions. There are also taste illusions. One of those is called thermal taste. And what happens with thermal taste is that if you have pure water in your mouth and you warm the tongue, or you warm that water from 20 degrees to a couple of degrees higher, up to about 37, 40 degrees Celsius, what will happen is that intensity of the sweetness will go from the intensity of the taste will go from no sensation to between weak and moderate sweetness for about 30% of people. So again, there's no taste present at all in this stimulus. And what is also the case is that those 30% of people that perceive thermal taste also perceive in general other tastes as stronger than people that do not perceive thermal tastes. And it has also been observed that in general, if you perceive one taste as stronger than, so say, for example, sweet quality, then you're also more likely to perceive salty, sour, and bitter as stronger in intensity. So all of this suggests that there is some kind of central mechanism. We know already that genetics also play a role, but that there is about 40 to 60% variance that is unexplained, and that is thought to be environmental, but that can include central factors. And so each taste is a good predictor of sensitivity to other tastes, which, knowing that the different receptors in taste have different mechanisms, this indicates that it might be central. So our question was, what might be that central mechanism? And there are some indications for that in the literature from patients who have had their amygdala resectioned for epilepsy. And patients will perceive taste as much stronger compared to after surgery compared to before surgery, or compared to people who haven't had any, who have intact amygdalas, which is kind of counterintuitive, because usually you would think that if you take away a brain region, this would lead to a weaker sensation. So our prediction here was that we will, like in those studies with thermal tasters, we would observe cross taste quality intensity correlations, such that when one person perceives one taste to be strong, they would also perceive the other taste qualities to be strong. And that it would be specifically connectivity between amygdala and earlier taste areas in the taste pathway. And we were focused on insula of thalamus in this case. And that connectivity would correlate with intensity, and that specifically that connectivity would be inhibitory, consistent with that observation that if you don't have an amygdala, your taste intensity perception is stronger. So in this study, we used 4 different taste qualities, so sweet, sour, salty, and bitter, and we presented the same intensity to all participants. And we collected data in 30 healthy weight participants. We first did a session, an event-related session, where we collected taste ratings, 8 ratings of intensity for each of the taste qualities. And then we ran a scan, an fMRI scan, where we presented the tastes in a block design, but people didn't perform any task, so passive sensation. And so this is what a trial looks like in the event-related design, where we collected the ratings. First, they get a taste stimulus, then they swallow, they make their intensity rating, they receive a tasteless rinse, and then they swallow again. While during the scan, we use the block design where we see multiple repeats of one and the same taste, and then at the end of a block there is a rinse. This is what our scan sessions look like, more or less. We have computer-controlled pumps that we use to control the delivery of tastes in the mouth. We have a special mouthpiece that sits comfortably between someone's lips and in a consistent manner drips tastes onto the tongue. Concerning the intensity ratings, here I graphed the intensity ratings for all participants for each of those taste qualities. And on the right side, you'll see the scale that we used with those labels of from barely detectable to very strong. The horizontal arrow bars actually indicate the variance from those 8 different ratings that a participant made, and then each little circle is one participant. And so what you can see is that we have a lot of variation. Some people perceive the tastes to be barely detectable or weak, and then some people very strong. And in general, we see more variance in the bitter taste, which is kind of a known thing because there's a lot of genetic variation in bitter tastes. But we also see that the different taste qualities are not on average perceived differently in intensity. And so, I'm sorry, I skipped through that a little bit fast. We're going to actually use the average of the sucrose, the citric acid, and the sodium chloride, so sweet, sour, and salty, to get our intensity metric here, because quinine showed a little bit more variation and correlates less strongly with the other taste qualities. So you can see that here too, that in the left column, you'll see the correlations between sucrose and the other tastants, where it's not significant for corrected for multiple comparisons if you look at the correlation with quinine, but it's very significant with sodium chloride and citric acid. And this agrees very much with other studies that used larger groups of participants that showed those cross-quality correlations. So when we look at the main effective taste in a voxel-wise analysis, we see sort of our usual suspects of responses in the insula and in the pre and post-central gyrus. I plotted here for the averages to all tastes and all tasteless blocks. And as you can see, almost all participants show a difference in the positive direction for taste blocks. So we have now addressed 2 of our assumptions, that each taste is a good predictor of sensitivity to other tastes, although quinine is not great, and that gustatory cortex robustly responds to the taste stimuli. So now we can go on to our prediction about connectivity. So where is in the brain is response to taste versus tasteless correlated with that intensity metric? So when we do a whole brain correlation, we observe a correlation, a positive correlation in the left amygdala. And we also had some unpredicted areas. I didn't include those in this presentation. And then if we ask the question, where is connectivity correlated with that intensity metric? We used a PPI analysis, psychophysiological whole brain correlation, and we used seed regions that showed a main response to taste versus tasteless, or regions that showed a correlation with that sensitivity metric. And we basically only observed significant correlations when we used that amygdala as a seed region, and we observed bilateral significant connectivity with the left mediodorsal thalamus and pulvinar, or the ventral medial area that is specific to taste, but that's also so small that it's kind of hard to know whether it's really that falls within one voxel pretty much. So then we wanted to take those regions, those 4 thalamus regions and the amygdala, and use a DCM model. And I thank Dr. Matsurana for introducing the DCM models, because I didn't prepare a slide for that. And we specifically wanted we used Bayesian pruning to find out which of those connections show posterior probability above 95, and whether those connections were inhibitory. And we observed that from the amygdala to all of those thalamus areas, that was the case, that there were inhibitory connections that correlated with sensitivity. And if you take that multivariate model, then you see a positive correlation with between the predicted intensity from that and the actual observed intensity. So concluding, that means that we identified a region of amygdala where response to a series of taste is correlated with overall gustatory system responsiveness, that amygdala region drives inhibitory outputs to the gustatory and limbic nuclei of thalamus, and that the dynamics and strength of these inhibitory connections predict individual variation in taste intensity ratings. And so this circuit acts to create gain in the gustatory system to regulate taste intensity perception. I would like to take a moment to thank Dr. Small and Dr. Green, who were involved in this study. And I would like to take questions now. And don't do this in your lab. I can do this in my lab, but nobody else probably can.
Thank you very much. Thank you, Maria. That was fascinating. What an interesting talk. We are taking questions. You can use the Q&A or put your virtual hand up for that. We have a couple of comments here about no questions, but what cool stuff and an interesting experimental setup. So definitely lots of compliments here.
Thank you.
I don't even know where to start, to be honest, to ask questions, because I've never even thought about this. But it's so fascinating, because I think we all have this experience, right? That, well, like for dinner at someone's place and it's either crazy salty or not salty at all. And yeah, Maria, if you're able to share any references to your work, obviously we'll probably find them on your Twitter account as well.
Yeah, so sorry, it's a little bit unclear to see here, but it's in the gray font in the bottom. It's the Journal of Neuroscience. It was published this summer. I can also, let me paste the link to
yeah, please do. Yeah, so we're in a lucky position that we don't actually have to vacate this room right now, because no one's in it afterwards. Thanks to all the speakers. Thank you so much. Like, fascinating, especially yeah, Xiao Li, like I kind of, there's so much overlap with what I do as well. And yeah, like, so interesting to see what you guys put out on anxiety and inhibitory control and things like that. There we go. So we got a link in the chat here to Maria's recent paper. Do check that out. And yeah, for the remaining attendees, I know people are potentially moving on to other rooms. Do follow the speakers on Twitter, find them there, and stay connected. And then, yeah, for me, finally, like, another thank you very much to all of you. That was some brilliant talks, and Maria, I think you're very right. It was great matching happening there. Big applause. And hopefully see you guys around on Twitter and then following talks this week and following Neuromatches. Hope everyone's having a great morning to evening, something in the middle.
Okay, I think it's time to start. So welcome everybody to another session here at Neuromatch 3. So I'm Christoph Metzner. I'll be the host of this session, and welcome also to our speakers for today, Victoria, Ram, Alexis, Nicholas, and Diego. So don't be confused. We only have 3 talks, Nicholas, Alexis, and Diego, because they have an interactive talk. They're all here, so we can have a lively discussion. Yeah, so we have a bit of a mixed bag because we start with a traditional talk, and then we have 2 interactive talks that focuses more on questions. But anyways, if you have questions, please put them in the Q&A window. And also for the interactive talks, please indicate whether you want to come on stage and ask the question yourself or whether I should do that. And yeah, so we'll start with our first talk by Victoria Manyukina from the Higher School of Economics at the National Research University in Moscow. And she will talk about MEG visual gamma oscillations reflecting individual differences in sensory sensitivity. So Victoria, if you would like to share your screen, and the floor.
Good afternoon to everyone. My name is Victoria Manyukina, and I'm going to tell about one of our projects, which is related to magnetoencephalographic visual gamma oscillations and their relation to individual sensory sensitivity. Gamma oscillations rise through interplay between excitatory pyramidal neurons and inhibitory interneurons, and this fact suggests that some characteristics of gamma oscillations may serve as a biomarker of the balance between excitation and inhibition in the brain. The excitation-inhibition balance is an important property of healthy brain that is disturbed in many neurological and psychiatric disorders, such as epilepsy, autism, schizophrenia, migraine, and many others. Therefore, potentially, it could be useful to somehow measure this balance to diagnostics of these conditions, for patient state monitoring, testing of treatment efficacy, some other things. The problem is that the only way to measure excitation inhibition imbalance is invasive. Therefore, it is possible in animals, but not in humans. That is why possibility to use some characteristics of non-invasive biomarkers, such as gamma oscillations, are of importance. The problem is, our problem is that typical characteristics of oscillations, such as power, frequency, seems not work quite good for gamma, because they are quite variable depending on the state of human in different individuals, depending on the type of stimuli. So they are not quite stable, but the balance seems to be stable. And in this respect, some previous researchers of our group suggest that excitation inhibition balance is best reflected not in absolute magnitudes of power or frequency, but in their related changes in individuals. In particular, we observed that excitation inhibition balance in the visual cortex may be reflected in how gamma power is modulated by intensive visual stimulation, such as rapidly moving high contrast gratings. So this measure that we use for estimation of the balance in the visual cortex works quite good. However, the thing that we were interested in is that if indeed this measure reflects the excitation inhibition balance, then individual variation in the balance should be somehow reflected in individual traits, or maybe some kind of perceptive phenomenon. And that is what I wanted to test here in this study. We wanted to, as we talk about the visual cortex, the thing that could be related to the balance in this area is sensory sensitivity to visual stimuli in the environment. So here they wanted to check if our measure of excitation inhibition balance, it is called gamma suppression slope, if it is associated with individual sensory sensitivity. Here we wanted to test it in Russian women, but previously in another work, we tested the same thing in Swedish males. And indeed we found correlation, but here we wanted to replicate these results if they indeed quite okay. So, and another thing they wanted to verify if there is association between other characteristics of gamma, such as gamma power and frequency, with sensory sensitivity, because previously we found that there is no association. So 27 females took part in our experiment. Because they were included in another experiment also, these women were recorded with MEG twice. And additionally, they were asked to fill Russian version of their adult adolescent sensory profile. This profile includes several categories, among which we are most interested in sensory sensitivity, which measures a behavioral response to some sensory stimuli in the environment, stimuli of different modalities, such as visual, auditory, olfactory, and others. So, to induce reliable gamma response, our subjects were presented with large high contrast circular gratings, such as those you see at the picture. And such gratings are known to induce strong gamma oscillations in the primary visual cortex. That is why we use them. These gratings were either static or moving with 3 different velocities: slow, medium, and fast. MEG preprocessing and processing steps were done with the MiniPython toolbox. Source time frequency analysis was conducted with LCV bioinformer. For each vertex of the brain, we estimated normalized spectral response power as stimulus minus baseline divided by baseline. Then, among all the vertices of the brain, we selected 26 vertices with maximal response in the gamma range. We selected these vertices, average spectra of the WEM, and based on this average spectra, estimated gamma power and frequency. I also want to say a few words about gamma suppression slope. This is our measure of excitation inhibition balance in the visual cortex. For this, let's look at the right upper feature. Here is the spectra that we get for each individual. Here are 4 spectra because each corresponds to each type of stimuli. So static, medium, static, slow, medium, and fast. You may note that with the increase of velocity of motion, gamma frequency constantly increases, and this is a very robust phenomenon. However, gamma power is usually bell-shaped, has bell-shaped relation with velocity. So gamma power first increases from static to slow stimuli, but then it attenuates from slow to fast stimuli. Actually, gamma suppression slope measures the efficacy of this attenuation from slow to fast stimuli. It is just a coefficient of linear regression, so the more negative gamma suppression slope, the sharper is this attenuation. Why we're interested in suppression? According to several previous studies, this suppression reflects the capacity of inhibitory neurons to compensate growing excitation, which is induced by increase of motion velocity. So for each individual, we estimated GSS for 2 visits and then averaged them for our purposes. So localization, here you can see group average localization of gamma response power for each of 4 types of stimuli. As you may note, this is occipital region, in particular this is primary visual cortex, so just nothing new here. And you may note again that maximal gamma response may be found at the slow velocity. After slow, for medium and fast velocity, it slows down. So we also observed that there are bell-shaped relations in gamma response power. with the increase of velocity of motions. Here you see spectra, not spectra, just a relation between gamma power and velocity for each of 27 individuals. And you may note that in most of them, gamma power first increases from static to slow stimuli, then decreases from slow to fast. So it is quite a reliable measure. And in most individual derived feature, you see average gamma suppression slope. So it indeed goes down. So what we observed, we again found that indeed individual sensory sensitivity correlates with gamma suppression slope index. So that the more positive gamma suppression slope, it means more the flatter gamma suppression slope, the greatest sensory sensitivity to visual stimuli. Yeah. And at the level of tendency, we also observed that the more positive GSS, so the flatter is GSS, the greater is general sensory sensitivity to multiple modalities: visual, auditory, olfactory, just average combination of them. So in this study, in general, what we observed, we replicated previously found correlation in men with this correlation of our gamma-based measure of excitation inhibition balance and individual variability in sensory sensitivity. The observed modulation of gamma power and frequency by velocity of visual motion can be explained by growing excitatory drive to a visual cortex, which is induced by increase of velocity of motion. And in this respect, this is quite well understood why gamma power increases from slow to static stimuli, because if excitatory drive increases, more neurons are recruited in synchronization, and gamma power grows. But why it starts to desynchronize, why gamma attenuation occurs? This is rather a question. And in one of recent study, Adesnik found that an increase of excitatory drive to the primary visual cortex leads to reduction of excitation inhibition ratio in mice. What does it mean? He observed that when excitatory drive increases to the visual cortex, the level of neural inhibition grows to much higher extent than grows the level of neural excitation. So in general, it even leads to decrease of excitation inhibition ratio. And we could suggest that in our case, this is also the case in our experiment in the primary visual cortex of humans. If indeed we suggest that level of neural inhibition increases with the increase of velocity of motion, to what it lead? It leads to a decrease of activity of excitatory pyramidal neurons. Thus, we will see decrease of gamma response power. We indeed see it. And this will also result to increase of gamma response frequency, because previous studies suggested gamma frequency is mainly regulated by tonic excitability of inhibitory neurons. So the greater is activity of inhibitory neurons, the greater is gamma frequency. And indeed, here we see that gamma frequency grows quite good. And this is a very reliable effect. So next, if indeed this effect occurs in gamma, there is also interesting thing that gamma synchronization enhances information transmission to cortical circuits. This means that attenuation of gamma response, which we observe to very intense, very strengthful stimuli, this attenuation would prevent perception of this harmful information by the brain. Yeah, so this gamma response suppression could serve as a safety mechanism of the brain, which prevents brain from hyperexcitation. And therefore, in such people in which this suppression is less efficient, it means less efficient regulation of inhibitory endurance of excitatory activity by inhibitory endurance, in such people we observe greater sensory sensitivity. So that is the picture as we see. So in general, the study replicates previous results that linked neural gain control measured for visual gamma oscillations to individual differences in sensory sensitivity. And it strengthens the previous proposal of our group that nonlinear behavioral gamma oscillations under varying sensory load may provide information about excitation, inhibition, balance regulation. Here we see it in healthy brain, but possibly it could also work in pathological conditions that are related to excitation, inhibition imbalance, and therefore it could be this result could be potentially useful for some patients that suffer from excitation, inhibition imbalance. So that is generally all what I wanted to say. Thank you very much for attention.
Okay, thank you very much, Victoria. Very nice talk. So we have maybe time for a very short question. So if there are questions, put them in the Q&A window. Okay, so far there is none, so maybe I'll ask one. So basically you showed that this suppression basically with increasing velocity gives you a measure of the inhibition. Do you have any idea of what types of inhibitory neurons are involved? I mean, gamma is probably is generated by probably positive interneurons, but do you know if that suppression is also mediated by this type of interneuron, or can you speculate?
It's difficult to say based on magnetoencephalography which type of interneurons are involved, because actually we see only activity of pyramidal neurons. But in general, this should be interneurons that are related to pyramidal neurons in the primary visual cortex. I don't know exact type of interneuron. So no, I know, I remember. For spiking GABAergic interneurons, so this is specific type of interneurons interrelated to pyramidal ones. Yes. Yes, and they are pyrrolbumyl containing spiking interneurons. That is what I've seen just from previous results, but it's difficult to relate it to human studies because mainly results are related to animal ones. So
Okay, thank you very much, and thanks again for the nice talk. So we move on to our second speaker. So this is going to be an interactive talk. So we basically also rely on the attendees to ask questions. We want to have a discussion. So our next speaker is Ram Kumapari. He is currently working at the Sacro Center for Consciousness with NMC3.0 at the University of Sussex. And he will talk about the neural dynamics of stroboscopic stimulation at different stimulation frequencies. Yeah, Ram, the floor is yours.
Great. So let me get the screen going. So can everyone see it?
Yes, that's fine.
Okay. So, hello everyone. My name is Ram. So today I'm going to be talking about the neural dynamics of stroboscopic stimulation at different frequencies. So you might be asking to yourself, what is stroboscopic stimulation? Essentially, it's basically just sitting in front of a stroboscopic lamp with so basically flickering lights in front of you and with your eyes closed. And this flickering lights essentially causes visual hallucinations. So historically, this effect has been mostly used in a sort of recreational way, was particularly famous in like the 70s and 80s with things just the Dream Machine, people just, and like other stuff that people are just building at home to basically get high on, I guess. So the type of visuals that you see with stroboscopic stimulation varies a lot based on the frequency and the participant itself. So you can expect to see, on the left, you can see form constants or patterns that are what we call simple hallucinations with stroboscopic stimulation. They vary a lot. So in this case, you could see a lot of intense color, kaleidoscopic effects, even complex imagery, such as faces, people, even sometimes feeling like you're floating, etc. People have reported a lot of different things. So in the study that I'm presenting today, we're interested particularly about the neural dynamics aspect of it. So one of the main things that we're looking at is conscious content. So any perceptual experience can basically be considered as conscious content. And one way of studying conscious content is through altered states of consciousness. Traditionally, psychedelically induced altered states of consciousness have been used to study conscious content. One recent example is Shartner et al. 2017, who used different psychedelics such as LSD, ketamine, and psilocybin to get at altered state of consciousness, with the psychedelically induced altered state of consciousness to affect conscious content. He measured conscious content, or the phenomenological diversity of conscious content, using a measure called signal diversity. In this case, he used MEG. Today in our study, we're going to use EEG to use the same technique of signal diversity to compare with baseline dark condition and different frequencies of stimulation. Another key aspect of psychedelically induced ASCs is the significant alpha power drop across such ASCs. So in our current study, we're going to be investigating signal diversity, spectral power profile changes in all the different bands, and the subjective experience of each stimulation frequency. So we used a stroboscope called Lucia. We use it at a free power luminous flux capacity of 5,600 lumens. The participant seated 50 centimeters away. And after each trial, we get the subjective experience measured using the ASCQ questionnaire. So results. These are the results of each of the frequencies that we've tested today in the study. So the top left, the first one, if you can see, is the baseline dark condition, and every single other one is like a spectral difference. I mean, the subjective experience in each frequency. One of the strongest frequencies here, if you can see, is 8 Hertz. That seems to produce the most intense, complex visuals compared to 15 Hertz, which produces a lot of more intense, less complex visuals from what we've seen. Another key result, so to the left here is the Lempel-Ziv complexity, which is our way of measuring signal diversity. So as you can see, baseline to the left and the different frequencies that we've tested. So compared to baseline, almost every frequency tested has an increased signal diversity measure, which is similar to like psychedelically induced ASCs. To the right, we have absolute alpha power across the entire brain. So again, compared to the dark baseline condition, all frequencies tested showcase a very distinct drop in alpha power, similar to that induced by psychedelically induced ASCs. So in the context of the psychedelic research that has existed before, we're presenting stroboscopic stimulation as an alternative to study the human psychedelic state, and as an alternative to psychedelically therapy with psychedelics, especially, how stroboscopically induced ASCs exhibit very similar effects to psychedelically induced ASCs. So that's what I'm really trying to get at here. We also have some other cool results in the study, which I can get to in the interactive bit. But yeah, we're also planning to, revamp everything with a lot more controls, a lot more fun stuff going on right now, which we are planning / working on right now, with like see what signal diversity actually translate to and stuff. And yeah, so feel free to ask questions.
Okay, thank you very much, Ram. Very interesting results. If you want to ask questions, put them in the Q&A window, and maybe also indicate if you want to come up on stage to ask them. Maybe I'll start with a question. So you've shown that this stroboscopic stimulation produces in several ways similar effects to these psychedelic drugs. Do you have any speculation or hypothesis on the mechanisms? So on the underlying causes?
In terms of the physical, the actual like visual hallucinations, the running hypothesis is that they are produced within the junction almost of the V1 and V2. It's specifically to do with how the hypercolumns deal with such visual signals within the visual system that results in these visual hallucinations at all. It's to do with their physical structure is the running theory there. In terms of the actual complex stuff, like seeing people, faces, all that, that's probably happening at a lot later stage with more complex stuff feeding into with the interpretation of what you're seeing. Your brain's, because essentially you're just sitting in front of a white lamp that's flickering off, and when you see colors or any of these patterns, it's all in your head basically. So they're all more like almost an HD, I would describe it as, compared to like any psychedelic induced hallucinations.
Great. So we have another question by Ralph Andrews. Ralph, do you want to come on stage, or should I ask the question? Okay, so maybe I'll just ask a question. So Ralph asked, is the signal diversity and/or alpha drop related to specific therapeutic component of the psychedelic experience, or is it more sensory hallucination related?
It is more related to the neural dynamics aspect of any altered state, psychedelically induced altered states of consciousness, basically. So it's almost like a characteristic feature, like that alpha drop, especially, is almost like a characteristic feature that you would see in almost any psychedelic experience. So I'd say it's the more of the neural dynamics aspect of it. How does that relate to any of the actual hallucinatory effects? We don't really know. I hope that answers the question. So any other questions, guys?
Yes, so there are two more questions. Do you guys, so Sander van Bree, do you want to come on stage? Oh, please ask for me. Okay, okay. Sorry. So Sander asked, what is your baseline in slide 8? Is it no light stimulation at all? If so, alpha is related to light level, so that finding might be trivial. If not, never mind.
So in this case, the baseline is a dark condition. It's basically sitting in a dark room for 4 minutes. All of these conditions are 4 minute long each. So in this case, the baseline is just a dark condition. We are in the revamp study that we're having a lot more effective controls, basically with like aperiodic simulation. All of these in this case are like periodic, so like 10 hertz is like it's consistently 10 hertz. So we're having more other kinds of control conditions in our revamp study for it, but in this study, the baseline is just a dark condition.
Okay, so then the last question by Sandra Sade is, nice talk. Any idea on the differential recruitment of excitation and inhibition at different frequencies?
Could you repeat that?
Any idea on the differential recruitment of excitation and inhibition at the different frequencies?
Okay. If I'm understanding right, I'm assuming you're asking why there's a difference across the frequencies, essentially. So our running hypothesis is that, so we see that almost like a maximal effect within, say, the alpha band frequencies or close to alpha band frequencies. The hypothesis is that it might be related to the natural alpha rhythm of the brain itself. Because like any of these frequencies basically shift the entire natural alpha rhythm to the frequency of entrainment. So if we're looking at like, say, even 15 hertz, it shifts the natural alpha rhythm from like, say, 10 or 11, if you want to say, to all the way up to 15 for the entire stimulation period, basically. One major thing across these frequencies is the difference in like this subjective experience. That, again, since the actual visual hallucinations are a result of structural differences in the visual cortex, there is a lot more factors such as the neurotransmitters, the actual physical structure in the visual system itself. And also in terms of the complex stuff, it depends on like how the individual is with mental imagery, because like the imagery that you see, the complex stuff you see is almost like a halfway point between mental imagery and physically seeing them in front of you.
Okay, thank you. I think we have to move on to our last talk. Thanks again, Ron. Very nice discussion. Also, thanks to everyone that asked the question. So our third and last talk, also an interactive talk, so please feel free to ask questions, well, we have all three authors on stage here. So it's by Nicolas Fuentes, Alexis Garcia, and Diego Mateos. Nicolas and Alexis are at the Universidad Nacional de Córdoba in Argentina, and Diego is at the National Research Council, also in Argentina. And they're going to talk about a study of states of consciousness under the effects of anesthetics through the use of information quantifiers. So the floor is yours.
Thanks, Christoph. Okay. Good morning to everyone. In this work, we analyzed the recording to electrocorticographic signals of monkeys under the effects of anesthetics for the development of biomarkers of consciousness. This research arises due to the importance of knowing the patient's state of consciousness in surgical practice. This work is done by anesthesiologists through a visual inspection, obtaining a qualitative measurement depending on the experience of the professional in charge. This generates the need to develop measures based on fast and open source algorithms. We analyzed 128 channels electrocorticogram signals from the left lateral cortex in 4 monkeys during the condition of awake, anesthetized, and recovery in anesthesia and natural sleep experiments. The anesthetics used were ketamine, ketamine plus medetomidine, and propofol. To analyze this data, we use 4 quantifiers from the information theory. These are permutation entropy, Fisher information, statistical complexity, and Lempel-C permutation complexity. In the analysis of the different states, we observed that all quantifiers turn out to be good measures to indicate the different state of consciousness, with the only exception of Fisher information, where difference between states were not shown. For example, in this case, statistical complexity for metomidine shows difference between anesthetized, recovery, and sleep conditions, without differentiating between open and closed eyes. Not so for propofol, which doesn't show any difference between states. For the rest of anesthetics, we can see difference in certain states, but not in all of them. These results suggest that the behavior of the quantifiers varies depending on used anesthesia.
You're muted. Sorry, we can't hear.
In another analysis, we compare the values of the quantifier for the different anesthesias and sleep conditions. This analysis show a similar behavior of each quantifier for all anesthesias. Permutation entropy is the only measure in which significant difference between anesthesias are seen. There are not significant difference between keramine and keramine plus metomidine, but there are differences between these ones compared to the rest of the anesthetics, which do not present difference between them, including natural sleep. In order to see whether the monkey recovered its initial state after anesthesia, a parallel t-test was performed between awake condition and recovery condition, showing that there are significant differences between both of the states. This means that the monkey doesn't recover its initial state. At least during the time the recovery studies were at least during the time the recovery studies were carried out. To display this analysis, we made bar graph making the difference between both of the conditions. In the minus, awake recovery well. Values above 0 or below 0 can be observed, indicating that the monkey doesn't actually recover its initial state. Finally, we perform an analysis per channel, plotting the values of the quantifiers in the monkey's cortex for the different kinds of experiment, with anesthesia and natural sleep. What can be seen here is that the different brain areas behave in a different way, and despite the fact that the anesthetic act on a global level. For example, when using keramine, we can observe that the permutation entropy shows low values in the occipital lobe, while in the rest of the hemisphere, these values are higher. So, from the analysis carried out, we can conclude that all quantifiers, except Fisher information, allow a clear distinction between the different states of consciousness. The quantifiers show a very similar behavior for all kinds of anesthesia used. There are some brain areas more affected than others beyond the anesthetic act at the global level, The recovery values are not the same as those observed in the initial state, and the quantifiers used in this work may be used in the future as real-time consciousness indexes. Well, thank you very much.
We are very thankful for your attention. If you have any question, we'll be pleased to answer.
Yeah, thank you very much. Yes, if you have questions, put them in the Q&A. Maybe I'll start with a question. If you can go to one of the first or second figure where you compare the results, the different states. Yes, these. So do you have an idea or a hypothesis why these different anesthetics have these different effects? So if you go up one slide, so so what is it, or if you take this figure, what is it that basically sets ketamine apart from the others? Is there like a mechanistic explanation why this would alter the brain dynamics in a different way? Or?
Christopher, would you repeat, please?
Oh, yeah. So, well, it looks like there's a difference between the two on the left, where ketamine is used as an anesthetic, and the other one. So, do you have like a mechanistic explanation for why they are different? So, is there something that suggests ketamine alters the brain dynamics in a different way than propofol or the other one?
Christoph, can I ask for them? Do you have any problem if I ask for them? Because
yeah,
the English is not so good to answer, and they have that problem. No problem if I respond for them?
Yeah, sure.
Okay. The thing is that, yes, we are talking to an expert to understand why these anesthetics change, the value of the anesthetic change. And this is because in the case of, for example, methadone is the anesthetic attack receptor in the brain being a more deeper anesthetic than the others. The thing is that we are not expert in the biology of the brain, and that is why we don't answer this question in the best way. But the idea is that all the different anesthetics actually work in different places of the brain. However, the anesthetic work in a global way in the brain, but as the guys show in the fourth figure, you can see that the anesthetic show changes in sometime in the parietal area, sometime in the occipital area, and that is one of the result of this research, no?
Okay, yeah, great. That's why. So because I have basically a bit of an understanding of what ketamine does, but I don't know what the other two anesthetics do. So
yes,
I was
ketamine
wondering
is very specific. Ketamine, do you know, I'm not an expert, no, but for what the experts say, that ketamine is one of the most heavy, for say something, anesthetic for human. Yes, and that's why it showed that difference between the others anesthetic. The same happened with the propofol.
Okay, yeah, thanks.
Example,
So indeed,
yes, sorry.
Sorry, in the interest of time, we have one more question, and
okay.
So let's go to that, and because we are running a bit out of time. Sorry to interrupt here. Yeah, so our next question, is a bit of a cryptic username, so maybe you just ask the question yourself.
Hi, can you hear me?
Yes, can hear
okay.
you.
Yeah, sorry. So I'm Nadine. I didn't intend to put it. It was an accident that I put that name. So yeah.
No worries, no worries.
Okay. So my question is why you specifically chose these measures, because there are so many out there which you could use to differentiate between different states. Yeah, so, and also could you say a bit more about what these measures measure, because they are quite different. So some of them are more complexity measures, while others are more like pure information theoretic measures. So yeah, can you elaborate a bit more on that?
Okay, thank you.
Okay, well, we use these 4 measures because we wanted to, first, we would like to use information measures. I understand that you can say that Lempel-Seed could be complexity, but come from the information theory, and the 4 measures are from information theory. We use this measure because they are very fast to compute, and the idea of this is to, in the future, try to use in a online time, yes, to use it in a surgery, for example, no? And information measures have the quality to be very fast to compute. The idea of what measure these 4 things are, well, permutation entropy is an idea of entropy, no? The idea of how I know about the system, and it's very useful in the analysis in the EEG. Statistical complexity is a measure of complexity, yes, based on entropy, and another functional called the disequilibrium. Is a measure of this divergence between 2 distribution or probability distribution. It's an idea of complexity about the yes, but idea of Lempel-Seed complexity is a complexity coming from an algorithm complexity, no an statistical complexity. And in Fisher information is a local information, yes, a permutation entropy measure and a global information, but Fisher information measure a local information in a distribution. But in this case, Fisher information didn't give us too much information, because we believe that because these states of consciousness are more global than global in time, no, than local in time.
Okay, thank you, Jacob. I'm sorry I have to interrupt. We are out of time, so we unfortunately we have to go on to the next session. So thanks again, everyone. So I guess if there are still open questions, just contact our speakers offline. Well, yeah, thanks to all presenters, thanks to the audience for a nice and lively discussion. I think when you have to shut the discussion down, that means that it's been going very well, right? So enjoy the rest of the conference, and goodbye, everyone.
Bye, thank you.
Actually, whether deep convolution neural networks explains recurrent processing in the brain. Stage is yours, Jessica.
Thank you. All right, so as mentioned, this is an ongoing project. I actually threw together this presentation in the last hour, so feel free to ask all the questions you have. So the question here is that do deep convolutional neural networks actually capture recurrent processes in the brain? And this is actually part 2 from a 2-parts paper. Part 1 is presented by Noor Seidel. It's on the necessity of recurrent processing during visual object recognition, and that will be Friday, 8:15 UTC. So you could also view that as the same data set. So current deep convolutional neural networks, they're feed-forward and therefore limited as recurrent processing is not incorporated. Because we know in humans, recurrent processing is known to be crucial, especially in perceptual grouping, figure ground segmentation, recognition under challenging conditions, and so forth. And so we also see in the last many years that these networks, they improved in classification accuracy from 2010 all to 2016. And the latest networks have hundreds of layers. So though it's clear that the depth of the network brings about improvement, it's not entirely clear why it does. And so some researchers have proposed that the improvement in deeper feed-forward convolution neural networks actually come from additional operations which resemble an unrolled recurrent neural network. And so we tested this with a family of ResNets, and these ResNets have different depths of 6 layers, 10 to 18 and 34. And in the human side, we manipulated the occurrence of recurrent processing. We did this in 2 ways. One is visual masking, and the second part is image complexity. And this is based on previous work in our lab that shows that based on how complex the image is, you see different amount of recurrent processing in the brain. So the prediction here is that under normal viewing conditions, without visual masking, the deeper models can explain more variance in the brain data. And with visual masking, all models explain the same amount of variance. And this is the experimental paradigm. So first you see a fixation for 500 milliseconds, then a flash of an image. And in the unmasked condition, there will be a blank screen for 500 milliseconds. And then you have 2 seconds to answer what object you see. And it's a choice between 5 categories: bird, cat, fire hydrant, frisbee, suitcase. And in a masked condition, instead of a 500 milliseconds blank, you see 5 subsequent 100 milliseconds masked. And so these are some of our stimuli across a different complexity condition. We have what we call the segmented condition, where this object, the segmentation is done. So there is, you can say there's no background. And then in the low, medium, high, they differ in terms of complexity of the background. So the assumption here is that with more complexity, it's more difficult to segment the object from the background, and therefore recurrent processing is necessary. And so here is the behavioral data. As you see on the x-axis, you see ResNet 6, 10, 18, 34, and then humans masked, unmasked, and masked. And then you see the accuracy. If you look on the human side, on the right side, you see that human unmasked have performance close to ceiling. So it doesn't matter whether the background is segmented or low, medium, high complexity. Humans perform very, very well. Until if you visually mask the object, then you see a difference in performance. So the highest performing condition is still the segmented condition, because that's the easiest condition. And then it comes down to low, medium, and high. And you see this parallel in these neural networks on the left side. So if you look at ResNet 34, you don't see a difference in performance in terms of the different conditions, just like in human unmasked. But when you look down at more shallow networks, ResNet 18, ResNet 10, you see a bigger separation between the easier condition, which is the segmented and low complexity condition, and you see a big drop in performance when the background is medium or high complexity, so more complex and more difficult. And so we will, since now we have confirmed that the more shallow networks sort of behave similarly to humans under mass conditions, and the deeper networks behave more similarly to humans in the unmasked conditions, we'll look inside of the networks and humans to compare the computation. So in humans, we do this with the EEG, the 22 posterior electrodes. We use a representational similarity analysis. And yeah, so, and in networks, we do the same, and for every layer, and we create an RDM per layer. And to do this, we just use a simple non-negative least squares to do the prediction. And so the deeper networks here do explain more variance for now. I only have results for ResNet 6, 10, and 18 because the ResNet 34 is currently still running. So in the unmasked condition, you do see more explained variance for the deeper networks 10 and 18 compared to 6. Although 10 and 18 overlap with each other somewhat. And in the masked condition, they also actually do explain more variance compared to ResNet 6. And so, and in a RIM conclusion is that, yes, deeper networks do, under normal viewing conditions, deeper networks do explain more variance in the data, but with visual masking, all models do not explain the same amount of variance. So that's not confirmed the second prediction. Another way to look into these networks is to correlate what we call the conceptual RDMs with the RDMs of humans and also these deep convolution neural networks. So you see 3 squares. The first one is the difference between segmented and unsegmented background. The second one is between the different object concepts. And the second one is between the different image complexity, and the third one is between the different object concepts. And so if we look at that, we see that the largest correlation still goes with segmentation. So if the object does not have a background, that is the first signal you see in the neurodata, which is correlated with segmenting the object from its background. And but with background complexity and object category, you see that arise much later and much lower in correlation. And we also see that in the networks actually. With ResNet 6, you see, and ResNet 10, in the early layers, you see a large correlation with segmentation and complexity, and a very gradual rise in object category in green towards from the middle towards the end. And even in the deeper networks, you also see the same thing, which is that the correlation from the early layers, they're mostly correlated with segmentation and complexity instead of object category, because that gradually arises later. And that's all I have. And some next steps is, I guess I'll go over that later. So that's it. Thank you.
Thank you, Jessica. Just slightly over 5 minutes, but I'm well aware that it is really hard to keep in 5 minutes. In the question and answer session, we have one question from Nadina Dijkstra. Sorry about the pronunciation. She says, this is very interesting, and she's surprised you don't see differences between the conditions in the shallowest network. For example, ResNet 6. Do you have any idea why this is the case?
Yeah, so I'm thinking that you are referring to this classification accuracy. And so the thing is that with the shallow networks, so I've also done a sort of based on the different image complexity. And so with deeper networks, you do see more activation on the object itself. So more activation in the pixels related directly of the cat or the fire hydrant compared to the background. With like ResNet 34 or ResNet 18. But with ResNet 6, it doesn't seem that it could locate where the object is. And it seems to put similar weight or similar importance on the background and also on the object. So I guess the simple answer is that it doesn't know where the object is.
Okay, thank you. Is there any other questions? I think I can see there are other questions. And for the next questions, Farahat is asking, do these networks match with the respective effective receptive fields?
Do they match the receptive fields? Is that the question?
Yes. I can read it again. Do these networks match with respect to their effective receptive field?
I'm not sure if I understand the question. If they match with the effective receptive fields, so I'm not sure I have a good answer for that.
Yeah, please ask your questions in a different way to Jessica, because we have some time for one more questions. If you just rephrase your questions, we are happy to ask it again. And Nadina is saying again for the first question asked by Nadina, and she is saying that if I understood it correctly, you now only did the RDM for the unmasked data. Did you also do it for the masked data? Would you expect neural activity to correlate mostly with early DNN layers for the masked data, or any other differences?
Yeah, so that's a nice question. I couldn't fit the results here. I actually also did it for the masked data and unmasked data. The interesting here is that although you see a difference between these explained variants over here for unmasked and masked, if you look at this graph over here, for the masked data, it's approximately similar, to tell you the truth. And you still see a large correlation for segmentation and very, very small correlation for background complexity and object category. Yeah. Does that answer your question?
I think so. And for the questions, I'm rephrasing the questions again. Do these networks match with respect to the field of receptive field of the last layer?
So okay, you're asking if do they correlate well with the fully connected layer? That's how I'm going to interpret the question. So all these regressions that were done with non-negative least squares, they were only done with convolution layers, and I also tried another model where I also included the pooling layer, or the last layer right before the fully connected layer, and it did not make a difference actually in terms of how well it explains brain data.
Okay, I hope these answer the questions, and we are in 15 minutes now. And if you have more questions, Jessica will be around to answer them throughout the question and answer panel. Please post them in the question and answer panel, and Jessica will answer them by typing the answers. And now we will move to our next speaker. Thank you, Jessica, again for the beautiful talk.
Yeah, thank you.
And for our next speaker, Parisa Abedi Kozani, she's doing her postdoc at York University in Canada. And her talk is titled Building a Convolutional Neural Network to Study the Combination of Allocentric and Egocentric Information. Stage is yours.
Yep. Okay. Let me share my screen. And what also I want to add. Good morning, everyone. Thanks for attending this session. In this part, I am going to quickly explain our project, which is at very early stages in understanding how allocentric and egocentric information are combined in the brain. So when planning to reach to a visual target, one can code the spatial position of the object with regard to body parts, for example, here with regard to the hand, or can code the information with regard to the other objects in the surrounding area, which the second one is called allocentric coding and the first one is called egocentric coding. And many studies, both behavioral and neurophysiology studies, show that actually this allocentric and egocentric information are combined for reaching purposes. For example, one of the recent studies by my colleague in Dr. Crawford's lab, when they recorded the activity in the frontal eye field of monkeys, they showed that the motor codes, in the motor code, there is an embedded allocentric information. So more specifically, what they did, they trained monkeys to reach to remembered position of targets in the presence of allocentric landmarks. So let's go through the task very quickly together. So here in the first image, the monkeys are trained to fixate on the orange dot, and the landmarks are presented as two crossing lines. And after a fixation period, the target appears as the white dot here, close to the landmarks, and then disappears after 100 milliseconds. And after the delay period, a mask appears, so both landmark and target, everything disappears. And after random delay, now the landmark again appears on the screen. And it could be in two different conditions, either in the same position as previous image or as a shifted position in different direction. And what they found, and then the monkey, of course, was trained to reach to the remembered position of the target when the fixation was disappeared. So what they found is that similar to human behavioral studies, also the gaze position, the final gaze position of the monkey was shifted toward the shifted landmark position, which is showing the influence of the landmarks. And this shift was around 30%. And when they look at the single neuron activity, they found that early visual responses were coding mostly information in egocentric frame, here eye-centered coordinate. But when they look at the later responses or motor responses, what they found is that those codes showing, again, mainly coding in egocentric, but within that coding was embedded about 30% shift toward the allocentric coding. And this was an indicator of the presence of allocentric coding also in the brain for reaching purposes. However, while all these studies showing the effect of allocentric information, it's not clear how and where in the network this combination might happen. So this is the goal of this study. And previously for egocentric framework, neural networks were a very useful tool to provide insights of like how these processes can happen. And our goal in this project is to use the same methodology. So there are several challenges that we first need to tackle. The first thing, so as I said, we are using neural network, but before I can go into the architecture, I'm going to quickly explain what type of signal we are feeding to the network and expecting from the network to generate. So since our goal is to replicate the result by my colleague, similarly, we will provide a visual input, here images with the landmark and target position. And then if you remember, we had two different images. One was landmark and target, and one only with the landmark. And in the first attempt of our project, we are only interested in the special coding. And so we remove the temporal aspect of this coding. And for that reason, currently, we are just stacking up the two images together. We hope to add recurrency or recurrent connection later on to our network. But so this would be our visual input. And in order to dissociate between egocentric and allocentric, then we varied the initial gaze position, and accordingly, we varied our visual input to replicate the retinal projection as an input for our network. So these are the two inputs. And finally, to make it more neurophysiologically feasible, we expect our network to generate the motor output similar to the FEF motor responses that my colleague recorded. So this would be a population code of the final gaze position. To very quickly go through the architecture that we are proposing here. So since we have images here, we are using convolutional neural networks to extract the information from images. And what we are doing here, we are just trying to replicate early visual responses. So we are aiming to use physiologically feasible. So here we use only two convolution layers. We use Gabor filters, different orientation, to kind of model the simple cells. And then we have rectification, normalization, and spatial pooling, and again Gabor filters. But before flattening the image to fit it to the final part of our network, then we included a feature pooling. And this part is the only part that we train in our convolutional network. And this part is the part that we allow our network to be selective and choose which feature map among all the feature map that was generated using our Gabor filters it wants to combine and generate the abstract feature maps of the visual input. And then we use this initial gaze position with the extracted information from the images, fit that to a two-layer fully connected network in order to generate the final mirror output. And this part is the part that we are planning to analyze to compare with the processes that is happening in the brain. Here, what I did, and so I just finished designing the network. Here, this is a very simple or very early result of the network performance. And what I did is that I got the population code and used a linear decoder to transform that into 2D gaze positions. And in the y-axis, I'm plotting the predicted gaze position from my network, and the x-axis now I'm plotting the desired gaze position based on the simulation data I generated, again, based on the behaviors. And you can see that the network is trainable and it can predict. And the next step for our network is now to analyze and go inside the individual units and compare them with the behavior of the network. And since my time is up, I would like to thank my co-authors and my lab member and the funding agencies. And now I'm open to questions for anyone who is interested to go deeper into the architecture of the network. Thank you.
Thank you, Parisa. And currently, we don't have any questions in the question and answer panel. And please post your questions in the panel so that we can go through it. We have actually time for plenty of questions. And could you please give us, could you please explore your network a little bit more, because it seems like you just go through it very shortly.
Yeah, so actually the network, the kind of maybe the main part of the network. So the goal of this is to create a model which is like replicating the brain as much as it's possible. So it's not like about like training just a convolution network to do a classification. So what we did is here I'm just giving a quick overview of the convolution part. Again, just a big picture. And so if this is my if this is the heat map of my input, this would be the final output of my network. It's kind of resized and creates a Gaussian like kind of activity of all of these pixels. And you can see that around the allocentric part and the target part, now I have higher activation. So one of the main part or challenges of this project was that to train it or to create the architecture that is more physiologically understandable. So if I go through it, so here, what I'm showing is that how I'm performing the convolution, rectification, normalization, and pooling. And as I said, I'm just simply using some Gabor filters. These are all predefined. And so Gabor filters are like they can just, here in this example, extract the orientation of the line for us. So if I have the example of like only having 2 lines here, so the first one would like, let's say, extract the vertical and horizontal. And then with this repeated filtering that we have here, and has been shown that this can be a process that is happening in the early visual areas, then I can create like feature maps with the combination of different line orientation. And this is the part that we allow our network now to choose. For example, if we have only like horizontal and vertical crosses or any other direction. So this is the part that our network will be trained to create the abstract features. And why we are selecting this and why this is important is that if you think about coordinate transformation, is that we have many options. And the issue in the brain is that we don't know what to look for and we don't know where to look for it. And by creating such a thing, what we are planning to see is that at what stage this information is coded. Is it like this allocentric information present all over the network, and at some point do they use it or not using it? So I didn't have the time to go through some of the results, but when we looked at fully connected layers, what we did, if we just extract here, if I go through like the weights, and what I can see is that from the early ones on my fully connected layer, I can see the presence of the crosses into the weights. So I can see that the network is in a distributed fashion, trying to extract this crosses position with regard to the target position and pass it to the other layers. So the next step for us would be for sure to just try now to simulate a situation that no allocentric information is coded and see if again this would be persistent in the network or not.
Thank you. One of our panelists audience is asking, Akel Labash is asking, was the images introduced to the network as RGB or binary?
No, they are binary. Right now they are binary, so they are not RGB.
Do you think it will make big differences?
If I introduce them as RGB? No, I don't think so. The plan is, interestingly, no one studied that as a frame of like allocentric coding. So this is the first step. But I know that in naturalistic images, the first image I showed you, that when it was like actual object and we have data, human data. And the next step is for our project is actually to then train them on naturalistic objects. And so that would be the next step for our project. But as I said, the main goal for us first now is that when we train the monkeys, we want to see what signal we should look for in the brain, what type of coordinate phrase we should expect to find in the brain, and which areas are the most probable to code those information for us.
That sounds very interesting. And thank you very much for your talk. Again, if you have any more questions for Parisa, please just type them in the question and answer panel. Parisa will be around to answer them. And now we will move on to our third speaker. And to our third speaker and the final speaker is Nil Shahdosh. Nil is going to present her graduate work from in which she worked under the supervisions of Dr. Mohamed Reza Abolghasemi Dehakani and Babak Najdar Arabi in Machine Learning and Computational Modeling Lab in University of Tehran. Her talk is titled Comparison of Cortical Object Representation with Neuroscience-Inspired Deep Neural Network. Stage is yours, Neil.
Hello, everybody. Is my voice clear?
Yeah, it is.
Thank you. Hello, everybody. My name is Nilüfer Şahduz. I'm going to give you a keynote about the comparison of cortical object representation with neuroscience-inspired deep neural networks. Okay, let's start the story. There was a paper published in Journal Vision written by my professor. And in this paper, they wanted to study the accuracy of people in naming the identity of 110 different images, line drawing images on white background. And for these images, there were 3 different kind of versions of these images. The complete version had edges and vertices, and the vertex version did have vertices but did not have edges, and the edge version did not have vertices but had edges. And in order just to save the time, I didn't put the paradigm of this task because it is not done by me, and but the results are here. And as you see, the accuracy of people on complete version of images are the highest, and it is worth mentioning that each image was shown about 250 milliseconds to each subject, and subjects were some teenagers. And then what was really interesting for us was this part. In this part, it shows that people tend to have higher performance for identifying images that had vertices. I mean, these images, and the performance was significantly high for vertex images in compared to edge images. It shows that for human beings, the vertices were much more important than the edges. And we wanted to explore, to investigate these results. I mean, we wanted to compare these results with deep neural networks. And at first, by going through the literature, we expected that the deep neural networks would have higher performance on images that had edges and didn't have vertices. I mean, these kind of images. So in order to do this, I had to create my data set. So what I did, I used a Python augmented pipeline function, and I just applied a random combination of rotation, scaling, flipping, and distortion transformations on each and every images of these three versions of my data set. And finally, I created a data set which had 110 classes of different identities, and each class had 30 images. And then I had to design a network, and I designed based on some brain-inspired networks like CoreNet, and I created 4-layer convolutional networks followed by 2 fully connected layers. And here are the characteristics of my network and the training of my network. And then what I did, I trained the network on complete version of my images, and then saved the network, and then tested it on vertex and edge version of my images. And unlike what we expected, as you see, the pattern of categorization, I mean, the pattern of accuracy was exactly as human, and it was quite interesting for us. And after investigating a lot throughout the data, and in this difference, I mean, difference in accuracy was not related to the statistics of the data set, but was related to the representation of my data set in my network. So what I basically did, I just created the RDM, I mean, representational dissimilarity matrix, for all the images in all the categories for all the 4 layers of my network for all 3 versions of my data set. By looking at this, we cannot understand that much. I mean, we cannot get to a lot of information, but here is the gist of this part, which is the RDM of RDM, or second order RDM, or the Kendall correlation between all the RDMs shown here. And I mean, the more blue and a cell is, it shows the more similarity between different layers of the network. And if you take a look at here, we will see that the difference between 2 results are due to the difference between the representations of images. I mean, the representation of images of vertex data set between vertex data set and complete data set are much higher than the representation of edge data set and complete data set. And especially for the first 2 layers, which was a really interesting concept that we understood. And here is another representation, a visualization of the result, which the blue curve is the Kendall correlation between the complete data set and vertex images, and the green part is the Kendall correlation. The red part is a Kendall correlation between edge images and complete images. And as you see, for all the layers of the network, the Kendall correlation between complete images and vertex images are higher than the complete, higher than edge version of images. And after investigating a lot, we thought that this difference between paying attention to the vertices of the images and the edges of the images, it is due to the inductive bias of deep neural networks. It shows that deep neural networks have good inductive bias toward vertices, and it will pay more attention to the vertices rather than edges, whereas in classical machine vision, the attention was more on the edges of images rather than vertices. And to be more specific, inductive bias is some set of assumptions about the target function and the input data that is injected to a network. And last, I wanted to thank you all for listening to my keynote, and I'm open to answer to your questions.
Thank you, Neil, for your nice work and for your nice, beautiful talk. And one question is asking for your data set, is it available publicly? I think he's asking for.
No, it is not available publicly. It is created by us, and I think that it won't be publicly available. But I think that there are some kind of images created by Google, I think, and there are some line drawing images, maybe.
Okay.
I think it is called Sketch Dataset.
Good to know. And Hojat Azimi is asking, why did you choose 4 layers?
Actually, this 4-layer represents four stages of the brain, and it is more inspired by the coordinate Z of coordinate Z network. The first layer represents the V1 part of the brain, the second layer represents the V2, and the third layer represents V4, and the last layer represents the IT part of the stages of human vision.
Okay, one quick question. Do you think if you use CNN, you will see these differences, edge and the vertex, what you're presenting here?
Could you please repeat your question? CNN?
Currently, I think you are using DNN, right? Deep neural networks.
It is a CNN. Yeah, it is a CNN.
It is CNN, okay.
Yeah, it is a CNN, yeah. What I want to do for the future work, I want to do the whole experiment on LowNet, AlexNet, and CoreNet Z, and even CoreNet R, because CoreNet R has some recurrent parts, which is more similar to human brain. Or maybe I can just design a new network with recurrent parts.
I think you have very nice and clear hypothesis to test on different models and then see how they are going to behave. Yeah, that looks good. Is there any other questions from our panelists to each other?
Well, I'm curious if you are going to test it on brain data to see.
Yes, yes. I have a brain data from a monkey, which the monkey was shown passively 3 different, all of these images, but they have just categorized, they haven't categorized the identity of these images, but they have categorized the version of these images. For example, if you take a look at here, in this study, in this test, they have, for example, they haven't written that a spoon was shown to a monkey, but it was written that an image was shown to the monkey, which is from the category of complete images or vertex images or edge images. So I can easily compare the data, the representational, the representation of all of these 3 versions of my data set with my network. Yeah, from the monkey brain with my network. And I think that I have data, spiking neural data for, collected from 4 neurons of a monkey brain.
I think one further steps can be, if you have a background, when you're identifying the object from the background, are you using edges or vertex, and how the models are performing to detect that? Because currently you're just using the object itself, right? But that's not real in the real life. We are always segregating these objects from each other. Yeah, I think you have a very beautiful direction to go.
Yeah. I will check all of the hypotheses. There are lots of hypotheses. I can create a lot of, many different data sets of this data set, and I can check everything on the network or any other network.
I think at this stage, I would like to thank all our speakers, Jessica, Parisa, and Neil, for your beautiful talks, times, ideas, and discussions. And many thanks to our audience for being very active and asking instructive questions. I should also thank to Zeynep for giving us technical help at backend, although you don't see it. It wasn't possible without her help. I hope you enjoyed the session and will enjoy the rest of the NeuroMatch conference and following the sessions. Thank you very much, everyone.
And I primarily work on this in rats. And today I'm going to tell you about 2 main stories. The first is regarding our first attempt at looking at kinship behavior in the brain. And I'll tell you about what we found in the lateral septum. We found a role for the lateral septum and some interesting cellular organization of responsive cells in the lateral septum. And in the second part, I'll tell you about some ongoing work that we're now pursuing in regards to a role for the lateral septum opioids in maternal attachment behavior. So kinship in biology has a very long history. This starts with Darwin, who talked about the concept of kin selection in the origin of the species while looking at colonies of honeybees. But the biologist who really brought the idea of kinship or kin selection to the forefront is Hamilton, who proposed the theory of inclusive fitness or kin selection theory and Hamilton's rule. So this is in very short summary. The idea that altruistic acts would be more readily performed towards genetic relatives rather than strangers. And this is actually a form of self-preservation of one's genes. So if you look around nature, you find that almost all organisms show kinship behavior, even single-celled organisms. I don't really have time to go into any specific details of examples, but hopefully you can read about this soon in our review that's now actually under review about kinship in biology and how neuroscience can approach this idea. So kinship we see throughout almost all organisms, but maybe the most peculiar example of kinship is in our own species in humans. And this is exemplified in the word nepotism, which is a really extreme form of kinship. It's the practice of those with power or influence of favoring relatives, especially by giving them jobs.
So there's so many examples of nepotism and kinship in humans that I can almost give a different example for every talk. So in this talk, I'm going to give you a UK example because that's where I am now. And this is the example of Robert Bob Cecil, who appointed his nephew as the Chief Secretary of Ireland in 1887. And this is actually the origin of the term Bob's your uncle, which I just learned about very recently upon moving to the UK. Somebody said this to me, and I learned from Wikipedia that actually this is related to kinship behavior in humans. And I like to give everyday examples that we can all imagine for ourselves as well in introducing this topic. So when I see this picture of a family having a good time in the park blowing bubbles, I think it's really cute. And well, I think it's nice, but I don't really feel that much about it. But if I see this picture of my family when we were all in New York one day, on a day it was too sunny, we had to walk around with umbrellas because we couldn't bear the heat. A lot of emotions come flowing in for me, and memories of each of these people. It means a lot to me. And I would think that if you saw a picture of your family, you would also feel something very different than either of these two pictures. So I hope you believe me when I say that kinship is really a driving force of behavior in nearly all organisms. But how do we study kinship in the brain? We really need a behavioral test to assess kinship preferences in animals to begin with. So we looked for this in the literature, and we came across the work of Peter Hepper, who was a really fantastic behavioral scientist who's published a lot of papers in his PhD actually about how to assess kinship in rodents. So in assessing sibling preference behavior, Hepper came up with this test, this apparatus where he put one family of siblings in one goal box, another family of siblings in another goal box, and he took each rat and put them in this start tube and looked at which direction they turned. So Hepper found that from birth, rats prefer their siblings. They turn towards their siblings, and up until about day 15 of age, then they start preferring non-siblings, or they're avoiding their siblings. It's up to your interpretation really at this point. So we found this to be a very interesting behavior and a very interesting way to study kinship. So we tried to see if we could replicate this behavior that Hepper demonstrated. So I built our own kinship apparatus, which was very, very similar to what Hepper did, where I made two goal boxes with computer fans blowing odors into the middle and out of this exhaust. And I tested the rats from birth for their preferences as Hepper did. And it turns out that I was able to replicate his results quite easily. So rats from birth, they overall preferred to turn towards their siblings. And then around 15 days of age, as Hepper saw also, the rats then turned towards their non-siblings. So these are quite different developmental phenotypes we found. So we divided the data up into these two groups of sibling preferring age group and non-sibling preferring age group. And you could see that the behavior is quite different across the population. But where do we look for kinship in the brain? So we were actually inspired primarily by a human imaging study done by George Moll and Roland Zahn, who are one of the few human neuroscientists who have really looked at the representation of kinship in the brain. And in their study, they found that there was the activation of the septal hypothalamic region of the brain in humans with affiliative kinship stimuli. So we decided to test the hypothesis that this sibling preference behavior might have something to do with the lateral septum. So we made lesions of the lateral septum first in the sibling preferring age group of rats, and we looked at then their behavioral preferences. So on the left here, this is the behavior with no lesion, and on the right, this is the lesioned animals. You can see that after a lateral septal lesion, that the rats' behavior goes to chance levels. They no longer prefer their siblings with a lateral septal lesion. So we next asked whether the septum might have something to do with this non-sibling preferring behavior as well in the older animals. So again, we performed lateral septal lesions, and we also performed cortical lesions of the cortex overlying the lateral septum. And what we saw was that with the lateral septal lesion, that the behavior went to chance levels. And then with the cortical lesion, the behavior basically looked like a non-lesioned animals. So this told us that the septum seems to be an important area of the brain for this sibling preference behavior. But we next wanted to find out if we could understand how this works at the cellular level. So we asked, what are the cellular correlates of kinship in the lateral septum? And the way that we address this is by performing single cell in vivo recordings and presenting to the pups, either in the young sibling preferring age group or in the older age, non-sibling preferring age group. We presented them with kin and non-kin stimuli. This was vocalizations as well as odors. So we presented either the pup's siblings, non-siblings, as well as their mother and another mother. And we looked at the responses of the lateral septal cells to these stimuli. I don't have time to go extensively into this data set. The paper is published though, so you're welcome to have a look. I'll tell you though that the responses to odors and calls in the lateral septum, as I said, they're complex. So this is just an example of a sibling odor response and a cell that responded to sibling odors and non-sibling odors. And in this case, this cell distinguished kinship by responding more to the non-sibling odors. And this is in the older non-sibling preferring age group. But as I mentioned, the data set is quite complex. We saw neurons that responded to calls of siblings, calls of non-siblings, siblings and non-siblings odors, as well as mothers and non-mothers. But there's two basic messages that I want to convey to you about what we found. And the first is that we found a developmental profile of sibling odor responsive cells, where the highest proportion of sibling odor responsive cells were in the youngest age group that we recorded from. And this declined with developmental days of age. We didn't see this developmental profile for the non-sibling odor responsive cells, but we found this to be very interesting because of the parallel between the behavior that we saw in the beginning and the proportion of cells that responded to the sibling odors. The next interesting thing that we found in the data set was that when we plotted the locations of the cells that were responsive to each stimuli, we found that there was an organization in the lateral septum. So in blue, in light blue and dark blue, this is sibling odor and mother odor. We found that they're deeper in the lateral septum than when we compared this with non-sibling odor and non-mother odor responsive cells. We were able to recover the morphology of many of these cells, and you could see that they're also show quite different morphologies. Depending on where they are in the lateral septum, we weren't able to make any conclusive statements about morphological differences. But what we were able to say is that there seemed to be a topography of kinship where the kin cells are deeper in the lateral septum and the non-kin cells are more superficial. We compared this across groups and found that there was a statistical difference between kin and non-kin in terms of the sibling and mother odor responsive cells. And overall, we found this to be a quite important aspect of the cellular organization of cellular responses in the lateral septum. So we decided to give it a name after Nepos and Topos. We call this nepotopy. So with this study, we suggest that nepotopy of the lateral septum, or this organization of cellular responses to kinship, may be an organizing principle for kinship behavior. And what I've been up to since this study is to, in my own lab, as Andre mentioned, which I'm now starting in Edinburgh, we're now looking at to see if we can find cellular correlates of learning, and if we can try to understand the overall circuitry of kinship. And this is kind of our hypotheses are centered around the lateral septum, but not restricted to the lateral septum. So I'm going to tell you next about an ongoing study that we now have partially going here. And this is looking at the role of lateral septum opioids in maternal attachment behavior. And specifically, we're looking at a circuit between the hypothalamus and the lateral septum. And I've worked on this, on the data that I'll show you with Gamze Guni and Lorena Jimenez Sanchez, who was my first PhD student now in Edinburgh. So the role of endogenous opioids in social behavior and attachment behavior is quite big. I don't have time to cover all of the background today, but I will just give you a really nice example of this. And this is a demonstration of the role of endogenous opioids in maternal attachment behavior in newborn lambs. So newborn lambs, if you give them a choice between spending time in a pen where their mother is or another mother is, from birth, they want to spend time with their mother. However, in this study, they showed that if they injected naltrexone, which is an opioid receptor blocker, if they injected this right after birth, that the newborn lambs no longer prefer their mother. You also see interesting effects of attachment behavior in mice, primates, voles, in many different species. So the endogenous opioid system seems to be quite important for social behavior, in particular, we think for attachment to the mother. So we were interested in the possibility that opioid action in the lateral septum might mediate maternal attachment behavior. And the reason why we thought that this might be a good hypothesis is the anatomical expression of enkephalins from the hypothalamus to the lateral septum. So this study in 1995 nicely showed it, and then we tried to replicate it actually in the brain of newborns. So this is an antibody against LU-MET enkephalin. And basically upon seeing the septum light up in this way, this really told me that this might be something important that we need to look at. So to test this hypothesis, what we did is then we looked back in the literature of what we know about, or to give us some indications of how we can assess kinship in terms of maternal attachment in rodents. And then we came, of course, across the work of Hepper again, who developed a really nice behavioral test of maternal preferences. In his study, he tested the preference for the amniotic fluid of mothers and non-mothers. So we modified this test to collect the odor of the mother and another mother, not the amniotic fluid, but of the mother's skin. And we also recorded the ultrasonic vocalizations of the pups during this preference behavior. So we used this, we established this behavioral paradigm, and then we next wanted to see if we could probe the role of the lateral septum opioids in this behavior. So the experiment was basically that we monitored the mothers for birth for 4 days, and within 9 hours of the birth, we then RFID chipped all the animals, and we injected naltrexone, as I showed. They injected in the newborn lambs to block endogenous opioid activity. And then we tested the behavior for 4 days. So naltrexone is a long-acting opioid antagonist. It lasts up to 48 hours. So we assessed for 2 days, and 2 days within drug and 2 days after. And what Gamze found after we injected naltrexone is that naltrexone injected in the lateral septum made this behavior go to chance levels. And on the left, this is the behavior of the rats when ringer is injected in the lateral septum. So they prefer their mothers normally if it's just ringer injected. However, when naltrexone is injected in the lateral septum, they no longer show this preference for their mother's odor. We also saw that the duration of calls and the frequency of calls was changed with naltrexone in the lateral septum. Right, so this is showing the duration, but we also have since seen that there's a change in the frequency of calls. So we are now looking into what's the role of, what are the cellular mechanisms again of this possible opioid action in the lateral septum. So we've just recently collected a data set of recordings from neonatal septal slices. So we patch cells in the lateral septum and we're stimulating synaptically. And we assessed the baseline synaptic responses and then the responses in the presence of opioid receptor agonists and vehicle. And what we've seen in a subset of cells is that there's a potentiation of the synaptic response with this opioid receptor agonist. And so now we're asking ourselves if this might be a cellular correlate of attachment. And this work is to be continued. I hope to tell you more about it soon when we find out more. So overall, I've told you that kinship drives natural social behaviors. I've told you that we found a cellular organization of family in the brain. And I think that this opens a new avenue of research for the study of kinship from a neuroscience perspective. Okay, so finally, I'd like to acknowledge all of the places and people involved. First is the University of Edinburgh and the Simons Initiative for the Developing Brain, who allowed me to start my lab recently. Lorena Jimenez Sanchez, who is joining in the opioid project and is the first brave PhD student to join me. We're also recruiting. We have now a PhD position open and probably more positions available soon. I'd like to thank the Brecht Lab and Michael Brecht, where I started the kinship work, and Gamze Guni, who worked with me on collecting the preliminary opioid attachment data. I'd also like to thank the MBL, the Neural Systems and Behavior course, which has really been an inspiration for me in the development of my scientific ideas, I would say, over the years, as well as the Grass Fellowship, where I participated in, which I virtually participated in over the summer and hope to one day actually participate in person. And I'd like to thank the funding sources in each of these respective places. And thank you for listening.
Wonderful, thank you. So there are some questions in the chat that I'll like to ask first, and then I'll use my privilege to ask a few. So that was a wonderful talk, Anne. Can everyone hear me? Great. So early on, Hadi Mota Mehdi, I'm sorry if I mispronounced your name, asked, I wonder why would be the reason that after day 15 that the pups preferred the non-siblings? What's the switch in the behavior signify? Can you speculate on that?
Yeah, that's a very interesting question. And that's one question that we would really like to address in the future. And there are a lot of different ideas. One indication, I think, could be the onset of sensory modalities at that time. So this day 15 is around the timing of hearing onset as well as eye opening. So it might have something to do with the introduction of these different sensory inputs that's then changing the behavior. The other thing is that, so weaning age of these pups is around 21 normally, but around 15, they're starting to become a bit more independent. So it might just be a matter of not relying anymore on the family and gaining their independence in a way. And another aspect of this is the development of fear. So if I remember correctly, there's quite a developmental switch as well in the amygdala. And we're wondering actually long-term how the amygdala and how fear might interact with this kind of kinship behavior. So it might be that animals are no longer fearing for their survival, and this switch is showing a form of self-reliance. So I could go on and give you possibilities of what might be happening, but this is something that we really have to experimentally probe to find out. Andre, I think you're muted.
Thank you. There's a siren here going by. So,
okay.
Thank you. And if any of the anyone in the audience would like to ask a question directly, please raise your hand and come on stage. I'd like to ask this. So the kinship assay, as we were looking at it, I imagine that was a one trial assay. Each pup was given the choice one time. Is that correct? And if it is, what happens if you have repeated trials within the same occasion? Do they do seeking behavior, or are they consistent in their choice?
Yeah, I didn't try that. So I did basically what Heifer did, which is to test them one pup. Sometimes we did a morning and afternoon test, but it was kind of one pup per day to assess the developmental profile as well. So we didn't do this repeatedly, but that's a good question, like to know if the initial response might be something different than once they're more familiar with it. Yeah, so I don't know. That's something that we can definitely look into, though.
Thank you. So Priscilla, why don't you come on and ask your question? Priscilla Ambrosi.
Yes, can you hear me?
We can.
All right. First of all, this was remarkable, wonderful, really. And one of my questions is about topographical organization of the kin versus non-kin responding cells. Yeah. I was wondering if that has anything to do with birth order of the cells. Could maybe the later born lateral septum cells be more dorsal and respond to non-kin stimuli?
Yeah, that's a question that's a thing that I thought about, but I actually don't know how the progression of the ontogeny of these cells goes in the septum, if there is a developmental migration of that. But one idea, actually, we have several ideas about how this might arise. Other than that, and that is the input from the hippocampus. So the input from the hippocampus is quite topographically organized as well, where, I mean, very coarsely described, the dorsal hippocampus goes to dorsolateral septum and ventral hippocampus goes to ventrolateral septum. That's an oversimplification, I would say, but this is one idea that we have, and I alluded to it in my circuit diagram, is that we're wondering if somehow like social memory, which has been demonstrated very much in the hippocampus already, and especially in the ventral, it's been shown that there are these dorsal ventral differences in the hippocampus. If this might be an interacting circuit with the lateral septum that might work to set up that topography. But then again, the development of these neurons in the lateral septum, that's a very interesting thing. And yeah, one other thing I would like to add is that we're not exactly sure how the topography arises in the very young animals, because we had a smaller data set from the very young animals, and teasing apart the anatomical locations of the cells in the neonatal brain, it's significantly more difficult than doing it in a little bit older animals. But the development of the topography, that's a very interesting question that we would also like to address by using some other methods, potentially like recording from large numbers of neurons with a single silicon probe at once. Our methods were quite arduous, and that we recorded from a single neuron at a time. So this topography and how it develops and how it interacts with circuits, those are all questions that we're now looking into.
Thank you. And one last question, and I'll take this. It's along the same lines. So the nepotypy that you've described, do you have any idea of how the cells in the superficial and deep layers are connected to each other? Are they independent? Do they interact? Is there any sense of that?
There is, right, there is quite a bit of interconnection between cells in the lateral septum. But again, what I can tell you about that from my data is somewhat limited because I was recording from one cell at a time. But the methods that we're developing right now in Edinburgh are to record simultaneously from a large number of neurons in the septum, and potentially to use some genetic methods to target the specific inputs and outputs. So yeah, I hope that in the future that we can have an answer to that, how these cells might be interacting, especially in terms of their response profile. So yeah, these are also questions that we're looking into now, and that's a very important question to ask.
Okay, well, thank you, Anne. Let's thank Anne for inspiring talk. There are some questions in the Q&A that you might want to address. Eden has a particularly interesting one that I think everyone might appreciate the answer to. And if we prepare now for our next speaker, this is Dr. Bianca Jones Marlin. I had the pleasure of being in the same institution with her as she did her PhD at New York University School of Medicine. She's now an EE Just Fellow and a Simon Society of Fellows Junior Fellow. She's in a postdoctoral fellow in the AXA laboratory and is just starting her own laboratory as an assistant professor in the Department of Psychology, the Department of Neuroscience at the Zuckerman Mind Brain Behavior Institute at Columbia University. And she's going to talk about a very important topic, survival. Bianca?
Thank you, Andre. Thank you for having me. I'm excited to be here, and I'm already so excited for the lineup that we have. So today I'll be talking to you about bridging the gap between innate and learned behaviors and using the senses to promote survival. Specifically, I'm interested in parents and their ability to promote survival in their offspring. And parents promote survival in their offspring in two distinct ways. One way is through passing on genetic information, so things that become innate in the offspring. And these things that are innate are usually instinctual and tied directly to the genetics. Another way parents promote survival in their offspring is through learned behaviors. So they experience and support by teaching their offspring how to survive. An example of an innate behavior is here. If anyone's dabbled with children, we all know that they love putting things into their mouth, even when it's not always something that's beneficial to them. However, they can learn to eat things that are healthy. And here's an example of a primate mother showing her offspring how to look for termites to consume, as opposed to pennies. My PhD work done at NYU in the Frumke lab looked at how innate behaviors can be learned. And specifically, I looked at maternal behaviors in virgins and how this can promote survival of their offspring. I'll talk to you a little bit about that today. And the work I'm doing now, and I'll continue to do in my lab opening next year, which is can learned behaviors become innate in the second generation? Specifically looking at the paternal adaptation to threat and how that can be inherited in the offspring for survival. And that's work done right now at Columbia in Richard Axel's lab, and I'll be continuing my own lab next year. So looking at the bridge between innate to learned behaviors and the learning and the expression of an innate behavior and experience. A great example is that of motherhood. So when a baby begins to cry, I have a 5-month-old now, so it's new and fresh in my mind. It begins to cry, and the mother or the caregiver will usually go towards the baby and pick it up and take care of it. Humans aren't the only animals to do this. Mice do this as well. And here's an example of a mouse mom I'll show you, where a pup begins to cool when it's left from the nest and emits ultrasonic vocalizations that we saw in Anne's talk prior. And this alerts the mom, so she'll go towards the pup, she'll pick it up, and she'll bring it back to the nest. And that's shown here. successful caregiver. Virgins, however, usually won't perform this task. They'll ignore pups, or even sometimes they'll cannibalize pups. Not shown here. Unsuccessful. So herein lies my question. How does an innate behavior and a standard sound, the sound of a baby crying, elicit different responses based on experience, and in this case, the experience of motherhood? We specifically looked at the neuromodulator oxytocin to answer these questions. Oxytocin has been known to be released during sex and orgasms, essential for pregnancy and the birthing process, in which oxytocin induces uterine contractions. It's continually released during breastfeeding, and it even released during touch, soft touch, and eye contact. It's been shown in mice to increase maternal behaviors, such as nest building and crouching. So then we asked, could oxytocin be the mechanism by which a virgin could potentially learn or unlock this innate behavior? Where, given the fact that the mothers will orient towards the sound of a baby crying, could the auditory cortex, primary auditory cortex, be a locus for these changes? And how, which we really didn't know, but how could oxytocin potentially change A1 to lead to these changes? In order to address the what, I performed a behavioral experiment where we had a mom who's an excellent retriever and a virgin who is not cannibalizing but not retrieving in the same cage. I then removed a pup from the nest, the mom's pup, and I gave the virgin a time to explore the pup while the mom retrieves the pup. The mom is then removed from the nest and the virgin is given a chance to retrieve the pup. When she doesn't, she's either given a cannula infusion of oxytocin into her auditory cortex, a cannula infusion of saline into her auditory cortex, or optogenetic activation of oxytocinergic terminals in the primary auditory cortex to release oxytocin specifically in those areas, and also systemically in the brain. What we observed is that when we induce oxytocin release in the virgin, she can learn to retrieve the animals more quickly. So with the presence of the mom, observing the mom retrieve, plus the presence of oxytocin as shown here in red and optogenetic activation in blue, the animals can learn to retrieve, unlock that retrieval behavior faster as compared to saline. So endogenous oxytocin enables retrieval. So oxytocin itself induces the learning of retrieval behavior in virgins. We then wanted to ask, is the auditory cortex important for this? Work done with my colleague Mariella Mitre and in Moses Chow's lab, we created a novel antibody to the oxytocin receptor. What we found was really cool. So here on the right is a coronal slice where the blue circles highlighting auditory cortex. And to the left panel, we see that every cell that's red has an oxytocin receptor made by a novel antibody, and blue is just DAPI, so cells that aren't expressing oxytocin receptor. When we compared the expression of oxytocin receptors in the mouse auditory cortex, we found something super cool, that there is an increased representation of the auditory receptor, oxytocin receptor, in left versus right auditory cortex. And that's shown graphically here. Moreover, what was really interesting was that in mothers and also in virgins, we see an increase in left over right. There's a lateralization of the oxytocin receptor in left versus right, a social cue, known to be a social cue represented in lateralization. We thought this was really cool because in humans, there seems to be a lateralization of social communicative areas, for example, Broca and Wernicke area. In right-handed humans, it seems to be lateralized to the right side. So to see lateralization of a social mechanism was extremely exciting. And to see that it was the same in both mothers and virgins shows that there's a network that's set up potentially to create virgins to become excellent caregivers. And here's an example of a mother. I'll show you a video of a mother who was retrieving at 100%. We then infused the GABA agonist musmol, so I'm transiently inactivating left auditory cortex. What we see is that inactivation leads to an activation of retrieval behavior. The pup is emitting ultrasonic vocalizations. And the pup is retrieved. This is after musmol. It's as if the mother acknowledges that the pup is out of the cage, but not that it's in distress. We wanted to see then if oxytocin, specifically in left auditory cortex, given our distribution of receptors, will induce retrieval behavior. So we took the virgin infused with oxytocin, saline as a control, or optogenetic activation of the terminals in left auditory cortex. And what you can see here is that oxytocin, specifically in left auditory cortex, enables retrieval behavior similar to what we observed with the systemic release of oxytocin, as shown here in red and in blue, compared to saline that's in white. So the left auditory cortex mediates oxytocin-induced retrieval. We then asked how. So in order to observe what was happening potentially in the primary auditory cortex, specifically the left primary auditory cortex, we asked, does learning change cortical responses to pup calls? In order to do this, we performed whole cell in vivo physiology, similar to what we observed before with Ant's talk, and we played ultrasonic vocalizations that were recorded from mice. So although the pups make ultrasonic vocalizations, we played them back with speakers. I then patched cells in anesthetized mothers, virgins, and experienced virgins and looked for the spike output. Here's an example of the ultrasonic vocalization recording. And each one of these rows is a current clamp recording. On the bottom, we have a raster plot where every dash in blue represents a spike that happened within 10 milliseconds of the former call. So these cells in a mother are expressing a time-locked and robust response to the sound of the baby crying. However, in a naive virgin, and I'll just point out that these two calls are different, the mother and the naive virgin, we use the best response call. So this call elicited the best response in this animal. The naive virgin's auditory cortex is active, it's responding, but it doesn't seem to be robust compared to the behavior or time to the mother or time locked. However, a virgin who has then learned retrieval behavior seems to have the signature of this robust and time-locked response to the pup call, similar to that of the mothers. So we asked, how is this happening? How could a virgin learn retrieval behavior and also have a signature that's similar to that of mothers? What's happening in the auditory cortex, and could oxytocin be mediating this response in real time? In order to do that, I patched in a virgin who initially was not retrieving, and I played the pup call. We then paired the pup call with optogenetic activation of oxytocin terminals in left auditory cortex. And you can see after 15 minutes of recording from the same cell, there's an increase in activity. After 30 minutes, we see this same increase, and also there starts to emerge a time-locked response. And after about 190 minutes, about 3 hours of recording from a cell in that same area, we see this emergence of the time-locked response. We have morphed the signature of a virgin into that of a mother by pairing oxytocin with the sound of a baby crying. And this is what we hypothesize is happening during birth, where the experience of motherhood with the flood of oxytocin that's being released from the uterine contractions is paired with the first salient cue. And that cue is the sound of the baby crying. So what we just demonstrated here at the 15-minute mark, leading to a change in that signature and therefore a potential change in the behavior by taking a virgin, leading them to successful behavior, a successful retriever, or taking a mother, first-time mother, and leading her to a successful caregiver. So synchrony, and I didn't show you this, but what we did observe is via the rebalancing of cortical excitation and inhibition, we see this emergence of spikes we can talk about later. So I showed that an innate behavior can be learned through experience, through support and observation, and endogenous release of oxytocin. I then asked if we can take a learned behavior and make it innate for survival. And this is work that I'm doing currently in the AXA lab. And it stems from work, data coming from the Dutch hunger winter, which is the last winter after World War II, in which the Netherlands were starved and they had food flown in. There's an example of this picture here. And what researchers observed is that those that were starved during the hunger winter, specifically males that were starved during the hunger winter, and they knew this because they took excellent note of those who wanted to serve in the military, their children and their grandchildren suffered from metabolic issues, such as hypertension, which is high blood pressure, diabetes, and even schizophrenia. As if the children and grandchildren were born for a world of paucity, but yet they were born in the world of plenty. As if the trauma of the parent was remembered in the kid and the grandkid. Work done by Oliver Rando's lab has taken this into rodents, looking at rodents with high protein and high fat diet, and observing that when you, the second generation, F1, filial 1, as shown here, has altered metabolism, altered reproductive success, and altered behaviors. Similarly, the Ressler lab at Harvard, and work that we're following up on, has looked at trauma in mice associated with odor. And what they've shown is that F1 generations seem to have a change in response when a parent is traumatized with an odor, shock and odor. And there also seems to be a morphological change. And this is work that I'm further examining. So we're asking, does odor fear conditioning lead to morphological changes in the olfactory system through learning? And this is in the F0. So first and foremost, can an experience actually change the morphology of our brains? And secondly, can this change be then inherited and potentially become innate in the offspring? In order to explore this, I'm using mice and I pair them with an odor. I'm using an odor such as acetophenone, which smells like almond, laurel that smells like flour, and propanol that smells like alcohol. And I pair them either with 10 seconds of an odor co-terminating with a shock, or 10 seconds of an odor, a minute in between, and then shock. And this is the unpaired paradigm. And what we observe is that top is paired, bottom is unpaired with acetophenone, propanol being the control on the left, is that paired animals will avoid the chamber in which the odor and shock were paired. There's no shock here, they're just now presented with two odors. Whereas the unpaired animal will explore both chambers. Although they got a shock and the odor, the co-termination seems to be important in this behavior. We then look at changes in the auditory, in the main olfactory epithelium. These are the first cells that sense an odor coming in. Here's an example here on the left of a laxi staining of main olfactory epithelium cells and their glomerulus. So every cell that is in the main olfactory epithelium expresses a particular receptor, and those cells that express the same receptor will converge in the glomerulus, shown here. This is a beautiful image, but it's difficult to count cells in an image like this. So what we decided to do is use iDisco, which is a brain clearing technique, to image these cells with single cell resolution. So here's an example of the main olfactory epithelium that I've extracted. I then embed in agarose and I clear, shown here. And I'm now able to observe the paired and unpaired cell counts after the behavior. And so what we observed, what we were able to follow up on, was that in animals that stayed in their home cage and never experienced acetophenone, in this example, we saw the average count of cells. Animals that were unpaired, which means they had the shock and the odor, but it was within a minute apart, also had a similar amount of cells as those that never experienced acetophenone at all. However, in the F0, animals that were shocked with the odor co-terminating seem to have an increase in the number of cells that express the olfactory receptor. And this was really exciting within itself because it's somehow saying that cells are either living longer because of this experience, or they're informing a stem cell population of this experience to have them bias their receptor choice. Moreover, what was extremely exciting was that we observed that olfactory fear conditioning in fathers increased the olfactory sensory neuron numbers in naive offspring that have never even experienced the smell of acetophenone. So we asked, are cells living longer? Are new cells being born in F0? And then what is happening in F1 in which that message can be carried down to the offspring? That learned message can be carried down and become innate potentially in the offspring. In order to look at how cells, the age of cells, we use EDU, which labels the cell birth date. And here's an example here to the left of the bulb, in which I have two glomeruli that are expressed in green. This is the M71, so it responds to acetophenone. And we're looking at the rostral migratory stream. So cells here at 3 days are time stamped. And then at 21 days, you see they go to their respective area to show that we can label these cells and label their birth date. We then wanted to look at how cells were being born and where they fall in the layer. So here's an example of the mantle factor epithelium, where here will be the nasal cavity. And we're looking at mature OSNs and also the stem cell population, and we're labeling them with EDU. And right here, green is GFP, so this cell responds to Lyral. And we want to observe if cells are living longer or if new cells are being born and are choosing to be the paired odor. And here's an example here of a co-labeled cell, which means this cell was born after the timestamp of injection of EDU and the behavior. We can then take these data and look at them in using iDisco to look for co-labeling of the paired odor and also the EDU to see how many cells are being born. These are questions that we're still continuing to answer. And so what I'm taking on into my lab is what regulates the change in cell number. Are mature neurons communicating with the stem cell population to inform this information? What does a difference, what does that 1 minute make? Why does 1 minute make all the difference? Between the paired and unpaired, we have activation of these MOE neurons, mantle factor epithelium neurons, and also activation of stress centers. So here's an example of a recording I performed with the oxytocinergic neurons. But are there stress-induced neuromodulatory centers that may regulate the increase in cell number, given the co-termination? And then how? What is the mechanism of this inherited experience in which an F1 generation will have an increase in cell number? And here's an example of sperm that I was able to remove from a anesthetized alive mouse. So we can look at the time points in which we see these changes over the experience of learning. And so with that, I'd like to thank the Frumke Lab, my PI for my PhD, Rob Frumke, and Joanna and Naomi are also following up on this, on oxytocinergic work, as well as Jen, she's had an amazing paper in Nature, and Mariella, who worked with me on the oxytocin receptor. And the team Transgeneration, my team now at Columbia, that are continuing to do the work on transgenerational epigenetic inheritance. And with that, I'd like to thank my funding sources, and thank you for listening.
Wonderful, thank you for that. Again, I invite you to ask questions in the Q&A, and if you'd like to come on screen, just raise your hand and you'll be invited. So that was absolutely fascinating, Bianca. I'd like to start with, one normally thinks of primary auditory cortex as simply representing sounds. What you've clearly demonstrated is it's not merely just representing the pup cries, let's say, the stress calls, but it's sufficient to mediate behavior. Were those recordings done in the fully awake animal, or were they under anesthesia? And the reason I ask is you can imagine that the sound itself and its representation might not be sufficient and would recruit the rest of the brain to actually entrain, if you will, the maternal or recruit the maternal behavior. And so I'm trying to understand how to think about that. Thank you.
Thank you for that. So the recordings, the whole in vivo recordings were done with lightly anesthetized animals. But we did follow up with these experiments, doing in vivo patch recordings, as well as tetrode recordings. And what we were able to observe is that with the tetrode recordings, the virgins actually had a response in primary auditory cortex that came online at the parallel point of retrieval behavior. So at the point of 12 hours, we started to see an emergence of this time-locked spiking at the point in which the animals were retrieving. So yes, during an anesthesia, we may, during awake behavior, we may be recruiting different aspects, but we're still seeing this similar time-locked response in both awake and also lightly anesthetized.
Thank you. Any other? Okay, so I had the great pleasure of being present at my daughter's birth, in fact, helping to birth her. And one of the things that I can gather from what you've shown is that it's actually fairly important for the mother to, one, not be anesthetized, two, to actually experience the oxytocin, and perhaps if we take it superficially, engage with the infant's early signals and crying. So how does that map to some of the birth practices that we see? For example, C-sections, or for example, induced births with Pitocin?
Excellent question. I think biology's got us. I can speak from experience of having two little ones, one which I was fully present for and one that I was not so present for. And although we are in the moment, for example, like, Pitocin is usually given to induce uterine contractions, especially if you're going to induce birth. We're continually releasing oxytocin. So holding one's child, breastfeeding if one chooses to breastfeed, breastfeeding one child, soft touch and hugs, these are continually releasing oxytocin. So if one does not have the ability to experience that fully awake and alert during birth, I don't think it negates that bonding. And moreover, like, my real interest in looking at parental behavior and paternal behavior is through foster care. I got the ability to grow up in a foster home, although my parents are biological parents, and they loved my siblings and me similarly. And so it could also be that these virgins are excellent foster parents through the learning of an innate behavior. You don't have to actually give birth to have those oxytocin released. And we know that because I assume that you also really do love your children, but you didn't have to be there to have the uterine contractions.
Yes. Thank you. Christine, you have a question?
Hi. No, sorry. Hi, Bianca. Thanks so much for the talk. That was great.
Thank you.
So it's pretty cool that you showed that there's a biological effect on the F1 offspring. I might have missed it, and if I did, I apologize. Was there a behavioral effect?
Yeah, you did not miss it. Following up on the DSM wrestler work, we did not observe a behavioral difference. So what we did is we did the same tri-chamber experiment where we had the mice with the F1s with acetophenone or propanol, and their parents were paired with acetophenone or propanol. And we did not see an avoidance to that odor, but that's okay because we're seeing an increase in cell number, and that may not be an increase of cell number directly tied with an increase of fear or threat, something that could potentially be associated with PTSD, where there's a stressor that is not really dangerous, but preparing the animal to learn more quickly by a change in gain. So an increase in cell number could say, if you do need to learn that this odor is terrifying, you can learn this more quickly.
So will that be like the next behavioral experiment?
Yeah, so actually the next behavioral experiment is paralleled with that. And we're actually looking at taste as well to see how taste, something that's more essential for survival, can be changed and how much of that is being tied onto the F1. There's some pretty cool stuff with that. Thank you.
Thank you. So for the last question, this is by Christian Ebsen. Interesting question. He says, can you speculate a little bit how you think the olfactory signal is being passed to sperm? The Diaz and Rensselaer 2013 paper showed methylation changes in sperm, but how does the signal get there from the nose?
It's the most fun question that we have. How does the signal go from the nose to the testicles? And what we're following up on is work that also was done in the Randall lab. And what they've shown is that the epididymis, which is a part of the testes in which immature sperm travel through to become sperm, give a payload via epididymosomes. So what this is it's a little vesicle that seems to have RNA that's similar to RNA that's observed in the liver. So they looked at metabolic issues and they saw that there were these RNA were in the liver and also in the epididymis and therefore in the sperm that passed through the epididymis. So there's some type of correlation with things that are happening in the sperm and things that are happening in the liver, but they haven't looked at it, they haven't observed it in a pathway form. And so when I talk to it with my students, we kind of joke that it's the quantum entanglement of the body. We don't know if there's an actual direct connection, like via blood, et cetera, from getting to the nose to the sperm, but somehow these two markers were found in these two areas. And so that's what we're looking at now, whether or not we can observe markers in both the nose and the sperm and how that possibly can travel that direction.
Awesome. Biology is pretty crazy. So thank you, Bianca. That was absolutely fascinating. Time for our next speaker. This is Christine Constantinople, my colleague. She's an assistant professor here at NYU at the Center for Neuroscience, and my neighbor just down the hall and down a flight of steps. And she's going to tell us about an analysis of decision under risk in rats. Christine?
Cool. Thanks so much, Andre, and the organizers for the opportunity to speak today. So I started my lab about coming up on 2 years, which is very exciting, but I'm going to talk today mostly about postdoctoral work that I did in Carlos Berti's lab at Princeton. Oh no. So as a neuroscientist interested in decision-making, I'm drawn to behavioral economics because I think it provides a really useful quantitative framework for describing human choice behavior. And I just want to give a brief background of some key concepts that are going to be important for the talk. So arguably the main workhorse of modern economic theory is the concept of utility. So the idea is that we evaluate outcomes or rewards, not based on their absolute amount, but based on the subjective satisfaction we derive from consuming them. And so you can formalize that with something like a utility function, which maps reward, here I have money on the x-axis, onto the subjective satisfaction or utility we derive from that reward. And the shape of this utility function has consequences for our preferences and for our choices. So for instance, this particular shape is called concave. It tapers off like this. And it captures a number of aspects of choice behavior. One thing is a phenomenon called diminishing marginal sensitivity for gains. And so the observation is that the difference between $0 and $1,000 feels a lot bigger to us than the difference between $110,000 and $111,000, even though the delta dollars there is the same, right? And so this function can account for that because 0 and 1,000 are more discriminable or farther apart on the y-axis down here, and they're closer together out here. So this particular example is probably familiar to the sensory physiologists in the audience, but in general is just meant to give you kind of a concrete intuition for one aspect of choice behavior that utility can account for. So the predominant behavioral economic theory of decision-making under risk is called prospect theory. It was developed by Daniel Kahneman and Amos Tversky and got them the Nobel Prize in economics. And it describes basically 4 main features of human choice behavior. So the first is diminishing marginal sensitivity, which I described, which you can account for by having a concave utility function for gains. Another phenomenon is called reference dependence. So the observation is that people evaluate outcomes as gains or losses relative to an internal reference point that at least some of the time probably reflects expectations. People also tend to be loss averse, so we're more sensitive to losses to equivalent gains. And you can account for that by having a loss function that's steeper than the gain function. And finally, people exhibit probability distortion. So we tend to overweight the likelihood of low probability outcomes and underweight the likelihood of medium to high probability outcomes. And so for economists, the equations that describe these functions are a set of tools that allow them to describe human choice behavior. But as a neuroscientist, I think these are a number of really interesting cognitive phenomenology. And so we would like to leverage rodent models to try to understand the neural basis at a more mechanistic level. And so this is basically what my lab is about. So during my postdoc, I decided to try to apply prospect theory explicitly to rats. So most economic theories have been developed to describe primate behavior. So obviously the field of economics was developed to describe human behavior, and there's been a lot of really beautiful foundational work in non-human primates that's shown that they exhibit some of the same phenomenology. And those paradigms elicit what an economist would call decisions from description. So think about choosing between explicitly described lotteries. And that's in contrast to most of the paradigms that have been used to study risky choice in rodents. And those elicit what are called decisions from experience. So think of something like a bandit task where the animal has to look by sampling them over time. And so we wanted to come up with a way of eliciting decisions from description from our rats so we could apply these same formal economic models. So to do that, we use sensory evidence to convey explicit information about reward attributes to animals on each trial. So in this task, the rat initiates a trial by poking his nose in the central port. He's then presented with a series of light flashes, the number of which convey the reward probability at each side. And simultaneously, he's presented with auditory clicks, the rate of which convey the reward volume that the rat's offered. So in this trial, the sensory cues are telling the rat, you can either choose between a water reward of 48 microliters with a probability of 0.3, or a guaranteed reward of 6 microliters on this side. And at the end of the trial, there's no right answer. The rat is free to reveal his preference and choose the side that he wants. So just a couple details. One side always offers a guaranteed reward. And in this example, the risky prospect is a higher volume, but that's not always the case. We have a very diverse choice set. And specifically, we offer 4 possible water volumes. They're on a log base 2 scale, so they're roughly equally discriminable to the animal. And the risky side ranges in probabilities from 0 to 1 and 0.1 increments. So this probably sounds like a really difficult task for the rats to learn, and it is. It takes them at least 4 to 6 months, sometimes longer. So to overcome that training bottleneck, we used a high throughput behavioral training approach. So this is an outdated photo of the high throughput facility in the Brody lab when I was there at Princeton. And at that time, it had 37 training rigs that trained over 300 rats per day. And the training is computerized and semi-automated. And so once you write the software that transitions the animals through the shaping procedures, basically technicians load the rats in and out of boxes. And so this allowed us to, again, overcome this training bottleneck, have a ready pool of trained animals for experiments, and also have a lot of statistical power for behavioral modeling for each animal. And we developed our own high throughput training facility in my lab. Okay, so using this approach, this is showing behavioral data from one animal, and the data is super rich and kind of a lot to look at. So just a couple things I want to point out. So each of these matrices corresponds to the animal's choices or the probability they chose the safe option on trials offering a unique safe reward. So 6, 12, 24, or 48. The y-axis of each matrix is the reward offered, the volume offered on the risky side, and the x-axis is the probability on the risky side. And so in general, the rats tended to choose the side with the greater expected value. When the safe side was the best option, 48 microliters, the animal chose it almost all the time. This is mostly yellow. When the safe side was the smallest option, the animal chose the safe option much less. This is much more pink. And we get a really rich description of the animal's choices across a diverse choice set. Oh, no, here's my mouse. And this is average data across 36 rats using more than 2.5 million trials. Okay, so one thing that we worried about a lot was, do the animals really understand the meaning of the cues, or are they using some sort of other strategy like a perceptual heuristic? So we looked at this a couple of ways, but one thing that we found was the following, which I think is pretty convincing. So the rats are supposed to keep their nose in the center port for the duration of the trial, but sometimes they would leave early and terminate the trial. And they would be punished with a timeout and a white noise sound. But we can look at those trials in more detail. So here I'm plotting the probability of violating or opting out of the trial as a function of the volume offered on the risky side versus the safe side. And what's interesting is that for this rat, when both sides offered the smallest possible reward, the animal was opting out almost 30 or 40% of the time. Like it wasn't even worth his time to complete the trial. Whereas when both sides were offering a large reward, the animal almost always completed it. And so that really indicates that the animal understood that the auditory cues were telling it or conveying information about the reward being offered. And this was highly replicable across our population of 36 rats. And so I would argue that on its face, this is inconsistent with something like a purely perceptual heuristic. Okay, so we used a behavioral model to try to estimate the animal's utility and probability weighting functions a la prospect theory. And this is just to show cross-validated model performance for one rat where the data points correspond to the choices on left out test data. And this is showing the model predictions for that data. And so these are the results of the model fits. So for our 36 rats, there's quite a lot of interesting individual variability in terms of the shapes of the utility and probability weighting functions, but almost all of the rats basically have this kind of concave shape to their utility function, which would produce diminishing marginal sensitivity for gains like humans. And similarly, most rats also exhibit probability distortion, overweighting the likelihood of most outcomes. So what's interesting is that this is kind of qualitatively different from the prototypical human, and we have some ideas of why that might be. But in general, it looks as though the rats are qualitatively similar to humans. Interestingly, most studies in monkeys found that the utility function looks different, and so it seems like our rats are closer to humans, but less, not so much monkeys. And again, that might be kind of task specific. It might have to do with the range of reward volumes that we offer. Okay, so we also found that the rats exhibit evidence of reference dependence, where they subjectively value outcomes relative to some internal reference point. And so one way that we can look at this is by looking at their trial history biases. So this is showing the change in the probability of repeating a left or right choice relative to the average probability of repeating, depending on the reward that the animal received on the previous trial. And so you can see when the animal doesn't receive any reward, they're much more likely to switch and go to the next port on the next trial. And so that's called a lose switch bias. And when they receive a really large reward, they're more likely to stay. But what's interesting, each gray line here is a rat. What's interesting is that for 6 and 12, a lot of the rats are actually pretty likely to switch after they receive those small volumes. And that suggests that they view them as losses relative to some internal reference point. So we were a little worried that maybe there's just something weird happening on the low end, and like the solenoid valves weren't reliable for those water volumes. So we took 3 rats, and through their lifetime, they trained on a version of the task in which all the reward volumes were doubled. And so now they range from 12 to 96. And so for these rats, they also exhibit loose switch biases, this time for 12 and 24. And so this really suggests that this reflects a subjective valence assignment that reflects something about integration to the mean reward offered. Moreover, the threshold for what the animal deemed to be a loss or a gain seems to be reward history dependent. So here I'm showing data from 1 rat, split based on whether 2 trials back was rewarded or not. And you can see that there's a clear shift. And so we think that for most of the rats, they're using their reward history to compute the reference point. And that's something my lab is following up on right now. And finally, at least at the aggregate level, the rats seem to be loss averse. So this is showing the median value function for 36 rats. And in general, the loss function is steeper than the gain function. And so we've basically demonstrated that rats exhibit, at least qualitatively, all the key features of this predominant economic theory in humans. And that's really exciting because it means we can start to leverage the powerful toolkit available in rodents to interrogate their neural basis. But that's not kind of the whole story. So the behavior was super rich and interesting and wasn't fully explained by these economic models. So in addition to these nonlinear value functions that were driving behavior, the rats also exhibited trial by trial learning, which I kind of showed for the Wednesday lose switch biases. And I call it deleterious because in this task, each trial was independent. And so it's actually bad strategy to use the outcome of the previous trial to change your behavior. And yet the rats did it. And so on our task, there were at least 2 distinct types of trial history effects that we observed. So let's say on one trial, the animal chose the left side where they selected the gamble and they were rewarded. On the next trial, there's at least 2 types of sequential effects we could observe. Either the animal's more likely to revisit the rewarded spatial location, and so we would call that a spatial Wednesday lose switch bias, or the animal's more likely to gamble again, irrespective of the side that the gamble is offered on. And so that would be a risky Wednesday bias. And we observed both of these effects in our rats. So again, I kind of showed you this already. This is collapsing across volumes. All of the rats were more likely to revisit a rewarded location and switch after an unrewarded location. And we also found that rats were more likely to gamble following risky wins. So this is the change in the probability of choosing safe, again, relative to the average, depending on whether the previous trial was a safe or a risky reward. And each line here is a rat. And so you can see that basically every rat is more likely to gamble following a risky reward. Moreover, the magnitude of the effect exhibited a graded dependence on the probability of the previous reward, suggesting it was updated by some sort of prediction error. So it's worth noting that the magnitude of this is small. It's about an order of magnitude smaller than the Wednesday lose switch bias, but it is reliable and present across all animals. There were also some interesting differences with the Wednesday lose switch bias and the risky bias. So for instance, the spatial bias seemed to decay with time. So if you looked at longer intertrial intervals, the magnitude of the effect was much smaller, whereas this bias was much more stable and didn't depend on the duration of the intertrial interval. Okay, so we were interested in how these kind of trial by trial learning effects might influence the animal's estimates of subjective value. And so the place we focused on was the orbitofrontal cortex, specifically the lateral orbitofrontal cortex in rats, which based on projections we think is somewhat homologous to the caudal third of the primate centrolateral OFC. And I'm happy to talk about homology in more detail. So unfortunately, because of time, I'm really not going to talk much about the neurophysiology. We perform tetrad recordings, and it's an extremely rich data set. You know, it's prefrontal cortex. But basically, I'm just going to show you two summary plots, which is that we saw strong encoding of risky and safe choice and also outcome at the time of the choice. So here I'm showing, this is a discriminability index, which is basically the average difference in firing rate of cells in these two conditions divided by the square root of their mean variance. So you can think of this as the magnitude of the encoding of whether or not the animal made a risky or safe choice. And so there's about 10% of cells, it's kind of a small subpopulation that encode this, but we did see encoding of whether or not the animal was choosing risky or safe on that trial, again, aligned to choice. And we saw much stronger encoding, both in terms of fraction of neurons and the magnitude of the discriminability index, reflecting left-right choice and also reward outcome. And so this time aligned to when the animal's actually executing the decision corresponds to really rich dynamics in OFC. But I should say that looking at these dynamics, it's not a priori obvious what we might expect photo inhibition to do, right? So it might be the case that this kind of small subpopulation of cells mediates something like the risky Wednesday bias because they're telling the animal, whether or not it's choosing the risky or safe side, or reflecting that. Or it might be that these more ubiquitous neurons are important for the spatial Wednesday loose switch bias. So because we saw all of this encoding during this period, we performed photo inhibition aligned to when the animal left the center poke to make his choice for 4 seconds, and then we analyzed behavior on the subsequent trials. And we took 2 approaches to inactivation. For 8 rats, we expressed halorhodopsin downstream of an excitatory promoter, and for 5 transgenic rats that expressed Cre recombinase downstream of the PV promoter, we expressed a Cre-dependent chondrorhodopsin. And so these guys were inhibiting excitatory cells mostly, and for these guys, we're exciting inhibitory cells. So they're just complementary methods. And so in both groups, we basically saw that we eliminated this risky Wednesday bias when we photo inhibited during the choice and then looked at the subsequent trials. And there's no effect on sham rats, and there was also no effect on the spatial Wednesday loose switch bias. And so again, it's worth noting that this is kind of a subtle effect, right? But the magnitude of the effect is going to be limited by the magnitude of the risky bias to begin with, which is small. And this was reliable across 13 rats and 2 different manipulations. And so it seems that orbitofrontal cortex is important for promoting trial-by-trial learning and specifically updating the subjective value of the risky option. So this was kind of surprising, and what was interesting about it is that this trial-by-trial learning was somewhat suggestive of reinforcement learning mechanisms, right? So, I opened with a description of economic theories of subjective value, but there are other kind of really powerful frameworks for thinking about subjective value in neuroscience, specifically reinforcement learning. So in reinforcement learning theories, there are these really early psychology and behavior studies. Here I've shown an image from Pavlov, where he trained dogs to make an association between a conditioned stimulus and an appetitive reward. An early learning theorist wrote down some learning algorithms. This is the Rescorla-Wagner learning algorithm that could explain how the animal might update the associative strength, and then later those models were extended to account for how one might learn values from experience. Basically by updating their previous value estimate by some reward prediction error multiplied by a learning rate. So what's interesting here is that these standard reinforcement learning algorithms are somewhat incompatible with economic theory because this equation will learn the expected value of R. And so what I mean by that is, let's say that the pairing between the bell and the steak is probabilistic. So only half of the time the steak comes. That means on half of the trials, R will be 1, and on the other half, R will be 0. And so with enough experience, the animal will learn 0.5 times the value, which is the expected value. And that necessarily implies linear utility and probability weighting. So just to make the point, this is a simulation of a Q-learning reinforcement learning agent performing our task. And sure enough, if you fit the prospect theory model, you recover these linear value functions. So the question is, how can we account for both the trial-by-trial learning that we saw in our rats, but also the nonlinear value functions? And we proposed a really simple hypothesis, which is that you can have a Q-learning agent, but if your reward prediction error is in units of subjective value according to prospect theory, then you can have it all. You can account for both the nonlinear value functions and also the trial-by-trial learning effects. And if we did model comparison, this type of reinforcement learning fit better across all of our rats than a similar model that had a more standard reward prediction error. And so we think this is kind of a parsimonious way of integrating concepts of subjective value from both economics and reinforcement learning. And also we think that it's biologically plausible. So there's about 20 plus years of work that suggests that phasic dopamine and stand sheets, a reward prediction error of the kind that is in these learning algorithms. This is a classic study from Wolfram Schultz's lab where they trained a monkey to associate a stimulus with a reward that came at a different delay. And what they found was that the phasic dopamine response reflected the subjective hyperbolically discounted value of those delayed rewards. And so we think that not only is this hypothesis parsimonious, we also think that it has biological plausibility. Okay, so I've shown you that rats behave according to prospect theory, which is really exciting because now we can explore these cognitive phenomenology in rodents. OFC promotes trial-by-trial learning, even in this case when it's maladaptive. And specifically, OFC promotes learning in task-relevant but not irrelevant coordinates. So in this task, it corresponds to risky and safe but not spatial coordinates. And I think this has connections to ideas about OFC reflecting a cognitive map. And finally, we argue that a teaching signal, a reward prediction error, and units of subjective value, at least according to prospect theory in the case of explicitly described lotteries, could reconcile economic and reinforcement learning frameworks. Okay, and all of this work was done in the Brody lab. So I'd like to thank Carlos Brody and other members of the Brody lab at Princeton, and especially Alex PA, who is a close collaborator on the project and gave a lot of guidance for the modeling. And thank you for your attention. And I'd like to thank my lab. We're following up on a lot of these questions now.
Terrific. Thank you, Christine. So I'll start with, there's a question in the chat. I'll postpone mine. Martin O'Neill asks, comments first, lovely talk, thanks. Can you say a little bit more on how you incorporate subjective value in your behavioral model, please? Is it an additional variable added to the reinforcement learning cue learning model?
That's a great question. Oh God, I have extra slides here. So I don't have a schematic of the RL model. This is just, so basically in the prospect theory model, we assume we parameterize subjective utility as basically with a power law. And then we tried a bunch of different parametric forms of the probability weighting function. We settled on the 2-parameter Prelec equation. And so the subjective value of each offer is basically the product of the subjective utility and probability weighting functions. And then we take the difference of those value estimates from the left and right side, and we treat that as the argument to a logistic function to get a choice probability. And then we use maximum likelihood procedures to find those best fit parameters in the case of utility, the power, the exponent, and then the 2 parameters for the Prelec function that basically maximize the likelihood of the data. So that was the standard prospect theory behavior model. In the Q learning model, we basically had, so we have, I'm trying to think about how to describe it. We have like a number of distinct trial types, right? So we assume that the animal learned the value of each unique trial type, which is actually feasible given how much training the animals required. Some of the animals in particular required to learn this task. And so basically they're basically learning a lookup table where they have some value estimate associated with a choice condition given each trial type. And the key thing is whether or not the reward prediction error itself is subjective utility times W times subjective probability, or if it's your more standard RL algorithm, in which case it would just be R on trial T. Does that make sense?
Great. Anne, you had a question?
Yes, very nice talk, Christine. So I'm not that familiar with economic theory, but my question is a somewhat basic one. Like, what does the influence of economic state have to do with the utility function? So for example, like in terms of the rat, if it's very thirsty to begin with, if it's very like water deprived, is the utility function different? And also like in human choices, if you're assessing somebody that comes from a very poor background, is their utility function different than somebody who has a lot of resources?
Yeah, so that's a great question. Oh wait, no, didn't mean to go forward. And Kahneman and Tversky, the prospect theory guys, kind of tried to account for this with the reference point. And so their initial idea was that the reference point corresponds to your status quo wealth. And so you're basically evaluating outcomes relative to how much wealth you have, in which case the actual shape of the utility function doesn't necessarily change, but just your place within it, where you start, changes. Now, it turns out that the status quo wealth idea doesn't really square up with a lot of empirical data. And there is some evidence from applied work that wealth status can have an effect on the shape of the utility function. So I didn't really get into it for the sake of time, but the shape also has effects on your risk attitudes, like how willing you are to tolerate risk. And it seems like that does vary with wealth. And there is some work from Paul Glimcher's lab in monkeys also that finds that, so there was a cool study, Yamada is the first author, where they basically measured the blood osmolality of the monkey every day to get a concrete metric that corresponded to their kind of physiological wealth, right? Which is how thirsty they were, how much water was in their blood. And they found that that actually explained a lot of the day-to-day variance in terms of the animal's risk attitude. And I don't want to get it wrong, but I think when the animal was thirstier, I think they were more risk averse. But we should check the paper. Anyway, so I feel like I just said a lot, but the answer is it's complicated and really interesting, and there are kind of thought to be strong effects.
Cool, thanks.
Thank you, Christine. So I was curious to know if you've looked at the large amount of data that you have as the animals learn this and become good at it, if you will. So what do the data look like, and what would you describe the models that they might have early in training, intermediate, and then obviously we know later? And the reason I ask this is because it seems to me that over time, the animals, just like a human would, is building some kind of a model of what this world is like, what subjective value is, has to be based on some sort of notion that's an internal model of the circumstances. And I'm curious to know if there's any indication of how that develops.
Yeah, so that is a question I get a lot, and it's I feel like I don't have an awesome answer to it. We've thought about this somewhat, and it takes the rats, and I feel like this is consistent with some of the other work in the Brody lab where people are training rats in these really difficult tasks, where you kind of get two groups of learners. Some of them are pretty fast learners, and some of them it just takes months, and you get this really slow, like iterative improvement that almost seems like a sensory motor kind of thing. And I think at some point I should dig into it, right? But it would be reasonable to think that those kind of different groups are using different strategies. I did have a rotation student that kind of played around with different artificial agents, including one that adopted a model-based versus model-free kind of strategy to learn the mapping of the different cues onto reward attributes. And just in principle, as you would expect, a model-based approach will allow you to learn faster with less data. And so it's probably the case that the fast learners are doing something like that and learning like an abstract rule to map the cues onto reward attributes. But beyond that, I don't have much more to say at this point.
Okay, well, I'd like to thank you for a terrific talk. It is now time to move to our next speaker, who is also NYU related, a former colleague of mine, just from a floor above. Actually, I'm a floor above. She's now at Harvard. Dr. Elizabeth Phelps is the Pershing Square Professor of Human Neuroscience in the Department of Psychology at Harvard University. And she has the Zoom floor.
Unmute. Thank you. Yes, I miss having Andre nearby. Although things at Harvard are nice, I guess for all of us now, we're just sitting in our houses anyway. So what does it matter? Any case, so I'm going to be following up a little bit on sort of a bunch of themes you've heard today, talking about decision making, but bringing it back to this notion of threat processing in the brain. So I first want to start, just talking about what we've thought about with the emotion and decision making broadly in humans, and the tradition from psychology and also somewhat from economics has been to think that when we talk about emotion and decision making, we think about deciding with our heart and our head. And this was you heard about Danny Kahneman and prospect theory, but of course he also introduced this idea that we kind of have two systems for deciding, one based on kind of intuition and one deliberation. And emotion was always thought to be this sort of part that was related to kind of intuition, right, and driving choices. And I'm going to argue, from a sort of affective neuroscience perspective, that doesn't make a lot of sense, right? So these are just two slides or two images from how we think about the interaction of affect or emotion and its role in cognition, broadly speaking, in areas such as perception, where we know that the amygdala modulates visual processing sort of throughout the ventral visual pathway, and also memory, where arousal will change the consolidation of memories. And so we always think about, in other cognitive domains in human psychology, we think about emotion as having a modulatory role. And I think that hasn't really been applied to the domain of decision making broadly, in part because of this history coming from economics and psychology. But I'm going to say, can we start with a neuroscience model of how emotion might change some choices and understand its role in decision making in a little bit more nuanced way. And the model I want to start with is the model of threat conditioning. So I've done a lot of work on threat conditioning in collaboration with Joe Ledoux, my former NYU colleague. And threat conditioning is just this very simple Pavlovian paradigm where you have here in a rodent a tone that sort of elicits an orienting response. But when you pair the tone with a shock, the tone itself comes to elicit a range of responses we call defensive responses, such as freezing, increases in autonomic nervous system arousal, and stress hormone release. And we know from lots and lots of work that the amygdala is central to the acquisition, storage, and expression of these simple types of associations. But there's really sort of two reactions you could have in this circumstance. One would be what I just described, this reactive freezing. But if you give the animal an option to take a choice, to do something, an action, to avoid the tone, given that it predicts that they might get a shock, the animal will. And what we know from a lot of the neurobiological research is that there are two different pathways by which, the amygdala can signal other brain regions to either reactively freeze when there's no option to make a choice to avoid or to avoid. So this is the amygdala here. This is a slide I borrowed from a review article by Jack Gorman and Joe Ledoux. So we know that the amygdala is the site of storage for the association between the tone and the shock, and particularly the lateral nucleus of the amygdala. When you now have no option for avoidance, the lateral nucleus projects to the central nucleus, that projects to the brainstem, that creates all these sort of reactive responses like freezing. However, when you have the option to avoid, the lateral nucleus projects to the basal nucleus, which then projects to the striatum, which then mediates the actions to avoid. And so the question I wanted to ask in the talk today was, does this amygdala striatal circuitry play a role when deciding to avoid the threat of monetary loss? So Christine already talked about this somewhat, but I'm just going to give you an example of what I mean by the threat of monetary loss. So we call this loss aversion, right? People are very sensitive to losses. They're more averse to losses than they are to gains often. And so here's an example of what I mean by that. If I were to ask you, would you just take this gamble? 50%, flip a coin. If you win, you get $10. If I win, you give me $12, right? Or would you rather just not play, get nothing? Most people would say that's a really bad deal. Of course I won't do that. And that would be the right choice. What about this gamble? Okay, 50%, you win, flip a coin, you win, you get $10. If I win, you give me $2.50. yeah, you know, odds are pretty good for you, so most people might take this gamble. How about this choice? 50%, you win, you get $10, or if I win, you get $7.50. A good number of people would avoid this choice, right? And this is thought to be because the loss in some ways hurts more than the potential gain. And this we call loss aversion. So let's go to a situation like this. So here's an individual, he has a choice like this, you know, 50-50, he could either do nothing, get 0, or win 14, or if he loses, he'll lose $11. Let's say he says, no, thanks, I don't want to play. And then you do some manipulation, and now he says, okay, yeah, I'll play. So what did you change? Did you change the sort of relative aversion to losses relative to gains, or loss aversion? Did this person simply decide, excuse me, that you know, they kind of don't mind risk as much as they used to, or are they just really inconsistent, right? And so in order to be able to look at loss aversion of the brain, we have to be able to differentiate these three possibilities. And so this is how we did this. We gave people a number of choices like this, just like I described, 50-50, win $9 or lose $11.25, or would you rather just not play? That would be one set of choice. So for each individual, they would have a number of choices. They'd either take a risky gamble or get a guaranteed amount. And the only choice is, do you want to play? And what we can do with this task, we have a number of trials, is we can separate out 3 different variables. Their sensitivity to gain, like their sensitivity to risk, right? Their sensitivity to loss, and their consistency. So loss aversion is, we use lambda for this. 1 means, you know, if it's greater than 1, it means they're loss averse. If it's less than 1, it means they're gain seeking. They also have risk. This is involved in all of these tasks. So they can either be risk averse or risk seeking, or mu, or they can be just inconsistent, right? They're just noisy. They change their preferences over time, or they're consistent. And so we can separate these 3 by giving different types of gambles. We can give gambles where we have gain loss possibilities, like the example you saw, or ones where there's only gains that they could play. So this would just get at risk, but no loss aversion. And we can do this a number of times and measure how noisy are people in their choice process. So this is what we did. And we wanted to say, how are we going to measure affect here? So we chose physiological arousal for a bunch of reasons. 1, that's one of the ones we often measure in threat conditioning. And 2, we also know that physiological arousal and the neurohormonal impact of arousal can impact sort of memory and learning, and particularly striatal learning, as Jim McGall has suggested. So we chose this as our measure of affect. And we asked the question, you know, is physiological arousal that you have, does it code for risk? Does it code for loss? Or is it related to consistency? So this is just to say, these are our individual subjects. We had several individuals do this task. Most of them were loss averse. Some of them were gain loss neutral. Some of them were gain seeking. So this is lambda, the loss aversion coefficient. We measured their loss aversion when they won or lost gambles. And what we found, and I'm plotting this here on the left, this is their skin conductance response, a measure, excuse me, I don't know why I'm moving around like that, a measure of autonomic nervous system arousal. This is skin conductance per dollar that they could potentially lose. We call this loss per buck. And this is the difference between your skin conductance when you lose money or when you win money. And what we found was it correlated with behavioral loss aversion. People that are over aroused to losses relative to gains are more loss averse. And you know, when we looked inside the brain to see regions that would correlate with loss aversion, there was one previous study that was published to look at loss aversion in the brain, and it found activation of the striatum, but this particular task didn't separate sensitivity to risk or sensitivity to loss. So in other words, it seemed to be coding something a little bit more like subjective value. In our imaging study, we also found that the striatum correlated with subjective value. This has been shown. There's been a meta-analysis of regions in the brain that correlate with subjective value, and you routinely see the striatum and also orbital frontal cortex. So we also found in our task, again, when you combine all of our gambles, including looking at risk aversion and loss aversion, that this region correlates with subjective values. That's consistent with the Tom study published earlier. But when we did a whole brain correlation of regions that correlated with loss aversion per se, the main region that emerged was the amygdala. So individuals who showed greater loss aversion also showed greater activation of the amygdala to losses relative to gains. And importantly, there was no relationship between either your physiological arousal response or your risk attitudes or your consistency with amygdala activation. So in other words, the physiological arousal and amygdala response were specific to loss aversion. So this again is consistent with this idea that you're getting a threat. When we have threat, we have this sort of autonomic nervous system response, and the threat of monetary loss leads to something similar. So this is a kind of a consistent with this model that when the striatum will code subjective value, but the amygdala may uniquely code the threat, and here the threat of monetary loss, consistent with this model, suggesting that the amygdala, which may code the actual loss signal or the threat of loss, corresponds with the striatum, which may then help code subjective value, which may lead to the decision to avoid that circumstance. So once we discovered this, we wanted to then ask an additional question. So this is just what I suggested to you, right? Arousal induced by the threat of monetary loss modulates subjective value. We think by the amygdala modulates the striatum and the choice, which is reflected in loss aversion. But one of the things that we've looked a lot in the laboratory in terms of threat processing and threat learning is not just how we acquire these threat responses in the role of the amygdala, but also how we can get rid of them when they're no longer adaptive, when it doesn't make sense to respond to the threat. And so we wanted to look at two strategies we might use to change arousal and then to see if we could also change choices. One of these was cognitive emotion regulation, and the other was to use pharmacological manipulation to manipulate arousal. So what is emotion regulation? Emotion regulation is, this is something that's used in cognitive therapy. It's our ability to use our thoughts to reevaluate or reappraise a stimulus or situation to alter our emotional response. This type of cartoon is often shown in cognitive therapy to conceptualize this idea. We've done several studies of emotion regulation in the laboratory in threat conditioning and also reward conditioning. So here's one example. I could ask you, when you see a stimulus that predicts you might get a shock versus another one that doesn't, to either attend to your natural feelings or to try to come up with a strategy to regulate your emotional response. So I could say to you, when you see the stimulus, I want you to use the color of that stimulus to think of something calming in nature. This would be basically trying to regulate your response to a stimulus that means you might get a shock, or perhaps one that means you won't. And when people can use this strategy, what they're able to do is to reduce their physiological arousal to the stimulus that predicts a potential shock. So here is the skin conductance response again, when individuals are attending to their natural feelings, to the stimulus that had been paired with shock on a portion of trials, versus when they're regulating, they're using a cognitive strategy to regulate their emotional responses. Again, no difference in the stimulus that wasn't ever paired with shock. And what we see when you use this type of strategy is a reduction in bold signal in the amygdala. So here's the amygdala response to the stimulus that predicts you're going to get shocked when you're attending versus when you're regulating. This is very similar to other techniques like extinction learning, where you may reduce a physiological response. We also see more brain activity in two regions. This ventral medial prefrontal cortex region, this is when you're regulating. This is a region that corresponds to the infralimbic cortex in rodents, which is important in inhibiting the amygdala and the expression of a conditioned threat response in extinction learning. And this region here, the dorsolateral prefrontal cortex, this is a region that's involved in emotion regulation, other emotion regulation studies. It's thought to be related to the executive control processes, the online manipulation or your interpretation of the stimulus that leads to emotion regulation. We know that this particular region does not have strong connectivity to the amygdala, whereas this region does, the ventral medial prefrontal cortex. And so we did a connectivity analysis with this region and showed that it mediates the relationship between the dorsolateral prefrontal cortex and the amygdala. So when you're regulating a threat response, just to sum this up, what we think is going on is you have the amygdala, which sort of codes the threat reaction, the learned threat reaction. The ventral medial prefrontal cortex, again, the human analog to what we think is the infralimbic cortex that can inhibit the amygdala and the expression of the threat reaction. And when you use a cognitive strategy, the dorsolateral prefrontal cortex co-ops this phylogenetically older circuitry to now let you use your thoughts to control your emotional response. Interestingly, if you do a similar paradigm when you're anticipating getting a reward, so now same task, but now one stimulus means you might win $4. One stimulus means you're not going to get anything. I'm asking you to attend or regulate. You can get physiologically aroused when you win money as well. People use this strategy and it reduces their conditioned response. It reduces their arousal response, their anticipation of reward response. And we also see changes in the striatum response. So here, I'm reducing the striatum response when you are regulating versus attending, and engages the dorsolateral prefrontal cortex. Again, this is a region that shows more activity when you have to engage in the executive control needed to regulate emotional response. So again, a similar circuitry. The striatum is involved in this anticipation of reward. The ventral medial prefrontal cortex can inhibit the striatum. I didn't show you that data, but again, it has strong connectivity with the striatum, and the dorsolateral prefrontal cortex plays a role in the cognitive strategy you use to reduce your anticipation. So how does this play into our decision-making task? Well, we did the exact same task I described earlier, but for one set of trials, we said, we want you to attend to each choice that you make. Think about this as your only choice in isolation. You're just making this one choice. For the other one, we said, we want you to regulate your emotional response. Think like a trader. Imagine you're assembling a portfolio of many choices. And again, we measured these responses. We measured loss aversion, risk sensitivity, and consistency. And this is the data I showed you earlier, same participants. Now I'm showing you the same participants in the same order, but the decrease in loss aversion when they were then regulating their emotional response. Okay, so most participants, this is the percent decrease in lambda. Most participants were able to use this strategy to decrease their emotional response. When I looked at individual skin conductance, here we see this again, increase in arousal to losses relative to gains, but that wasn't there when these participants regulated their emotional response. So they also regulated their arousal response. And we saw this difference in the amygdala, again, that was correlated with their reduction in arousal from attend to regulate. Okay, so again, we reduced the amygdala bold response as participants regulated their emotional response. And finally, we saw this engagement of the entire circuitry of emotion regulation as well when they used the regulation strategy. So in the cognitive regulation of decisions, we're using a similar circuitry as when we're regulating our threat reactions. And very briefly, I want to get to the last version. Oh, and just to sort of say, what's the real world implication of this? This was a paper that was done by Andrew Lowe, and he showed that physiological arousal responses to market events correlates with poor performance in traders. And he argued that maybe this is because, some people were just born to be less reactive. It could be that experienced traders actually just learn to regulate their emotional responses. And this leads to better performance in these traders. All right. So last thing I want to talk about very briefly is, can we also reduce loss aversion by pharmacologically reducing arousal? And we know that when the amygdala modulates the striatum, at least in terms of its memory representation, this is due to the neurohormonal changes that occur with arousal. Essentially, the adrenaline release that occurs, the epinephrine, and the neuroadrenergic system. So we gave participants a selective beta-adrenergic receptor antagonist, a beta blocker. This is highly lipophilic. That's relevant because it passes the blood-brain barrier, but it also relates to kind of your body weight. So it acts centrally and peripherally, but it has a dose-dependent pharmacokinetics. So individuals with higher body mass will experience lower peak concentrations of this drug. So it blunts arousal without a sedative effect, and we expect a stronger effect in smaller individuals. So individuals were given the drug or placebo on 2 separate days, about a week apart. Again, we did the same task, measured loss aversion, risk sensitivity, and consistency. And I should just point out, people were about 47% correct in guessing which drug, whether they got drug or placebo. In other words, they were at chance. So what we measured is the change in loss aversion from the day they were on the drug to the day they were on the placebo. And across all participants, we didn't see a difference. But if we now separated individuals out by their body mass index, essentially people that would be technically overweight versus not, we saw a difference. So those individuals with a lower body mass index that got a higher dose of the drug showed a significant change in loss aversion when they were on the drug. They reduced their loss aversion when they were on the drug. Again, there was no impact on risk, sensitivity, or choice consistency. So what this suggests to us is propranolol reduces loss aversion in a dose-dependent manner, no effect on risk attitudes or consistency. This corroborates studies linking loss aversion to arousal, to amygdalostriatal circuits, and to the adrenergic system. And it aligns with studies from other domains concerning the amygdalostriatal systems in terms of avoidance actions and the adrenergic system. So what I've done today is I've taken on sort of inspiration from what we know about threat conditioning and the circuitry related to threat and avoidance, and I've applied it to monetary loss. And I've suggested that specific affective responses modulate your response to components of a choice, and arousal is specifically linked to loss aversion, the threat of loss aversion. And that changing emotion can also change the choice. So if I change arousal either via cognitive regulation or pharmacology, I can reduce loss aversion. And I just want to point out this was work by Peter Sokol-Hessner when he was a grad student in my lab. He's now a professor at University of Denver. And Colin Kammerer, a behavioral economist who's at Caltech and funded by the National Institute of Aging. Thank you. I'll take questions.
Thank you, Liz. That was great. Christine has a question. I'll start with her.
I do. Thanks so much. That was really interesting. So in terms of, the kind of top-down regulation of loss aversion with cognitive control, that kind of makes me think that there might be different timescales on which these systems might be operating. And I'm not sure that the particular paradigm you presented would let you assay reaction time, but can you speak to that? Do you think if you gave subjects kind of longer periods of time to respond, that would also have an effect on loss aversion?
Meaning, well, 2 things. So this particular task was asking on a trial-by-trial basis, attend versus regulate, attend versus regulate. That's very different than what you would do. So it suggests people can flexibly use them. But if you were to go to cognitive therapy, they would try to teach you to regulate kind of always, right? They would try to make that your habit, right? Not kind of go back and forth and flexibly decide what I'm going to do trial by trial. And so that's, I think, more likely what we talked about in the sort of trader domain, right? These are people that have learned not to overreact to bad situations in the market. So this is not quite getting at your question, I think, in the sense that we're not looking at super fast responses. But yeah, I mean, I think getting more of what you're trying to get out, and just correct me if I'm wrong, is if I didn't give people time to have the top-down regulation, would you not see this effect? I suspect that would be the case, but that would be forcing them to make a very, very quick decision. That would be kind of, I would wonder they'd really fully be able to process the entire decision in that time frame. I should point out, however, that in this particular experiment, we gave them the 2 things. We gave them the cue before they saw the choice. So we were trying to get them into that mindset before they saw the choice itself, right? So, and in the decision-making task, at least, I should say we blocked them. So we gave them the cue and then we blocked them. So like 10 trials each. In the actual emotion regulation task, we gave them the cue right before the stimulus, and it was trial by trial. So we were giving them an advanced opportunity to engage in that cognitive mindset of, how I'm going to approach this situation. But if we did it very, very quickly, would it be hard to engage the cognitive strategy? I think it would be. We haven't done that.
Thank you, Liz. So I'll ask, does the same sort of rule apply in children? So before the frontal cortices are fully developed, because it makes sense to train your whole society to be able to do this sort of regulation. School is a great time to do it, but when's the right time?
Great question. And this is a big, question of people who study the development of emotion, like how do you do emotion regulation, especially when the prefrontal cortex connectivity with these brain regions is developing. So most of the work that I know of has not been done on this types of decision tasks, but have been done on things like just responding, trying to reduce maladaptive responses to aversive pictures and things like that, sort of standard regulation. And what we know is there's a trajectory, right? So at least in terms of extinction learning, younger children look more like adults, and where you see differences are more in the sort of middle ages, teenage years, et cetera, where there might be some sort of, inverted U type function, but it may actually be that different systems are involved, at least in terms of regulating for the older adults and the younger children. They just look, the behavioral pattern looks more the same. In terms of cognitive regulation, obviously that's a lot more kind of complicated. And what I've heard developmental psychologists talk about is, a lot of these strategies are learned from parents. So parents are kind of emotion regulation, people. They are the ones who are guiding you on strategies you need to use and sort of providing the scaffolding of children developing their own strategies. So I think there's really two things. One is learning the strategies. Two is having the connectivity to work in the same way, which it doesn't. We know it's different across development, and that's really why we think we see many, many changes, in responses to things like risk and threatening responses across development. So, I think it would be different in children, but I can't say that we've looked at it or I know of anybody who has looked at it very explicitly.
Thank you. Okay, I'll ask one more follow-up question, and I think we'll have to thank the speakers. So are you aware of any computer implemented games for training this kind of thing? That would be another way to, to approach the topic.
Training regulation?
Yeah.
I mean, there are online, this type of regulation is basically what you do in cognitive therapy, right? And so the notion of sort of I don't know of any particular games, I should say. Like, I don't know of any, but there are many, many training games of things like working memory. The same regions we think are involved in the executive control for cognitive regulation are also involved in working memory. As far as I know, there's not good evidence of transfer for those types of things. In terms of cognitive, there may very well be kind of emotion regulation, cognitive training games. It seems like a pretty logical thing. I just don't know of any.
Okay. Well, thank you. Mindful of the time. If there are no other burning questions, I just want to thank you for speakers. This was a really fantastic symposium for me. I don't work in any of these areas, but it's an extremely ripe and creative area, and thank you for sharing your work.
Simulations to look at the contributive factors for this donut shape. We assume that the brain averages over 3 guesses based on 3 distinct sources of information provided in the paradigm. The first 2 being the memory of the target in ego and the allo coordinates, and the third being the prediction that can be made during the initial target presentation in a quasi-predictable fashion. The resultant average estimation of the desired gaze goal is then weighted with the learned reward function. And what we get is a distribution which is very similar to the 2-dimensional projection of the data as you see in the middle. And if we take one of these components away, we lose this donut shape. So we recorded from the supplementary and the frontal eye field simultaneously. The green corresponds to the supplementary eye fields and the gray corresponds to the frontal eye fields. The red and the blue dots, they correspond to recording sites from both animals. We recorded 68 neurons from the supplementary eye fields, and that we included for this analysis, and 147 spatially tuned neurons from the frontal eye fields, and we further divided that into visual, motor, and the motor neurons. To note that many of these neurons have delay activity, and here we are specifically analyzing the activity from the VR onset, which means the visual response onset, which is indicated by the dotted blue line. And we analyzed it until the end of the landmark shift or the mask off. The green shaded area corresponds to the area analyzed, to the time period analyzed in this study. So we are interested in the underlying neural codes, and how we do that is we plot the response fields in different coordinate systems, and we fit neural data against different spatial models and computed residuals, responses, and fits based on these spatial models. So here, the pink is the fit to the blue data. And if a response field is plotted in the best coordinates, it will lead to the least residuals, or if it is, on the other hand, if it is plotted in an inferior coordinate or an incorrect frame, it will lead to the higher residuals. So to the left is the best coordinate system for a given neuron. We have recently found in the frontal and the supplementary eye fields that the visual response codes for the target relative to the eye, whereas the motor response codes for the future gaze relative to the eye. And most of the transitions occur between this continuum, between the target and the gaze. So we divided this space that is the target and gaze, which is essentially the error line between the target and gaze, into 10 different steps, and see where the response field fits best along this continuum. Here's an example of an SCF neuron. The blue line corresponds to the target on. The vertical broken blue line corresponds to the visual response onset. Every little tick here is an action potential, and the red curve is the spike density plot. And we took the area from, so the pink shaded area is the mask, with the duration of the mask in our task. And we took the area, as we showed you in the previous slide, from the time from the visual response onset until the landmark shift, and divided that into 7 half overlapping beans. This green block here corresponds to one bean, and see where exactly in these beans the response fields is best fitted. So we then took the target to gaze continuum and see where exactly along this continuum the response field fits the best. Here is an example of response field. Every circle here corresponds to the size of the response, the magnitude of the response. The bigger the circle, the higher the response, and the smaller the circle, the least the firing frequency of the neuron. And the heat spot corresponds to the non-parametric fit to these data. And here, along this target to gaze continuum, this response field fits best at 8th step from target. That is very close to gaze.
1 minute left, sorry.
And this corresponds to the time step 5th. That means along the target to gaze, this response is fitting best at 8th step, but at the 5th time bin, which almost corresponds to the time step just before the mask onset. That is, it in a way is predicting the future gaze. And then we looked at the population. To the left is the spike density plot from all the neurons from the supplementary eye fields, and in the middle is the spike density plot of all the frontal eye field neurons. And on the x-axis is the time steps, the half overlapping time steps, and on the y-axis is the spikes per second. And to the right is the spatial code evolution with time. So along the y-axis is the target to the gaze continuum. The gray curve is the frontal eye field spatial co-devolution, and the green curve is the supplementary eye field spatial co-devolution. Every solid dot here corresponds to a significant shift from time point from point T, that is from the target. So here you can see that the frontal eye field, the evolution of the population code is gradual in the frontal eye fields, but for the supplementary eye fields at the time step 5th, there is a predictive shift towards the future gaze direction. And overall, what this slide suggests that the supplementary eye field neurons display a predictive shift towards gaze just at the mask onset, whereas frontal eye fields do not. So in summary and conclusion, the donut-like gaze distribution is attributed to the weighting between the ego, allo, prediction, and the reward components. The SCF neural activity predicts the future gaze, whereas the FVF does not. The future directions, we will investigate the effect of independent landmarks on gaze behavior and the corresponding codes in the neural activity. Thank you.
Thank you for the wonderful talk. So we are waiting for some questions if they will appear. Otherwise, we have 1 minute for a question. And it's all really fascinating, but I was just wondering when I look at this study, what do you think would be the, I don't know, translational part of it? Translational application, maybe?
Well, we are already exploring that in a collaboration in Ottawa. So, like for example, there are many patients who lose their ability to use agocentric or allocentric information. So, by with these kind of tasks, or even like maybe with some like interventions where we can like stick electrodes in the brain, we can look at the underlying egocentric and allocentric mechanisms of binding in these patients. So this is one of the applications, but to further look into that, maybe we can get inspired from this paradigm, to train the subjects or the patients.
Yeah, great. Thank you very much again. And we're moving forward, and now we have second speaker, Veronika Natcher. And Veronika, she's a colleague of Vishal at York University. You can find Veronika's recent research in the chat. And you can check, we encourage you to check her Google Scholars. While now, we will learn more from Veronika about visual-motor transformations in ventral premotor cortex during head unrestrained reaches. Please, Veronika, go ahead.
Thank you, but Vishal hasn't unshared his screen, please.
Oh, yeah. I'm sorry, Yeah.
Yeah, okay. Okay, so thank you very much for this kind introduction. And I am going to talk about visuomotor transformations in ventral premotor cortex during hand unrestrained reaches. And this is an ongoing project done in Dr. Douglas Crawford Laboratory at York University. So in natural conditions, reaching involves a coordinate, reaching involves a coordinate sequence of gaze, head, and arm movements towards a visual target. And several studies have examined the eye, head, and hand coordination in the human, but the underlying mechanisms, especially those controlling head motion, have not been studied. So to this goal, we trained 2 monkeys in a reaching paradigm that allow an uncomputed head motion and reaching in depth, and we recorded the neural activity while monkeys performed the task. So when the animal arrives to the laboratory in a prime chair, we attach a LED bar on the waist level. The LED bar has 3 LEDs, and in front of the animal there is a touch screen. So a typical trial starts when one of the LEDs is lighted on, the animal places its hand on the LED and waits 700 milliseconds. After appears a fixation dot on the screen, and the animal has to fixate between 400 and 800 milliseconds. And as soon as the fixation dot disappears, a target appears on the screen, and the animal has to release the hand from the LED bar and touch the target. So in this task, the animal is rewarded for touching the target. So in this task, we have 3 different LEDs that can be, one of them lighted up from trial to trial in a random fashion. So we have 3 initial different hand positions. And also we have a different window here around the fixation dot of 7 degrees in which the fixation dot has a jittering. And also the animal is working with the head free. So we have different head positions. We have also studied a non-reaching task. It's a control task in which the trial the evolution is exactly the same. The animal uses the hand, but when the target is on the screen, the animal doesn't have to reach. The animal just have to look at the target and is rewarded by that. So we record gaze and head motion using a search coil and touchscreen technology. And we also are recording the extracellular single unit activity in the premotor ventral area, in the premotor dorsal cortex, and in the frontal eye fields. So we have a chamber that covers all these areas. And we have done recordings in 2 animals. In monkey W, this is a schematic recording of the chamber of the penetrated sites. It's gray dot, and the red dots are the ones who we could elicit evoke saccade using microstimulation. So we were mapping the frontal eye fields. So we obtain neural activity. This is an example for a hand touch event when the animal touches the screen. This is a raster. This is a replica of the neural activity. Each line is a trial and each dot is an action potential. And here we have the spine density plot. And what they're going to do. So for this neural activity in a specific time window, we are interested in to know what are the neurons encoding. What of the task parameters are the neurons encoding. So the first thing is to test for gaze, head, and hand gain fields during these different neural responses across the task. So a gain field effect is the scaling of a tune response by a postural signal. So remember that we had different initial hand locations, different initial eye positions, and different initial head positions. We can see how firing rate can change, is modulated by these postural effects. And after we feed residual data against various spatial models. So we use the same model fitting, and the main spatial models tested might fall into 4 groups. One group is related to the eye in blue. This is a schematic drawing of a subject. This is in orange, the head models. In gray, the body space. And in purple, the arm movements. So the types of neuronal responses that we have found in premotor ventral, this is the data that I am going to show you, just premotor ventral today. So we have found 3 groups of different responses. So here are the task events: target on, when the target is presented on the screen; gaze onset, when the animal looks at the target; hand onset, when the animal releases the hand from the LED bar; and hand touch, when the animal touches the target. So the first group of neurons increase firing rate abruptly after 80 milliseconds in latency after the presentation of the target. The same for gaze onset. And for hand onset, the firing rate is usually low or is decreasing. And these neurons are not responding when the animal touches the screen. The second group of neurons, we call it early-late. So these neurons increase firing rate for target presentation and maintain the firing rate increased until the animal touches the screen. And the last group of neurons, we call it late. And these neurons start to increase the response a little bit before the hand onset and reaching a peak when the animal touches the screen. So interestingly, we can compare this neural activity during the reaching and the not reaching task. So this is an example for a rich neuron, is an early neuron again. When we test the same neural activity during the controls, the neuronal data is not, the neuron is not responding. So it seems that if the neuron has a motor plan or something encoded here, when the control task is, the animal is working in the control task, the neurons are not responding. So this is an example for an early neuron, how we fit the models. So we will start with the target onset. This is the spike density plot and the neural activity in this window. So we are plotting receptive field maps. The colors are the number of spikes in the sampling window, and the dots are this is the representation of the targets on the screen, and the circles means the firing rate. The bigger the circle, the higher the firing rate. And we fit models, these spatial models, and this is the plot that represents the p-value of the models. This is our threshold. So all the models above the threshold are significant, the models below the threshold are not significant. In this particular case, for target onset, none of the models has a strong preference, but it was gaze in future eye coordinates, one of the top. But the situation changes. For the gaze onset, when the animal looks at the screen, just one model is significant. This model is the one that explains completely the data. So the model is displacement of the arm. It seems that when the target is on, the neuron is encoding a space or eye information, but as soon as the animal looks at the screen, the neuron is encoding the displacement of the arm. And it maintains this code during the hand onset, when the animal is releasing the hand from the LED bar at this point. Even more, if we study the gain field effects, we can see for this specific event, hand onset, how the receptive field responses are changing according to the different initial hand positions. So the neurons are encoding the displacement of the arm with these gain field effects. For the population results, we have these preliminary results for early neurons. So during the target onset, the neurons best encode space models. During the gaze onset, displacement of the arm, and during the hand onset, head in future space. For early late neurons, the situation changes during the target onset. Now, the neurons start to encode displacement of the arm, change for head in future space during the gaze onset, and during the hand onset is displacement of the arm. And for the late neurons, during the hand onset, target in space and arm in future space. So the conclusions are the following. So early premotor ventral responses initially encode after population analysis target during target presentation, but this was rapidly transformed into arm displacement code. And this rapid transformation occurred in a window ranging from 100 to 200 milliseconds. The rest of the neurons, early, late, and late, seems to prefer encode arm and head models. And these three types of neurons show initial hand position gain fields and sometimes gaze gain fields. And during control conditions in which reaching is not required, neurons are not responsive. And a more complete analysis will describe the complete coding and distribution of gaze, head, and hand and reach signals in this region. So thank you very much to my supervisor, Dr. Douglas Crawford, and all the lab members and co-authors, and the fundings, Canadian Institutes of Health Research and VISTA funding. Thank you very much.
Great talk. Thank you very much. It's
Thank
interesting. Do we have any questions? Okay, then I have a question. These three types of neurons, neural responses, do you think there is some cellular mechanism behind? Would it be different type of neurons? Would you be able to have to classify them not only based on the temporal aspect of their response, but I don't know, maybe there's something morphology or they express particular electrophysiological features?
So these three types of neurons are based on different response profiles across the task. And as you could, and I showed that, for example, during the target onset, when the stimulus is on the screen, early neurons encode the space models, right? But in the same event, a neuron that is early late is encoding displacement of the arm preferentially. So these neurons are, in principle, our hypothesis is that they are different. But a final population analysis with a complete database, there are just 271 neurons, we have more neurons, will give us a better response of what the neurons are doing. And also we are planning to do a spatiotemporal analysis to study all these dynamics from the all these visual motor transformations across these three different types of cells.
Okay.
I am planning also to see if during the penetrations of the cortex, we can find specific spots for this group of neurons, or all of them are intermixed in the cortex, in the premotor ventral.
Okay, thank you very much for this answer. We also have two questions that unfortunately I have no time to read them out, but please feel free to go to KA and answer them directly. We have two questions from Gunnar, and we move forward again.
Okay. Thank you very much.
Thanks again. It was wonderful. So now we have Ihsan Nijad, who is a PhD candidate and Reza Shadmehr Laboratory in Johns Hopkins School of Medicine. Please follow him on Twitter. You will find his Twitter handle in the chat, and also check out his latest paper. Without any further ado, please go on.
Okay, thank you very much, Maria, for introducing me. And I want to first thank the organizers, the NeuroMatch Academy. I also wear my NeuroMatch conference. I also wear my NeuroMatch Academy t-shirt here. I had a great time at NeuroMatch Academy, and I'm really grateful that they gave me the opportunity to present my work at NeuroMatch conference. Before I start, I should thank my great colleagues, Paul Hage and JP, that helped me in this work, and specifically my amazing mentor, Reza, that's kind of the backbone of the lab. So without further ado, so my talk today is about population coding of saccadic eye movements by Purkinje cells of the marmoset cerebellum. So my talk today will be about this cute, very kind of amazing and new animal, non-human animal models, namely marmoset monkeys. And I'm going to present the data that are recorded from cerebellum. So the other two talks were about the cortex. This talk is more about the cerebellum, and specifically about the Purkinje cells of the cerebellum. Just to give you a background, the cerebellum is where that we think is that responsible for encoding of the internal models and the motor commands. But a major difficulty in the field has been to understand the coding of the cerebral Purkinje cells are doing. Let me actually. Sorry. And so recently we published two papers in the Nature and Nature Neuroscience that we show the results that we can use a framework, and this framework is about the population coding of Purkinje cells, and how this population is getting formed is based on the preference for error of each Purkinje cell. And when we put the Purkinje cells based on the preference for error, then we can have a coordinate system, and using that coordinate system, we were able to make sense of the activity of the Purkinje cells. That work was in macaque monkeys. Here we expand that work, and we expanded to using marmoset monkeys. So first of all, we want to prove that marmoset monkeys are reliable and very promising new non-human animal models for studying sensory-motor learning and specifically saccadic eye movements. And we test kind of the generality of our hypotheses. So we record from Purkinje cells of the cerebellum and see if we can use the same coordinate system and the same framework to make sense of the activity of the Purkinje cells of the marmoset cerebellum. So we previously published our findings about how to train marmoset monkeys, how to do surgery on them, and also how to, because there is no reliable brain atlas for marmoset monkeys right now, kind of general use like what we have for macaque and also the mouse. But we also introduced how we can come up with a very accurate targeting of different brain areas. Please go ahead and read that paper. Anyway, okay, so now let's focus here on our task. So our task is a saccadic eye movement task. What we do is that we'll present a center target, and the monkey is tasked to fixate at the center target for about 200 milliseconds. After the fixation point, we'll make a sound, and then we'll jump the target to a new location. So this target will disappear, a new target will appear, as you can see here, and the monkey is tasked to make a primary saccade. When we detect the onset of the primary saccade, we jump the target to a new location. So this will disappear, and target will go to a new location. So what will happen is that since we do the secondary jump at the saccade onset, so this will happen during when the eye is kind of in the travel. So, and as you know, the eye is basically blind during the saccade, so the monkey will not see the second jump. And when the eye lands at this location, the target is no longer there, it's a new location. And this is what we call the sensory predictioner, because the monkey predicts that the target should be there, but the target is a new location. And as a result of this secondary jump, and the monkey will make another saccade, what we call a corrective saccade. And monkey's task is to fixate at the secondary location for 200 milliseconds, and after that, the monkey will receive its reward. Here is the eye trajectories for a sample session. Monkey is fixating at the center point. The target is jumping to one of the randomly 8 choose locations, and the monkey makes the primary saccade. Then after that, we again jump the target to one of the 8 new locations, and the monkey will make a corrective saccade. Okay, while the monkey is doing these tasks, the saccadic eye movement tasks, we're recording from the cerebellum. And we are specifically recording from a specific type of cells, and those cells are Purkinje cells. And how we define that we are recording actually from Purkinje cells is by using the simple spike and complex spikes. So we are detecting these complex spikes, and these complex spikes are the unique signature of Purkinje cells. And using these complex spikes, seeing that we have the complex spikes is a proof that we are actually recording from Purkinje cells. Okay, so what is simple spike and what is a complex spike? So simple spikes are the usual spikes that other cells also have. They are about between 50 hertz to 60 hertz. And one of the signatures of the simple spike of the Purkinje cells is that they had this kind of extra bump after the refractory period. But what is unique about Purkinje cells, as I told you, is that they also have complex spikes. Complex spikes are complex, but these are unique spikes. These are rare spikes, so they fire about 1 hertz, just 1 hertz. So every second or every other second, you get one of those. And they're actually getting produced by a different mechanism in the cell. So as simple spikes are the usual sodium-potassium channels, complex spikes are getting generated by calcium channels. And the way to make sure that you're recording from a Purkinje cell is by what we call a suppression in the simple spike due to a complex spike. So what I plot for you here is a cross probability. At time 0, we had a complex spike. And what you can see here is that after happening of a complex spike,
there is a longer refractory period for a simple spike, and simple spike, the cell does not fire any simple spike for a longer period of time. The same data has been plotted here. So what you can see is that at this time, we have all the complex spike. Before a complex spike happening, you can have simple spike all the way up to the complex spike. But after complex spike is happening, we have a long period of time of the silence. And then after that period, simple spikes start to fire and coming up. So we use these simple spike and complex spike framework to make sure that we are actually recording from Purkinje cells. Okay, great. Now let's focus on the encoding of the ongoing saccade in the cerebellum. So first, what I'm plotting for you here is 8 directions that I show you as a sample session. So we had 8 directions. I marked for you the saccade onset and saccade offset. If I plot the raster plots for simple spikes, what you can see here is that this Purkinje cell, the sample Purkinje cell, do actually show modulation for saccadic hive movements. But if you zoom in a little bit more, what you can see is that the activity of this Purkinje cell is very complex and it's not consistent. For example, for some direction is bursting before the saccade and pausing during the saccade. For other direction is pausing and bursting. In some direction it's just pausing without any bursting. And it's very complex. So there is no consistent kind of behavior of the cell for different directions. And this has been a very, very challenging task in the field, that by looking just at the Purkinje cell, there is no very clear understanding of what does this Purkinje cell is doing during the saccade. But the Purkinje cells does not just have simple spike, they also have complex spike. Now, what I'm plotting here for you is the complex spikes of this Purkinje cell. If you now look at the complex spike activity, what you can see here is that the complex spikes in the right saccades are having more probability of firing than the left saccade. And if you put them as a tuning, what you can see for this cell is that this cell is actually have a complex spike tuning for rightward saccade. So although the simple spike might be very complex and does not have a very unique way of encoding, and complex spikes do show directional tuning for different directions. Okay, now using this framework, let's look at the population of Purkinje cells. So now in this slide, I'm going to show you the complex spike population tuning or coding, and then the next slide I will show you the simple spike coding. So as I mentioned, for each cell we detect the direction that cell has the maximum probability of firing a complex spike, and we call that direction CS on. And of course, since we define the CS on as the maximum probability, you will have the maximum probability of CS probability in the CS on. But what is happening is that the direction opposite to CS on will always have a reduction lower than the average firing rate of the complex spike. And so the CS off, just by aligning things to CS on, you will get a CS off or CS 180 that will has lower than the average firing for the complex spike. Now let's focus more on the CS on direction. So what we saw is that after presenting the primary stimulus, we saw an increase in the firing of the complex spike. And if we now align the same data to the saccade onset, so not just presenting this, but when the saccade is actually happening, what we saw is that right after the saccade onset, although that we had an increase in the complex spike firing rate before the saccade, just right after the saccade onset, the complex spike will go to the baseline, and after the saccade, it will go all the way even more down. Now, if I focus on the corrective saccade, we saw again the same thing, that we had increase in the activity after experience of the sensory error, and going to the baseline during the saccade and going below the baseline after the saccade finish. Now, if I this was for CS on. If we focus on CS off, and what we see is that, so this is the same thing just for reference, what we see is that in the CS on, we actually see a decrease in the complex spike firing when we experience the sensory error. Okay, great. So we had this framework. Now, if we put all these Purkinje cells at the population all together, what we see is that as a population, we can see that the Purkinje cells show an increase in their activity before saccade onset, and during the saccade, they will decrease their activity, or we had this disinhibition of activity of Purkinje cells. But what is interesting is that if we now use our
30
framework,
seconds, really. Please, we're short on time.
Okay, I will just finish here. If we use this framework, we see that CS off or CS 180 show the maximum firing, although CS on does not show that. So overall, I show you the results that the marmoset cerebrum is a reliable animal model for the sensory motor learning. We recorded from 110 Purkinje cells. When we put the population of the Purkinje cells and when we account for the preference for the error, then we were able to see the SS response of the Purkinje cells. Thank you very much.
Okay, thank you. That's great. Cerebellum is awesome. And we have one question, really one, and really short. Yeah, please. How difficult to train marmosets in this task compared to macaques? This is a question from Edmund.
That's a great question. So in our 2019 paper, we explain in great details how we trained them to do more than 1,000, and all the way we had a monkey that do 3,000 trial per session. We spent about 1 month for training them on this saccade task. So I would say they're very convenient and very comparable to macaque monkeys.
Okay, cool. So let's have now one really quick one because I thought that we'll have less time. Question from Michelle.
Yeah, where do you think the input for this complex spike is coming from? And do you have any circuit for that?
So complex spikes, the input for complex spikes come from inferior olive, and inferior olive receives inputs from all over the brain. And this is an ongoing kind of research. But the simplest spikes and complex spikes actually have different inputs. So simplest spikes, the input for that come from mossy fibers, but the input for complex spikes come from inferior olive. So they also have different inputs. Yeah.
Okay, cool. Thank you.
Okay. Thank you very much. It's like amazing session, and I wish we had more time for your talks and for questions. I hope to hear more about your studies soon, and please, audience, follow our amazing speakers, ask them more questions, and I just can wish you a nice evening or day or whatever you have in the place where you are. Thank you very much again.
Thank you very much.
Bye-bye.
Bye-bye.
So welcome to NeuroMatch. I'm Andre Fenton. I'm a professor of neuroscience at the Center for Neuroscience at New York University. This is a symposium that I'm very excited about. I have friends and colleagues and people who I've been following the work of for quite some time, and they're gathered here in this symposium that we've titled Cellular and Molecular Mechanisms of Memory, Allocation, and Storage. Just to introduce you to the session rules in case you're not that familiar, please ask. We invite you to have a vibrant discussion, so please ask questions in the question and answer. You can type them in there. If you want to ask the question live, we'll bring you up in a Zoom box, and you can ask the questions directly if you raise your hand. Otherwise, I will ask the questions on your behalf. And please, if you don't mind, reserve the questions for after the 20-minute presentation. We'll have a full 10 minutes for discussion. So with that, I'll start the symposium. It's my pleasure to introduce Professor Jingji Lin. She's a professor of psychiatry and behavioral sciences at SUNY Upstate Medical University. and she'll be talking to us about memory allocation by transcriptionally defined neuronal ensembles. Welcome.
Sorry. Can you hear me now?
Yes, we can.
All right. Sorry. I'd like to thank Andre and NeuroMatch for organizing such an exciting symposium. As you know, memory is the mental capacity to encode, store, and retrieve information of our past experiences. So I think we still don't have a full picture of what really constitutes memories at a cellular level. But we and many others in the field are focused on the so-called active ensemble neurons as strong candidates for the sites of memory encoding. This is because that these neurons are activated during learning, and they undergo long-lasting synaptic changes as a result of learning. And they have been shown that they are essential for expressing the encoded memory. And these neurons sometimes are called inguine cells, as Steve, our next speaker, will like to call them. They're inguine cells. So when a neuron is activated, a series of cellular responses are triggered that can be used to identify and capture these neurons. For example, synaptic transmission will trigger membrane depolarization and calcium influx, which can be monitored by electrophysiology recording and calcium imaging. And for long-lasting cellular changes triggered by neuronal activity, the signals at the synapses are transmitted from the synapses to the nucleus to turn on gene expression. Showing here is an example of one of probably the most famous such genes that are activated by neuron activity, CFOS. So FOS actually belongs to this group of genes called immediate early genes, or IEGs. And this group of genes has very nice features. So they are induced very rapidly within minutes and very robustly. And that activation is also rather transient. So their induction has some kind of nice temporal resolution. So if you look at the group of these genes, let me change my pointer. If you look at this group of genes, you'll notice that the majority of the IEGs are transcription factors. So they can turn on the expression of second wave of genes. And this group of genes can function directly to modify neural circuit at a variety of different ways. So because of this nice features of the IEGs, and the activation of IEGs have been used to identify active neuronal ensembles. So this approach has proven to be quite fruitful. So works from many labs, including that of Mark Mayford, Arsenio Silva, Sheena Josling, and Susumu Kongawa, including Steve's early work, have provided many very exciting insights into cellular mechanisms of memory allocation and storage. Here, though, I want to draw your attention to the fact that each IEG actually represents a distinct activity-dependent transcription pathways that can lead to very distinct and different downstream effects in neural circuits. So let's look at two of them, FOS and MPAS4. FOS, being the most famous IEG, has been shown to regulate neuronal excitability, and it's required for LTP. On the other hand, MPAS4 is an IEG that I identified when I was a postdoc and continue to study in my lab. And MPAS4 seems to function rather differently from FOS, and it often recruits inhibitory synapses onto principal neurons. So some of our early work also suggests that FOS and MPAS4 can be activated, induced in different populations of neurons. For example, after contextual field conditioning, as you can see, that MPAS4 induction is largely highly enriched in the CA3 regions, also a little bit in DG, which you cannot see here, but it's largely absent from CA1. This is very different from FOS, which is induced throughout the whole hippocampus. So these early observations suggest that memory allocation can be very heterogeneous. So this is more like the picture. And so there may be different active neuronal ensembles that can have very distinct functions within the memory inlet. So in order to really to test this, to prove whether this is the case, Andreas and Yoni in my lab led the effort to develop the robust activity marking system, the RAM system. So using the RAM system, we are able to design, develop a series of different RAM reporters. So at the heart of each RAM reporter is a RAM promoter. The RAM promoter is activated by the binding of one of the IEGs in activated neurons. So the RAM promoter drives the expression of reporter molecules within to be expressed within the active ensemble neurons. The RAM reporters also have a temporal control system, which I won't get into the details, which prevents the expression of reporter molecules unless it is within the designed experimental window. So by designing a different, so before I get ahead, I want to show you an example. So this is what an example of the active ensemble neurons labeled by one of the RAM reporters in DG after contextual field conditioning. So consistent with the sparse encoding in DG, and the RAM reporter labels, this reporter labels about 2% of the DG granule cells. So by designing different RAM reporter that can be activated by different IEGs, we were able to investigate whether indeed there are different active neuronal ensembles that represent the same memory, and whether these ensembles have very distinct functions. So Xiaochen Shang, a very talented graduate student in my lab, decided to test this hypothesis in the dentate gyrus. And he worked together closely with Max and Xiaochen, and three of them form a very productive team. So because we know DG is a region known to be involved in memory discrimination, so they designed a very simple memory discrimination assay. Mice were contextual field conditioned in context A. So 1 or 2 days later, their contextual memory was tested when they are exposed to the same context, context A, or in a very similar but different context, context B, or in a neutral context, context C. So you can see from the freezing levels, the animals' freezing levels in these three different contexts, and the animals can really tell these three contexts apart, and they freeze at high level in A because they remember this is where they got shot. And the freezing in context C is very low because this is a neutral context, it's considered safe. They freeze very high in B because they realize that B is very similar to A, so it could be dangerous. But they also realize that B is not the same as A, so perhaps it's not as dangerous. So the freezing level in B is significantly lower in A. So you can plot out that freezing level as a function of the similarity between the memory recall context and the condition context. what we hoped to find and define was that some ensembles will function to push the curve to the right to improve discrimination, while others will push the curve to the left to promote generation. So it turns out that neurons are defined by the activation of MPAS4, which I color-coded here in blue, are important for memory discrimination. We call them NREM neurons. So because they are labeled captured by the MPAS4 dependent REM reporter, NREM reporter. On the other hand, the fast dependent active ensemble, the FREN ensemble neurons, they are, I'm going to show you, they are important for memory generalization, and I color-coded them in red. So our first clue that that the FREN and NREM ensembles can have very distinct function came when we measure their electrophysiology properties on those neurons. And it turns out that FREN neurons receive much stronger excitatory inputs compared to their neighbors that are not labeled, or we call them FREN negative neurons. As you can see, the mini EPSC frequency on FREN positive neurons are much higher. It's much higher than the FREN negative neurons. And the inhibitory inputs between these two groups are very similar. NREM, though, it's very different. And the excitatory inputs on NREM positive or NREM negative neurons are very similar, but the NREM neurons receive much stronger inhibitory inputs. As you can see, measured by they show much higher mini iPSC frequencies. So we've done a lot of experiments, which I don't have time to show you, which I'm just going to summarize here. So we show that the FREN and NREM ensembles are largely non-overlapping neuronal populations, which is not surprising given that they're very distinct electrophysiology properties. And their electrophysiology properties are actually induced by learning. And we also found that the FREN and NREM ensembles have very distinct activity patterns during memory discrimination. So now the question is, do they have very different functions? So in order to do that, we decided to manipulate their activities during memory discrimination. And so we've done both excitation and inhibition experiments, but I'm going to show you just inhibition experiments. So when we inhibited FREM during memory recall, what we found is the discrimination between A and B was greatly improved. You can see based on the freezing levels in A and B, and also by the discrimination index shown here on the right, calculated using this formula. This suggests that probably normally FREM ensemble functions to push towards generalization. NREM, when we inhibit NREM, we got a very different result. The discrimination between A and B is completely abolished. So which suggests that NREM norm is required for memory discrimination. Consistent with DG is really only required for discrimination between really two similar contexts, but not the contexts are very distinct. The discrimination between A and C was not affected by these manipulations. So next, we wanted to figure out how actually FREN and NREM ensembles achieve their very distinct functions. Remember, I showed you that the FREN neurons receive much stronger excitatory inputs. Within DG, each DG granule cell receive excitatory inputs from three major areas: the lateral entorhinal cortex, LEC, or the medial entorhinal cortex, MEC, or the hilus. So we wondered which one of these inputs have been strengthened on the FREN neurons. So Xiao Chen used dual patch, which means he patched simultaneously on a pair of afferent positive and afferent negative neurons, and recorded the excitatory postsynaptic current EPSC simultaneously on those pairs of neurons when he stimulated one of these three areas inputs. What he found is that it is the input from MEC that is selectively strengthened on the afferent positive neurons. So this result suggests that the MEC input into DG could be could function the important for afferent that could drive memory generation. So Xiaochen went ahead to inhibited the MEC during memory discrimination, and he found that just like what we saw when we inhibited afferent ensemble, when he inhibited MEC, the discrimination between A and B is significantly improved. And when LEC is inhibited, there's no effect on the memory discrimination. So how about NREM? Remember, NREM is a different story, and it receives much stronger inhibitory inputs. And there are so many different GABAergic interneurons in DG. So here, there are PV and CCK and SST are the three major classes. So we wonder whether the NREM neurons selectively recruit inhibition on any one of these subclasses of GABAergic neurons. So we expressed, Xiaochen expressed channelrhodopsin in all the GABAergic interneurons so that he could use light to activate all the GABAergic neurons and record optically induced inhibitory postsynaptic currents on pairs of NREM positive and NREM negative neurons. So as expected, that the NREM neurons receive much stronger, the iPSC frequent amplitude is much stronger, larger in NREM neurons. And when he did the experiment in the presence of blockers, the two different blockers blocks the input from CCK interneurons, this difference is completely abolished, which is not the case when he blocked the PV inhibiting neurons, PV expressing interneurons. So this result suggests that now that NREM neurons selectively recruit inhibitory inputs from CCK interneurons. It also suggests that CCK interneurons may be important for memory discrimination, which had never been examined before. So because CCK is also expressed in some excitatory neurons, so Xiaochen used a genetic intersection strategy to selectively target CCK expressing interneurons in CCK Cui mice. So when the CCK expressing interneurons were inhibited, and it completely mimicked what we saw with inhibition of NREM neurons, the discrimination between A and B context was completely abolished. So suggesting the CCK neurons are required for memory discrimination. And when we inhibit either PV or SST, the discrimination was not affected. So this brings me to the summary slide that I hope I convinced you that memory is allocated very heterogeneously among distinct neuronal ensembles that can be defined by different activity-dependent transcription pathways. So within DG, the contextual memory is encoded in at least two different ensembles, the NREM ensemble and the afferent ensemble and the NREM ensembles. And learning selectively strengthens the input from MEC to the afferent ensemble neurons. On the other hand, and learning selectively recruits CCK inhibition onto NREM neurons. And the MEC and afferent pathway promotes memory generation, and memory discrimination requires the activity of the NREN neurons and the CCK interneurons. So I'd like to acknowledge these are the folks in my lab that are involved in the work that I just showed you. In particular, Xiao Chen carried out the majority of the work that are in the DG discrimination generation work. It's a huge study, and he really did a great job. And he just graduated and started his postdoc with Mark Snitzer, I think last month. And Andrea and Yoni really led the effort to develop the RAN system, which proved was very critical for all the things that we wanted to do. Andreas now runs his own lab at University of Copenhagen, and Yoni is in the middle of his MD PhD at UCLA, and our collaborators and our funding agents. And I also like to do an advertisement if you are interested in what we are doing, and please check us out, and even better, join us. And thank you. Am I on time?
That was wonderful. Thank you. I remind the audience, please put your questions in the chat or raise your hand to come forward. I'll start with a general question. From the immunohistochemistry that we saw, there seemed to be specialization in different compartments, if you will, of the dentate gyrus. For example, the CCK neurons seem to have their cell bodies in the hilus. In the Sorensen image, it looked to me that the NPAS4 positive cells were specifically expressed in CA3 and so forth. To what extent are those sub-region specific features reproduced across the different subregions? So for example, a CCK neuron in the hilus, if you patch a CCK neuron anywhere else, would it have the same electrophysiological properties? Are these features generalized across the hippocampus, or are they location specific or subcircuit specific? Thank you.
So in terms of the CCK, I mean, like it's just the nature of interneurons. So we call them CCK because that's the marker we use in different regions, and they tend to sometimes have different subtypes. Sort of, they're all CCK, but they have different subtle features. And one thing very unique about the the CCK neurons in dentate, they really synapse onto this very tight band onto the inner molecular layer on the dentate fields, and which is coincide actually very interesting. This is something that we haven't really addressed very much. It's the same region that's targeted by Morphy cells. In a way, it's very interesting. We were interested to explore whether there's any it's only coincidence, or if there's some it's scientific biologically significant, something like that. And also in DG, the CCK neurons are the only ones that have expressed at the terminal, the CB1 receptors. So a lot of the features are the unique features of CCK neurons are depends on the endocannabinoid pathway. So there are a lot of very interesting features about those CCK interneurons in DG that we are interested in further explore.
Cool. Okay, so another question then. Do you have any evidence that once a neuron for this particular memory is, if you will, allocated by, let's say, NPAS4 expression or FOS expression, does that neuron stay that way? So if you had another memory, another experience, could it switch its affiliation, or is it a neuron committed to being inhibited through the NPAS4 mechanism, or excited through the FOS mechanism? Do you have any sense of that?
So it is a question that we wanted to address, because one thing it's basically those features intrinsic or extrinsic, right? So it is a very important question, and I think in general we assume that the allocation is random, but we don't know. Actually, nobody has done the experiments. I think we are actually in the middle of some experiments to address those questions and see whether it's not easy to address, but I think we have a way to really address and to see whether it's really intrinsic or extrinsic. And in terms of different memories, I think in a way, you will imagine, because I think the manipulations we do, we optimize our conditions so that we can sort of, we deprive the animal so much so that we can sort of really isolate the single event that we can manipulate. It is a little bit artificial, may or may not apply to what's happening in the real world, but we are limited by what we can do, right? We wanted to sort of have the selectivity, specificity that associated with our analysis. And so you will imagine that I actually think it's likely, but I don't know, that the different neurons not every neurons are born similarly. So they might have different features that allow them to be segregated into one pathway versus another pathway. And it's still an open question whether all the neurons have the ability to be sort of recruited to encode the memory, right? Where is the capacity of the whole? I mean, again, we are using a very low resolution method, to really say, oh, this neuron is involved and those neurons are not involved. But in reality, it's a lot more complicated than it's not a black and white, right? It's not a yes and no answer, rather than how they are subtly tuned to different episodes of memories.
Okay, great. Well, I'll ask one last question in our last minute. So have you looked at extinction? So when people talk about extinction, they talk about the memory actually being preserved and, if you will, being suppressed, if you will, by the extinction memory itself. You have a powerful system to be able to evaluate that, because you can actually measure whether the physiological change is altered by extinction. Have you explored that?
We haven't, but great question. This is the question, the experiments I wanted Xiaochen to do, which he didn't get to do before he left. Also impacted by the pandemic, unfortunately. But it is a great question. It's something that we would like to address.
Okay, and a question just came into the chat. How much overlap, if any, is there between the FRAM and NRAM neurons?
What did you say?
Be the last. How much overlap is there between the FRAM and the NRAM neurons?
Yeah, it's a very tough question to address. I'll tell you why. And because the system we are doing, but although for the ongoing experiment, we might be able to give you an answer, hopefully within a year or so. But the reported system didn't allow us to label the two ensembles simultaneously. So that's sort of, so our analysis is truly based on, which I don't have time to show you, but based on their very distinct electrophysiology property, you imagine they are not, the overlap cannot be very significant. But we also did another experiment where we look at the overlap between either FRAM or NRAM with the ensembles labeled by the false trap mouse. And that way, and because one is in the false trap mouse, what you have is the neurons are labeled by the activation of false, and the FRAM neurons are labeled by sort of downstream effect of false. So there we saw a very significant, like 50% overlap in that way, but they're not identical. And NRAM and the false trap ensemble, the overlap is very minimal, like 5 or 6% in our hands. So suggesting that we cannot say they are sort of black and white, totally no overlap, but it largely no overlap.
Great. Well, thank you very much for a wonderful talk. And it's time now to move to our next speaker, Steve Ramirez. He's an assistant professor in the Department of Psychological and Brain Sciences at Boston University. And he'll talk to us about social transmission, I believe, of fear memory engrams. Steve, the Zoom room is yours.
Great, thank you very much. And can everybody hear me okay? Great. Thank you, Andre, so much for organizing all of this. Like it really is a privilege to be here up with you guys. And I want to say that I am having this kind of Lion King circle of life moment right now, because it was almost exactly 10 years ago that I was rotating in Yingqi's lab as a graduate student. So it's really amazing to be able to be here and share the stage with her. And Yingqi, if you still have lab openings, I'd love to join. It'd be fun to do science again. So I want to split this talk into 3 brief sections. I want to start off with like a little bit more big picture of what does it mean to go in and start causally probing neural circuits. And then I'll go into some data about an ongoing project that we're hoping to submit in a few weeks on the social transmission of fear engrams. And then if we have enough time, I do want to end big picture with what does this all mean? Like, why do these experiments work? Why does activating these cells in very artificial patterns, why does it still yield a behavioral response? So I kind of want to end with trying to at least tie a little bit of the field together and see where we're going for the next decade. So when I was a first-year grad student, I read this really influential review from Richard Morris's lab that really began to set the criteria of what it means to go in and start causally probing the neural basis of memory. And the word causality gets thrown around a lot these days, right? Especially with opto and 2P imaging in the works. They at least propose that most experiments can be demarcated into 3 different styles of experiments. So one, for instance, is we can go in and say that learning happens in the brain, and areas like the hippocampus and plenty of other areas start lighting up with activity. And we can go in and observe that activity. We can eavesdrop on it with fMRI, histology, 1 and 2P. And we see areas like the hippocampus active during memory formation. So from that, we get this idea of correlation, that hippocampal activity may or may not correlate with memory expression. Then usually if we want to understand how something works, right, we go in and break it and see what kind of output is broken as well. So let's say we can find the brain cells that are involved in forming a memory. And if we can block the activity of those brain cells, and that seems to impair the expression of memory, then we get this concept of necessity, that some set of cells are necessary for the expression of memory. And then more recently, what we've been doing, or what the field really has also been doing, is going in and trying to mimic neural activity. Now, I use mimic loosely here because we can't exactly play Beethoven yet with the brain and basically recapitulate sequences and endogenous firing patterns with maybe some rare exceptions. But we can go in and at least hit chords on that piano and activate brain cells with tools like optogenetics. And we can ask, if we activate a set of cells that are active during memory formation, and if that seems to be enough to drive the expression of a memory, then we get this concept of sufficiency, that activating a set of cells is sufficient to drive a given behavioral output. So I'll focus mainly on this idea of activating brain cells and then try to make sense of what that means in terms of the subsequent outputs that actually emerge. So just to, I'm going to go over one of our first published study for 2 seconds, just because it really sets the basis of what my lab does, and especially with the data that I want to present today. But we started off with this hypothesis that cells in the hippocampus that are active in this case when a memory is being formed may or may not be sufficient to drive the expression of that memory. So we went on to iterate on an activity-dependent tagging strategy, as Yingqi mentioned. And in this case, we were using a CFOS-based system to drive the expression of channel rhodopsin. So the green cells here are dentate gyrus cells that were active during the formation of a fear memory. They express channel rhodopsin, and then we asked, does going into the brain and optically activating those cells drive the expression of that memory? In this case, our index of memory recall here would be freezing behavior. So this was the one mouse that worked on the big experimental day, but it was the one that worked the best, where the first half of this video, you can ignore the caption, but the first half of this video, this animal does not have a fear memory. So we're just gonna go in and shoot light into the brain to control for non-specific effects of light. So for instance, this animal is in a neutral environment right now. It has no reason to be afraid of it. You can see it foraging around, and we'll go in and just shoot light into the brain. And there's no obvious detectable change in behavior. So at least it was one of our first pass at controlling for the effects of light. Now this is the same animal. It does have a fear memory, and the dentate gyrus cells that process that fear memory express channel rhodopsin. And when we go in and optically activate these cells, the animals almost immediately show this stereotyped freezing posture, which at least convinced us that perhaps we were actually enabling the recall of that particular memory. And then from this work, we went on to show that we could activate cells that were active during a neutral memory and artificially associate it with aversive information to thereby create a false memory, quote unquote, and then also activate cells that were involved in forming a positive memory and drive things like preference and more active appetitive behaviors. So when I started the lab, one of the projects that we had in mind was instead of activating fear per se or something that's rewarding per se, what about something that might be, maybe it's a different dimension of modalities. So something like social behavior. So this was a project that started years ago now. And the idea here was when animals are interacting, is there any bits of information that are being transmitted in this case through those interactions? So we thought, like, what if the interactions were negative, for instance? Was there bits of that kind of negative information that was being transmitted as well? And this was a project led by a postdoc in the lab, Abby Finkelstein, as well as her remarkable team of researchers in the lab, who really set out to develop a different behavioral assay for measuring this, and then to go in and use these activity-dependent strategies to ask what happens when we go and start tinkering around with the hippocampus here. So the way this experiment goes, I'll walk through it because there's multiple steps in the behavior, but let's say we have a cage of five mice, right? So just a standard cage of mice and their conspecifics. We fear condition each of them individually, and then we take all of the animals and then we put them through extinction protocols so as to suppress that original fear memory. Then what we do is we have a cage of extinguished animals now after this protocol. So let's say we take one of the animals out of the cage, out of the extinguished cage. We take one of the animals out and we give it a reminder shock to give rise to the reemergence of that fear memory, to drive reinstatement in this case. So one animal is taken out of the cage, it's given a reminder shock, and then it's put back in the cage of extinguished animals. But when the reinstated animal, we'll call it the transmitter is always the animal that's in a high fear state. Now, we put it back in a modified cage where the transmitter here gets put behind a one-way mirror. So the transmitter has visual access to the extinguished conspecifics. We'll call those the receivers. So the transmitter is behind a one-way mirror. The transmitter can see and presumably smell also the receivers, but the receivers here can't see the transmitter. Presumably they can smell the transmitter and so on too. But the point is that the transmitter is behind a one-way mirror, has visual olfactory access to the receivers, but the receivers can't see the other way. So then the next day we test all of them for reinstatement and ask, did any of them actually display this kind of reemergence of fear based off of this one-way mirror test. So what we noticed was that in a subset of animals the following day, it's not exactly bimodal, it's maybe about a third of animals, did show in this case a kind of fear reinstatement. In other words, they showed heightened fear responses during the reinstatement test relative to the last extinction test. So something about placing the transmitter behind the one-way mirror was sufficient to drive reinstatement in a subset of the receiving animals in this case. And then one of the controls that we did here was that rather than having a one-way mirror, we just have it be an opaque, kind of like an opaque wall. So neither of the animals can see each other in this case. So when we put the transmitter behind an opaque wall, as opposed to the one-way mirror, then none of the animals show fear. They don't show reinstatement on the following day behaviorally. So the summary for this slide, because it's important for the subsequent slides, is that for some reason, putting the reinstated animal, the transmitter, back in the cage behind a one-way mirror elicits the reemergence of fear in a subset of the receiving animals during a reinstatement test. So this begs the question of why. So one of the things that we decided to do next was, let's say it's a similar behavioral protocol where the animals they undergo fear conditioning, they undergo extinction. One of the animals now actually gets the immediate shock just as before. But when the transmitter animal, the animal in the high fear state, is put back in the one-way mirror, test, the receivers here have that window open for tagging cells during this particular experience. So the receivers in this case have hippocampal cells that are active during this one-way mirror test, now express channel rhodopsin in the dentate gyrus. So the transmitter is put back behind the one-way mirror. The receiver animals now have cells that are active in the dentate gyrus tagged with channel rhodopsin. And then the next day we go and optically activate these cells to ask what happens. And then interestingly, when we put the animals back in this case, when we put the animals in a completely different environment, when we activate the cells in the receivers that were active during this one-way mirror interaction test, we see in this case this seesaw shape where optically activating those cells increases freezing, goes back to relatively low baseline during the off sessions and back on. So activating cells in the receivers that were active during this one-way mirror test is sufficient to elicit freezing responses when activated. And then as a quick control, what we did was, instead of having this reinstated animal placed back in the cage, we just put a naive animal back in the one-way mirror. So basically, it's still a mouse that's being put back behind the one-way mirror, but that animal did not receive the reminder shock. And then when we do that, we don't see this increase in freezing. So basically, this animal has to be reinstated or in a high fear state in order to elicit activity in the hippocampus that subsequently becomes sufficient to drive freezing responses. So one of the experiments that we thought of from here was maybe doing this, maybe the animals here, for instance, are recalling the original fear memory for some reason. So to at least begin to probe this, one of the things that we did was, imagine once again, we fear conditioned the animals, but during the fear conditioning session, the cells that are active during fear conditioning are tagged with GFP in this case. So we have the same snapshot of dentate cells that are active during fear conditioning. We do the same protocol of extinction, give one of the animals a reminder shock. We put the animal back in the one-way mirror. We test for reinstatement the following day. But during reinstatement, we sacrifice all of them an hour and a half later and stain for CFOS, because the prediction here is perhaps CFOS active during reinstatement in the reinstating animals, in this case, might overlap with the original set of tagged GFP cells. And then this is what it looks like in this case, where, as a reminder, green are cells active during fear conditioning, red are cells that are active during the reinstatement test in this case. And then interestingly, in the animals that reinstate, so the socially reinstated animals, they show a large increase in overlap in this case between CFOS, the cells active during this reinstatement test, and with GFP, the cells active during fear conditioning. Whereas the animals that don't reinstate, they don't show this kind of overlap. They don't show this kind of overlap with the originally tagged ensemble. So one of the working hypotheses here was that that these cells that are active during fear conditioning they come back online during this one-way mirror test. And we did a bunch of other experiments where during this one-way mirror test, for instance, if we chemogenetically inactivate the fear ensemble during the one-way mirror test, that actually completely blocks reinstatement in this case. So it seems like blocking the original fear ensemble during the one-way mirror test also blocks the reemergence of fear on the subsequent day. So we think that the fear memory is coming back online during this one-way mirror test. And in some animals, it's sufficient to actually be able to elicit freezing responses during reinstatement. Now, there's a lot of other data that unfortunately don't have enough time to show here, but the first prediction was that maybe it was something more odor-based or pheromone-based, and that there's kind of a distress kind of signal that's being sent. But we tried a lot of different ways of like swabbing the animals or basically getting the pheromones off of the reinstated animals, and that surprisingly wasn't sufficient to elicit reinstatement. If we introduced an aggressor, so maybe it was just some kind of like social stress, that didn't seem to elicit reinstatement. Also surprisingly, in this case, if we immobilized the receivers, if we immobilize them, so we apply a physical stress, that also didn't induce reinstatement. So it does seem to be specific to the reinstated animal that's placed back in the one-way mirror test, and then kind of consistent with the idea that maybe it's the original fear memory that's coming back. It's a very context-specific effect. So these animals don't generalize on subsequent days to new environments. They only reinstate to the original environment where fear conditioning actually happened. And then right now, one of the things we're trying to do is maybe there's kind of a literal distress call that's being sent from the transmitter, and maybe it's USV mediated. This number 4 here is a technical disaster, and we're trying to figure out what's the best way of actually doing this, but that's one of the predictions. And we had some preliminary data that in the receiving animals, if we blocked the medial geniculate nucleus, so we block some auditory cortex, that also seemed to block the effects, which is what led us down this rabbit hole of thinking that maybe it was USV mediated. So in the last couple of, okay, good, I do have a couple of minutes. In the last couple of minutes, I kind of want to wax poetic on, where are we in the field? And, have we discovered the engram? Like, the spoiler alert is almost certainly not. I think it's just a very snapshot of a subset of cells that seem to be sufficient to drive behavior, which is great because it keeps us all in a job. And then, where are we now, and where is the field going, and the way, and again, why do these experiments seem to work somehow? So we started in the hippocampus, and this figure is antiquated now because this is 5 years old. But these are all the different areas where sort of the observational and necessity and sufficiency experiments have been or were performed. And it begs the question: what happens if we activate a set of cells active during a learning experience in the amygdala or in the olfactory bulb or in the PFC? To anthropomorphize it, what's the animal thinking? Like, what's the mind of a mouse here when we activate dentate gyrus cells or amygdala cells? Is there different mnemonic content that's coming online that's contingent on the area that's actually perturbed? And I think that this is fodder for discussion later on, but I think we can design some clever experiments that tease out what kind of mnemonic information is coming online that might be different depending on the area that we're actually perturbing, for instance. So one of the questions that I struggle with again is that when we activate the hippocampus, this is activating thousands of cells at a very artificial frequency, so 20 hertz usually, for 15 millisecond pulse widths for 3 minutes. And I'm willing to bet anything that the dentate gyrus does not fire at 20 hertz with 15 millisecond widths for 3 minutes in order to elicit memory recall. So why would these experiments still work, even though, again, we're not playing Beethoven on the piano of the brain, we're kind of hitting it with like a hammer and hitting some chords. But it's amazing that maybe the brain's smarter than we think, and it can somehow take those chords and translate it into Beethoven. It can extract signal out of that, out of a lot of noise, and still extract a behaviorally meaningful signal. And I think that by analogy here, right, there's no drug that perfectly recapitulates the endogenous firing patterns throughout the brain, but sometimes they work, right? If you have 2 beers, the brain doesn't explode, right? Even though it doesn't naturally work on 2 beers, it translates that into a behaviorally meaningful output nonetheless. So maybe what we're doing with these kinds of tools is kind of a, I guess, 21st century drug work where we're still imposing a lot of noise into the brain, but there might be still some signal there that can turn into a behavioral output in general. And then the last part here, which is an analogy that I often use in talks, but one of the things I think of is, what if we take something way less complicated than the brain in this case? Like, we can take something like a car. So we can ask in this case 2 types of questions as a conceptual bifurcation here. One question is, how does a car work? Right? You go in, you turn the key, or you press the on button and the engine starts. That's just how it works. There's the other question of, how can a car work? And you can go in and put the screwdriver in and twist it around, or MacGyver it and connect the 2 red wires, or get it to run on canola oil or solar panels, even though it wasn't designed, quote unquote, to run on these artificial things, but they still jumpstart the engine somehow, right? Because there's something there that the car is still making sense out of. And I think that we can ask the same thing with memory, where there's one question of, how does memory work? These are the observational studies, the histology, the MRI, the 1 and 2 Ps, the parts where we can go in and just listen in on the brain as it's doing its thing when memories are being formed. That's just the code, right? That's the code that we're trying to make sense of. But there's the other subtly different question of, well, how can memory work? And maybe this is just getting memory to run on canola oil and solar panels, right? It's like we're asking how can memory work? And I think that any exogenous perturbation that we impose on the brain might be revealing how can it work when we're really pushing it to its limits artificially. And just because that may work and we can reveal maybe different ways that memories can be jump-started, it doesn't mean that how can memory work has to be superimposed, therefore, to how does memory work. Because in this case, the artificial methods that we use to manipulate memories may not actually be how memory works, but it may be a way that it can work nonetheless. So it's an interesting way, I think, of thinking of how any, again, exogenous perturbation, what it means to have one actually occur in the brain. So with that said, in the last minute, yeah, I'd like to, thank everyone for the attention. It's so like heartwarming to actually see everyone here and to use these 2 hours as a kind of scientific escapism from the world. And like on the lab website, for any trainee that's listening or postdoc that's listening, all of our, for instance, grants that are funded, unfunded, pending, actually got to update it because there's a lot of unfunded ones recently, but basically all of those are up there, as well as interview tips, faculty job talks, basically anything that helps champion open science, which I think is the way to go. And that way we all stand on each other's shoulders, right? And not each other's feet. So yeah, I'm really happy to take questions, and thank you everybody for your attention.
Thank you, Steve. That was terrific. So I'll start with the question, sort of riffing on the last 3 minutes. You're stimulating, or you're optogenetically activating the tagged cells in the dentate gyrus. And so we know that the dentate gyrus projects to CA3, which projects to CA1, which projects out to the rest of the brain, if you will, to elicit the behavior. What happens if you move the stimulation or the activation down that path? So for example, if you were to stimulate CA3 or CA1, do you get the same robust effects, or is the translation of into the symphony muddled?
Yeah, it's a great question. And I guess there's 2 quick answers where if we were to tag cells in the dentate, CA3, let's say the BLA and the accumbens simultaneously, when we activate the DG, we do see preferential reactivation of those originally tagged cells. So somehow the information is still making it downstream to the same set of tagged cells. But in terms of if we were to start stimulating now downstream, that unfortunately is a big like monkey wrench in this whole problem. Because one thing is, for instance, when we first started these experiments, we were initially aiming to try to activate fear through CA1, and we could never really get it to work. And then other groups had better success in CA1 by changing 2 things. The first one was by changing the frequency of stimulation, and CA1 seemed to like 4 hertz, and the BLA also, like, and PFC for that matter, that 4 hertz seemed to be this kind of magic number, whereas 20 hertz was what we used in the dentate. So for an area like CA1, it was frequency dependent, but then it got a little more complicated because other groups, for instance, did get CA1 to work at 20 hertz, which is what we used in DG, but they tagged way less cells, because with our system, we tag about, let's say, 20 to 30% of CA1 cells in general. And we don't, with 20 hertz, we don't get a freezing. But when other groups tag about 12 to 13% of CA1 cells and stimulate them at 20 hertz, that does work. So it seems like it's both frequency dependent and population size dependent. So maybe those 12% or 13% of cells that they tagged using different viruses are the most engrammy of cells, and for some reason they have access to these downstream to ultimately to lead to behavior. But and again, the same thing for areas like the BLA and accumbens and so on. Like population size, I think, really matters here, and sometimes less is more, as well as frequency, or titrating the frequencies also seems to matter. So I think both make our lives a lot harder, but I think that those are at least 2 boundaries that we usually consider when doing these simulations in other areas.
Yeah, it'd be fascinating to record in those downstream areas when you do the stimulation in the dentate. So Anna asks in the chat, have you tested whether olfactory cues are signaling fear reactivation? I think you answered that, and I'll just add to that. There's an interesting paper by Marta Moita, where social transmission of fear, where it's actually silence that's the salient cue.
Yeah.
So you might want to look into that.
Yeah, absolutely. And like we admittedly haven't systematically ruled out olfactory or anything odor or pheromone based, but at least with some of the initial experiments, it didn't seem to be enough to elicit this kind of reinstatement.
Okay, so Sander Van Bree in the question and answer asked, once we do clearly have an engram, is that the end of the memory storage problem? Could one not say that finding the engram is analogous to finding the brain area that causes behavior X without any sort of mechanistic explanation?
Yeah, I hope not. And I think that, it gets a lot harder because when the second after you form the first memory that you've ever formed, every subsequent memory enters a brain that has a history, and that modifies and resculpts the brain with every subsequent memory. So in this case, the snapshot of an engram that we might see, it's going to change both over time and as subsequent experiences are imposed on it too. So I think that it's almost like the engram is nowhere and everywhere at the same time, because that kind of dynamism I think is very real, especially when we start considering brains that have histories on the order of decades, for instance, like that. One of the examples being with like systems consolidation, where when it comes to tagging, usually we see cells in the hippocampus, even a month later, they can still kind of drive freezing, but not as much as on day one. But when you look at the overall levels of FOS in the hippocampus, they seem to remain steady at day 30, but it's not the original set of cells. So it's not that the hippocampus necessarily goes offline. It's just that there's a shift in which cells may be contributing to the recollection of something like an old memory. So I think that, that example again is one of those things that, why that's the case. I think that maybe there's a kind of transformation in the contents of the memory, or as they go from recent to remote and so on. And I think that is something that I hope is more tackleable these days than not. But I think that one of the things that I only briefly touched on is, that in order for that symphony to make sense, we need to not just have the conductor and the instruments, but like we got to hear it in real time. So being able to go and record in real time and actually listen to these on a large scale is just as much, like that physiological aspect of it is just as much as the engram as the molecular and cellular aspects of it as well. So it's almost like there's the structure of memory, which might be a set of tagged cells, and then the function, which is memories working in real time. And that's where, again, that kind of symphony emerges, I think.
Great, there's time for just a minute left, but here's a question from the chat. Is it known when mice are deprived from adrenaline, for example, that they still have a similar fear response? And so the main question is this: what is the difference with knocking out or inactivating the neurons responsible for the fear response? If you were to activate the neurons optogenetically, but block the neurons responsible for the expression of fear, what do you expect?
We only have one data point on this, and I think it's the hardest experiment I've ever had to do because it required 4 fibers in a brain and just like plugging 4 things in simultaneously. But we can block. So we activate the dentate. Actually, let me take a step back. Let's say we tag cells in the hippocampus and in the amygdala and in the accumbens that were active during fear conditioning. When we activate the dentate, if we simultaneously inactivate those tagged cells in the BLA or in the accumbens, that does seem to block the process. So the way I think of it is just kind of like a big domino set where in the DG, we kind of found this golden domino that we can knock over and the chain reaction of recollection happens. But when we remove a set of cells in like the BLA or the accumbens in real time, we take away that domino that would keep that chain reaction going and it stops there. So I think that there are ways of blocking that expression and really tracing like where this memory is going from the DG to behavior. And the way I think about this is that when we activate the dentate gyrus, somewhere between there and the spinal cord, there's some magic happening to actually be able to elicit a freezing response. And I think that that's a career's worth of work to try to trace out that kind of circuitry, but it's definitely a fun one.
Cool. Well, thank you, Steve. That was fascinating.
Thank you so much. I really appreciate it.
So it's time to move to our third speaker. This is a great pleasure for me to introduce, and since I organize a symposium, I can invite my friends. So my close friend and collaborator, Professor Todd Sackler, He's a distinguished professor in the departments of physiology and pharmacology, as well as neurology at SUNY Downstate Health Sciences University. And you can see the title of his talk. He's ready to go. Todd?
Okay, thank you. Andre, you can hear me?
We can hear you.
Okay, great. So as we've heard, whoop, oh dear, let's see. It's not advancing, sorry. Now it is, okay, thanks. So memories evolve over various phases. So there's a short-term memory that becomes consolidated into a long-term memory. And we know a lot about the molecular mechanisms of this consolidation processes. NMDA receptors are activated, multiple protein kinases are activated, following by gene expression. But the thing is that each of these has its own temporal window. And as we heard in the first talk, even gene expression, it lasts only for an hour or so. And yet memories last much longer. So the fundamental question in terms of memory storage.
Todd?
Yeah?
You don't have screen share enabled.
Sorry. Thank you. Let's see.
There it is.
There it is.
All right. Sorry, guys.
All good.
So have you heard any of this? You heard this about the key question being, what is the storage mechanism of memory? Andre?
Yeah. Yes.
Okay. Thanks. So one of the hypotheses is that receptors that activate kinases, that these protein kinases, which are able to enhance synaptic transmission, can become autonomously active. And CAMK2 is well known, but I'm going to focus on protein kinase C, for which there is a lot of evidence. And the mechanism for the formation of a constitutively active form of protein kinase C is through the formation of what's called a PKM form. So here's protein kinase C. It comes in many different isoforms, but they all have a similar structure with a catalytic domain in green, a regulatory domain in red, and the regulatory domain contains a pseudo substrate that inhibits the catalytic domain. Second messengers such as diacylglycerol can bind to the regulatory domain and cause a conformational change, opening up the enzyme, allowing a substrate access to the catalytic domain, and that can get phosphorylated. Each of these PKCs can also be cleaved, separating the regulatory domain with its pseudo substrate from the catalytic domain. And this free catalytic domain is called PKM, and it is an autonomously active kinase. So when I started, I asked the question, is there a PKM form in the brain? And it turns out there was only one isoform, the zeta isoform, one of the two atypical PKCs that has a substantial amount of a PKM form. So we called this PKM zeta. And remarkably, it doesn't have any PKC zeta. And that's because the mechanism for the formation of PKM zeta is not by that cleavage. It's by a unique mechanism in which the gene for zeta has an internal promoter, which makes a messenger RNA that contains only the catalytic domain. The regulatory domain, with its pseudo-substrate inhibition, is not expressed at all. So this internal promoter is active only in the nervous system. And as I said, it makes a messenger RNA, but the messenger RNA is translationally blocked under basal conditions. It doesn't make protein. Unless the NMDA receptor is activated, calcium comes in, multiple signaling molecules, all of them transient, but they're important for releasing this translational block and forming PKM zeta. So then the question was, what's the function of this PKM zeta? Is it necessary and sufficient for the maintenance of LTP, for example? So we recombinantly expressed and purified the PKM zeta and put it inside a whole cell recording pipette and then patched onto a CA1 pyramidal cell in a hippocampal slice. And as the PKM zeta floods into the neuron, there's potentiation. And so it's sufficient to cause potentiation even without strong stimulus like LTP. To examine necessity, what we did is we took the sequence of the regulatory domain, that's the pseudo substrate, myristylated it so that it's now a cell permeable form, found the dose that we can then bath apply this peptide that will suppress the potentiation of PKM zeta, and then applied it to the bath in LTP. And what we found is that that dose can reverse LTP maintenance even 5 hours after the tetanization that induces it. Totally unprecedented in the literature. It also had no effect on basal synaptic transmission recorded in the same slice. And then with Andre, we found the initial evidence that PKM zeta could maintain a long-term memory. We used active place avoidance, which Andre has invented. And basically, the mechanism is the apparatus works by putting a rat or a mouse into this rotating arena. And so the animal will get a shock in a stationary, non-rotating sector. So the animal has to learn to avoid getting this shock. So you put an animal in, it will wander around the apparatus. Then during the training, it learns to avoid. And then you could then take the animal out, put them back in, and even a day later, it will avoid, even if they're not receiving a shock here. So we trained an animal, waited 22 hours, injected this peptide called ZIP, and then tested its memory. And ZIP appeared to erase the memory that, that it had previously learned. Now, over the years, there's been evidence both for and against the hypothesis that PKM zeta is necessary and sufficient for long-term memory storage. It's not just ZIP. It's also another inhibitor of the catalytic domain of PKC called chelarethrine. There's viral expression of a dominant negative PKM zeta. There's PKM zeta shRNA virally expressed. They all disrupt well-established long-term memory. But on the other hand, ZIP and chelarethrine affect other PKC isoforms. And in addition, ZIP has non-specific effects. PKM zeta knockout mice compensate for the loss of PKM zeta in LTP and long-term memory by persistently activating another isoform, that other atypical that I mentioned, PKC iota lambda. But on the other hand, ZIP still works to disrupt the maintenance of LTP and memory in these knockout mice. ZIP affects not only PKM zeta, but also iota lambda. And there's so far no pharmacological evidence that PKM zeta maintains the LTP and long-term memory in wild-type mice. And then the third, overexpression of PKM zeta enhances new and established memories. But what mechanism is there for that? As I showed before, if we flood the neuron with PKM zeta, you'll enhance all the synapses. That should degrade memory, not enhance them. So what I'd like to show today is answers to all of these questions. And the clue to them came when we first started looking at where PKM zeta is increasing during LTP and long-term memory. Here's an example of 2-hour LTP and where the PKM zeta is using immunocytochemistry. It's mostly next to the stimulating electrode and in radiatum and cell body. We also looked at memory, again with the active place avoidance task, in which we trained a mouse, waited 1 day, sacrificed, and looked both with Western blots and in other mice with immunocytochemistry. And what we see is that there's an increase in PKM zeta in the hippocampus, primarily in CA1, less so in CA3 and dentate, but also in the overlying neocortex. There is a preponderance of the increase in PKM zeta in these active ensemble neurons that Yangtze and Steve have been talking about. And we don't have time to go over in detail, but in a mouse in which tamoxifen injections will activate the ability of ARC promoter to make a marker, in this case, EYFP, that's been fused to channel rhodopsin so that it's membrane bound. We see that there's an overlap of the increase of PKM zeta in these active ensembles. And this can be quantified even 1 month after the training. So if we do training and then inject the 4-hydroxytamoxifen, continue training, so it's well-established memory, and then look 30 days later, what we see is comparing the trained and untrained mice, if looking at CA1, there's increases as expected of the marker EYFP in the pyramidal cell layer relative to the untrained. There's increases in PKM zeta, and then there's an increase in the overlap. And that's true both in the cell bodies, it's true in the dendrites of radiatum, where there's increase in EYFP, PKM zeta, and in the overlap. But interestingly, not in laculosa moleculare. So this is suggesting that there is a compartmentalization of the PKM zeta within these ensembles, active ensemble neurons. In fact, if you look in more detail in the radiatum, we see the PKM zeta is in some spines, but not in many others. So then the question becomes, what could be sequestering? What could be compartmentalizing the PKM zeta? So we asked the question, maybe it's one of the known protein binding partners of PKM zeta called KIBRA. And KIBRA is particularly interesting for 2 reasons. One, it's a human memory associated protein. That is to say, alleles of KIBRA, for humans, people have either good or poor memory, normal people. And also, as I mentioned, KIBRA binds to and stabilizes PKM zeta. So we first asked the question, okay, do these 2 proteins actually interact closely? So to do this, we use what's called a bimolecular fluorescence complementation assay, in which KIBRA is now labeled with an fusion protein with half, in this case, the N-terminal half of a fluorescent protein called Venus. PKM zeta is labeled with the other half, the C-terminus of Venus. But each individually, these proteins do not fluoresce. If KIBRA and PKM zeta do touch within 10 nanometers, there will be fluorescence, seen here as puncta. So here are some controls. So if you take the KIBRA labeled with the N-terminal half and overexpress it together with just the other half of Venus without PKM zeta, we can see that the KIBRA is in these punctate regions, and these are in kidney cells for ease of transfection. If we put in PKM zeta, again with only the Venus not attached to KIBRA, we see the PKM zeta is more diffuse in the cell, not so punctate. But again, no bimolecular fluorescence. They're not touching each other closely. But if you overexpress both of them, then there's this bimolecular fluorescence very clearly. What about in neurons? If you take these same two constructs and transfect them into hippocampal neurons in culture, what you see is the PKM zeta is extensively in the dendrites of neurons. But the bimolecular fluorescence, the interaction between KIBRA and PKM zeta, is very discrete in these puncta, typically in these spine-like processes, but not all of them. There are other spine-like processes that, in Philopodia, that do not have the interaction between PKM zeta and KIBRA. So now the fundamental question is this the molecular engram? Is this what is crucial for the action of PKM zeta? It has to be bound to KIBRA. So we tested this hypothesis using a new inhibitor of PKM zeta called zetastat, that instead of like ZIP, which affects the catalytic site, affects the binding of KIBRA to its putative site here, that's on the outside of the of PKM zeta. So we tested that and found that inhibiting the PKM zeta KIBRA interactions with the zetastat blocks the formation of these fluorescent complementations, so they're no longer acting. We also tested the other atypical PKC called iota lambda, which interacts much less with KIBRA than PKM zeta, but the drug has no effect. So then the question is, if we can block the interaction between KIBRA and PKM zeta, what does it do to LTP? So we tetanized in hippocampal slice, waited 3 hours to get into the late phase of LTP, and then added zetastat. And zetastat did what ZIP did, but what nothing else could do, which is to reverse the maintenance of LTP while not affecting baseline synaptic transmission within the same slice. But unlike ZIP, which also affects the compensatory kinase iota lambda, zetastat had no effect on the PKM zeta knockout. Now, if something is really involved in maintenance, you should be able to wash it out and yet not have the potentiation come back. If the potentiation comes back, then it's more involved in expression of LTP, not in the maintenance. So we waited 3 hours after a tetanus, added the zetastat, reversed, and then washed out. And the potentiation did not come back. How do we really know that we've washed it out? Maybe it's just sticking around in the slice. Well, we added the zetastat and then tetanized one pathway, saw that it repressed the late phase of LTP, washed out for 1 hour, and then was able to show that the LTP in the second pathway was persistent. How about the memory? We trained mice, waited 1 day, injected zetastat or this vehicle, waited 2 days for washout, and tested retention. And just like ZIP, zetastat could disrupt the established memory. But unlike ZIP, its effect was specific to mice that had the target for the drug, PKM zeta. What about the washout? Well, we repeated the experiment, trained the animal, and then immediately retrained on active place avoidance, but within a different context. And again, the memory that had already been established in the hippocampus was erased. And yet once the zetastat was washed out, new memory for a different context was indistinguishable from an animal whose memory had never been erased. And so in conclusion, Is there pharmacogenetic evidence directly demonstrating PKM-zeta maintains LTP and long-term memory in wild-type mouse? Yes, an allosteric PKM-zeta inhibitor, Zetastat, reverses the maintenance of late LTP and long-term memory in wild-type mice, not in compensated knockout mice that lack the drug target. What is the mechanism by which PKM-zeta could enhance new and established long-term memories? Well, at least now we have a hypothesis. The hypothesis is that it's really the persistent synaptic tagging by Kebra of a diffusible PKM-zeta that is the molecular engram of long-term memory storage. And it explains how memory enhancement by PKM-zeta overexpression might work, because as a memory degrades, we do know that the levels of PKM-zeta drop in parallel. But if the Kebra PKM-zeta interaction is still there, then you could overexpress PKM-zeta and it will be targeted specifically by persistent synaptic tagging to the synapses that have Kebra. So our overall conclusion is it's the PKM-zeta Kebra interactions that are maintaining late LTP and long-term memory. And that's it. Silence.
My computer's reluctant to unmute me. That was wonderful, Todd, but I'm biased. If there are any questions.
No, you're not.
Marco asks, do you think LTP is necessary and sufficient to explain long-term memory?
Well, Yingxi, again, am I pronouncing your name right? Close enough? Okay.
Yeah.
So she just showed in her first talk how the network is molded by both changes in excitatory and in inhibitory transmission. So LTP is a persistence enhancement of excitatory transmission. And that's what PKM-zeta is maintaining. Whether it has a role in the persistent changes of inhibition, we don't know. We do know that PKM-zeta is present and expressed in GABAergic interneurons, or at least some of them, as well as excitatory principal cells.
Thank you, Todd. Santiago Oye, I hope I pronounced your name correctly, asks, did you look at transcriptional modulation of PKM-zeta? I'm thinking what happens after protein turnover? Memories last way longer than proteins do. So how can PKM-zeta sustain memory over the protein turnover?
Okay, well, there's two possibilities. One is that, so let me just first say that for the initial formation of PKM-zeta in both LTP and in memory, it's a translational upregulation of protein synthesis from the dendritic messenger RNA that's already there. So it actually isn't a transcriptional change initially. So there's two possibilities. As I showed, increases in PKM-zeta can last a month. So that could be either due to a persistent increase in synthesis or a persistent decrease in its degradation. That is to say, an increased stability. A year ago, I had put my money on an increased synthesis. But times have changed. And so now that we know one of the functions of Kebra is clearly to make PKM-zeta last longer. It extends its half-life. So it could easily be that the level of PKM-zeta synthesis returns back to its baseline level, but the reason we see a steady state increase that can last a month is because of the decreased degradation due to the binding of PKM-zeta to Kebra at particular sites.
Okay, so Todd, I'll ask one other question. So it looked from some of the images that you showed, and maybe you can comment on this, where is PKM zeta expressed? In the detailed experiment that you showed, it was in stratum radiatum, but not in lacunosum moleculare. How about in CA3 and then in the dentate gyrus? Is there compartment-specific expression there in response to learning?
Well, in active place avoidance, CA3 is very silent. We don't see the increase in the ARC in the ARC promoter driven marker in CA3. In dentate, we see it somewhat more in the upper blade versus the lower blade, but it's really very small amount for this spatial memory relative to the changes we see in CA1. So we focused our attention on CA1.
Thank you. One last question. This is from Jantina Brook. As the change from LTP to long-term memory also involves synaptic homeostasis, do you see differences in PKM zeta that correlate with synaptic homeostasis?
I think it's hard to measure that. I would say no. What we do see, and I'm repeating myself, is if the memory lasts from a day to a month, the overall steady state level of PKM zeta lasts from a day to a month. If the memory training is not as strong and the memory lasts a day but then fades by a month, the PKM zeta is increased at 1 day and then fades by 1 month. Doesn't quite address the issue that you have mentioned.
Okay, wonderful. Well, thank you, Todd. That was a fantastic presentation. We'll move to the final speaker.
Okay.
I'm definitely biased. But setting us straight will be our final speaker, Professor David Glantzman. He's a professor of integrative biology and physiology and also neurobiology at UCLA. And he'll tell us about towards a revival of the molecular code hypothesis of memory. David, Zoom room is yours.
Okay, thank you very much, Andre. And I want to thank you for inviting me to this symposium. And I have to say it's a real pleasure to be here listening to all this exciting data. So I'm going to, this talk, I should say, is going to be part data, part intellectual archaeology, and a fair amount of speculation. So just to give everybody a heads up. So as everyone here and most of the participants know, one of the major hypotheses in neuroscience today is that learning memory are mediated by changes in the strength of synaptic connection. Now, this idea was one of the earliest proponents of this idea, and one of its stronger champions is shown here, the Spanish neuroanatomist Ramon y Cajal. And the synaptic hypothesis of memory has recently received the imprimatur of the Brain Prize in 2016. And the winners of this prize from right to left are Timothy Bliss, who's the co-discoverer of LTP, Richard Morris of Morris Watermaze fame, who pioneered investigations into the role of LTP in learning and memory, and Graham Collingridge, who identified the molecular mechanism of LTP, the NMDA receptor. And I've been in the field of learning memory for 40 years now, and I'd say for about 36 of those years, I implicitly accepted the synaptic plasticity hypothesis of memory. I began to go astray when I first heard about these results by the gentleman shown here, Kareem Nader, and his colleagues. And they showed something, or really they re-showed something quite remarkable, which is that under certain circumstances, a memory could appear to be erased. And the experiments that they did were they gave fear conditioning to rats, and 24 hours later, they injected a protein synthesis into the rat's amygdala. Of course, that's the site of one of the critical sites of fear conditioning. And then when they tested the rat the next day by giving the tone CS, the rats didn't freeze. And that made perfect sense, because the idea is that after 24 hours, the memory's been consolidated, all the protein synthesis that's necessary for consolidating the memories is over. And so if you temporarily inhibit protein synthesis, you're not going to have any effect on the long-term memory. However, when Nader and his colleagues tweaked this experiment just a little bit, they got a very different result. It's the same experiment. They fear conditioned the rat on day 1. On day 2, they injected a protein synthesis inhibitor into the rat. And on day 3, they retested the rat. But the difference was, before they injected the protein synthesis, they gave the rat the tone CS, so a reminder of its training experience. Now, when they tested the rat 24 hours later, the rat did not freeze. They acted like they had never been fear conditioning. And I found this result truly remarkable. In fact, I didn't even believe it when I first heard about it, but my friends convinced me that it was right. And so I started thinking, well, what's the implication of this for the synaptic hypothesis of memory? When the fear memory appears to be erased, do the synapses that are formed during the learning experience, do those get erased as well? And this question, of course, is very difficult to address in the mammalian central nervous system, but I thought I could address it in the system that I work on, which is learning and memory in the marine snail, Plesia. So this animal, which is shown from the top down here, has a defensive withdrawal reflex, and that's illustrated in part A of this slide. If you give this organ called the siphon a weak tactile stimulus, you produce a contraction of the gill with which the animal breathes. It's a marine organism, and the siphon. So this is a defensive withdrawal, and it's designed, of course, to protect these critical organs. The siphon is used during respiration to flush seawater out of this mantle cavity here. So this is a defensive withdrawal reflex. Now, if you give the animal a painful or noxious stimulus, and in these experiments, the animal was given electrical shocks to its tail, then when you test the defensive withdrawal reflex, you find that it lasts a lot longer. It becomes sensitized. Now, depending on how many shocks you give the animal, you can get sensitization that lasts for a very long time. So if you give it a single shock, you get sensitization that lasts for maybe 30 minutes. If you give it 4 shocks, you can, as shown in the graph in B, you can get shocks that last for a day. And as shown here, if you give 4 days of tail shock training or 4 trains of shocks, you can get sensitization lasts for a week. So I should say these are data from Bill Frost's PhD thesis done in Eric Kendall's laboratory. Okay, so you can also demonstrate a synaptic homologue of long-term sensitization. And this was first shown by P.G. Monterola, Sam Shacker, and Eric Kendall. And we've reproduced this phenomenon in our laboratory. And by we, I mean my research associate, Tian Kai Kai. And the way this is done is you dissociate sensory and motor neurons that mediate that defensive withdrawal reflex. You put them into cell culture, and after 3 or 4 days, they will have formed robust synaptic connections so that if you fire an action potential in the sensory neuron, you get a very lovely EPSP in the motor neuron. Now, this synapse can be trained, and the way one trains it is one gives the culture 5 pulses of serotonin spaced about 15 minutes apart. When you do that, you will get, as shown in B3, you'll get long-term facilitation of the synapse. And I should say, the reason that serotonin is used for this is it's known through the research of Eric Kandel and Jack Byrne and a host of others, that it's serotonin that mediates sensitization of the withdrawal reflex. So repeated stimulation with serotonin produces long-term synaptic plasticity. And Dee and Kai also showed that this long-term synaptic memory can undergo reconsolidation. So as shown in the experiment here, he gave synapses in culture 2 rounds of this serotonin training. And 48 hours later, some of the cultures got a reminder stimulus, and the reminder stimulus was a single 5-minute pulse of serotonin, which produces short-term facilitation, but not long-term facilitation in naive cultures. And then some of the synapses were treated with anisomycin, which is a protein synthesis inhibitor. And the results are shown here. Control synapses are synapses that had no stimulation. The reminder group are synapses that only got this 5-minute reminder pulse at 48 hours. And the yellow group is, the yellow bar represents synapses that received the serotonin training plus the anisomycin, but not the reminder stimulus, and they showed long-term facilitation. And the synapses that got at 48 hours after the serotonin training got the reminder plus the anisomycin, they showed this blockade of long-term memory. So having demonstrated the reconsolidation phenomenon in cell culture, we were prepared finally to do the experiment that I had been curious about, and that is, do the physical synapses that mediate long-term memory get erased with reconsolidation blockade? So we carried out morphological experiments on sensory motor synapses in culture. These were performed in my laboratory by Shunping Chen. And Shunping filled sensory and motor neurons with fluorescent dye. And so the motor neurons were filled with rhodamine, the sensory neurons were filled with fluorescein. And you can see these puncta that are associated, these green puncta that are associated with this red neuron. The red neuron's a motor neuron filled with rhodamine, and these green puncta represent presynaptic boutons, or we call them presynaptic varicosities. And those are sites of synaptic contact between the sensory neuron and the motor neuron. Now, what Shunping did in her experiment was fill the cells with dye and then image them using a confocal microscope, and at 3 times. First, before serotonin training, and then 24 hours later. And then some of the cell cultures got the reconsolidation blockade treatment, single reminder pulse of serotonin followed by anisomycin. And then all of the synapses were re-imaged at 48 hours. Now, in part B, you can see a synapse in culture, and the blue arrowheads represent synapses that were present at the first imaging session at time 0. And then you can see a micrograph at 24 hours, and now you can see those same blue varicosities, but you can also see varicosities that are labeled red. These appear in between the first and second imaging session. And finally, if you look at the 48-hour imaging session, you can see some new varicosities that are indicated by yellows. Now, I should point out that this is in a control synapse that did not receive serotonin. So you can see that there's synaptic, at the synaptic level, there's stability, but also a certain amount of dynamism, even if you don't do anything to the cultures. Okay, so what happens if you give the synapses this reconsolidation blockade protocol? So here are the results. So the synapses that received the serotonin treatment by themselves, the light blue, they show an approximately doubling of the number of varicosities, and that doubling persists over the next, persists for 48 hours, okay, between the second and third imaging session. Synapses that got the serotonin stimulation and anisomycin at 24 hours, they also show a persistence of this increase in the number of varicosities. The group that received the reconsolidation blockade phenomenon, they show the doubling at 24 hours, but at 48 hours, the number of varicosities reverses. And what I should point out is that the number doesn't revert to zero. It reverts to the original value, approximately the original value. These are normalized. The number has been normalized to the number at 0 hours. So this supports the idea that reconsolidation blockade involves synaptic erasure as had been originally proposed. However, we then decided to ask the question, okay, do the synapses that get erased, are they the same ones that grew, that appeared in response to the serotonin training? And here we got a surprise. So these are the results. So first of all, if you look at A, this is the varicosity number at 48 hours normalized to the number at 24 hours. The blue bars represent the groups that got the serotonin stimulation, but no reconsolidation blockade. And the red bars represent the group that from the previous slide that got the reconsolidation blockade protocol. And you can see that there is significantly more retraction of the 5-HT-induced varicosities, the varicosities that appear at 24 hours after the serotonin treatment. There's greater retraction in the reconsolidation blockade group than in the control group, which is exactly what you'd expect. However, there's also greater retraction of the original varicosities, that is the varicosities that were present before the serotonin treatment in this reconsolidation blockade. And finally, if you look at the varicosities that appear between 24 and 48 hours when there's no serotonin stimulation, there is a greater growth in the control group than in the reconsolidation blockade. So what you see is a sculpting of the varicosities. It's a you get a combination of retraction and growth, but this is designed to correlate with the experience of the synapse. And the synapses do not appear to be identified with respect to memory. There doesn't seem to be any memory tagging in the synapses. So we were kind of dismayed by this result, and it took us a while to accept it. And then we started thinking, well, if the synapses, the memory's not at the synapses, where could it be? And many years ago, a molecular biologist by the name of Robin Holliday proposed that epigenetic changes, particularly DNA methylation, might mediate long-term memory. And so, as you know, chromosomes are made out of chromatin, which consists of DNA plus these proteins called histones. And the three-dimensional structure of chromatin can be altered by enzymatic reactions, making the genes easier or harder to be transcribed. And DNA methylation is one of these so-called epigenetic changes, and it causes the methylation of the DNA for a gene makes it harder for the DNA to be transcribed. It silences genes, in other words. So Casey Pierce in my laboratory looked to see if the maintenance of long-term memory required ongoing DNA methylation. So here are her experiments. These are intact animals now, so she gave the animals 5 bouts of tail shock, and the details about the tail shock are shown in the bottom left of the slide. Then at 24 hours, the synapses were tested, after which they were given this drug, RG1OH, which is an inhibitor of DNA methyltransferase. They were retested at 48 hours. Then some of the animals got this partial training with 3 rounds of tail shocks. And we did that because we showed that following the apparent erasure of long-term memory by reconsolidation blockade, we could bring the memory back by this partial training. We wanted to see if inhibition of the memory would come back following inhibition of DNA methylation. And what we found was that the DNA methylation inhibition blocked the memory, and it wasn't reinstated. It didn't show spontaneous recovery, and we couldn't reinstate it with the partial training. Now, we thought, well, maybe the drug's just doing something to mess up the animals. So we also carried out a new experiment where after the RG108, this DNMT inhibitor, was applied, we gave the animals the original training once more. And now you can see that we were able to restore the memory. So the inhibiting DNA methylation looks like it really alters the stored memory. And we also showed that inhibiting DNMT would block the consolidation of long-term memory. So we thought, well, what is it that's responsible for the DNA methylation? And it's known that non-coding RNAs play a key role in epigenetic changes. So then we reasoned, well, look, maybe what we could do is train animals, extract the RNA from their nervous systems, inject it into naive animals, and we get the long-term memory. So that's what we did. So we took aplesia, we gave them sensitization training, and these were donor aplesia. We then took out their nervous systems, extracted the RNA, purified it, and then injected the RNA into naive animals that had not been trained. And we also took RNA from untrained donors, injected that into RNA. And we did some experiments on neurons and culture, and I'm not going to have time to talk about those results. What we got is shown here. So these are intact animals. We gave them 2 rounds of tail shock training. Then we tested them for sensitization. Afterwards, the RNA was extracted, and it was then injected into naive animals, and they were in turn tested 24 hours later. So these are the 2 groups that we took RNA from, the sensitization trained group and the control group. And these are the results for the 2 groups of recipient animals. And this is the group that got the RNA injection from the trained animals, showed enhanced siphon withdrawal reflex and indicative sensitization. And the group that got the control RNA did not. Now, we also carried out an experiment to see whether or not this apparent transfer of memory required DNA methylation. And if we repeated the experiment with using only the RNA from a trained group, injected that into another group of naive animals, we got the sensitization. But in a in a companion group, we gave the trained RNA followed by an injection of RG108, and the memory did not appear. All right, now here's where the intellectual archaeology comes in. These experiments are reminiscent of experiments that were carried out by James McConnell and his colleagues in the 1960s. And he worked on this animal here, which is a flatworm called a planaria. And what McConnell showed is that you could train planaria on a classical conditioning task. And these are aquatic flatworms. And he had them go down a single arm maze in an aquarium. And at a certain point, he would give them a light and then pair that with an electric shock. So it was a classical conditioning task. Then after he trained the animals, he cut them into pieces and fed them to naive planaria. So planaria eat carrion, and they're perfectly willing to eat cut up compatriots. And what he found was, then he tested the cannibal planaria, and he had two groups, the cannibals that ate the trained planaria and the cannibals that ate the untrained planaria. And he tested them for, he trained them again on this maze, and he looked at the number of contractions that they showed in response to the light. That's the conditioned response in the experimentals and controls. And he reported that the experimentals showed greater number of contractions. So another student of his, Jacobson, repeated McConnell's experiment, but instead of using cannibalism, he used injection of RNA, the same thing that we had done, similar to our results. And he reported that you could transfer the conditioned response using RNA. Now, this result stimulated a host of other experiments in which people tried to get learning transferred by RNA or cannibalism. And some people reported success, but there were reports of failures as well. So here, this group failed to get the transfer in planaria through cannibalism. This group and these other groups failed to get transfer of memory in the mammals that they were working on for a variety of different learning paradigms. So eventually this idea basically died out. And there are interesting reasons for this, and I'll just go over them quickly because I see I'm running over my time. But one of the reasons why this idea died out is that, as I said, people couldn't get the classical conditioning in planarian, and they failed to replicate the cannibal effect. And so, but it's now known that invertebrates can be classically conditioned, including planarians. And this memory transfer was not isolated to worms. People reported it also in fish and rats. Another major problem is that people were never able to identify a molecule that mediated memory transfer. Was the molecule RNA? Was it a protein? They didn't know. And even if the phenomenon of memory transfer proved to be replicable, because molecular biology was in its infancy, it would have been impossible for proponents of the molecular code hypothesis of memory to hit on a plausible mechanism. Back in those days, there was no knowledge of non-coding RNA or epigenetic changes. So the molecular biologist Günther Stent has talked about scientific prematurity. And I think the memory transfer is a perfect example of an idea that was scientifically premature. And the other argument, of course, is that the competing hypothesis, the synaptic plasticity hypothesis, was supported by a lot of impressive science. Kandel's work on learning memory and amnesia, and Bliss and Lermo's discovery of LTP in the hippocampus. So I think I just want to summarize by saying that I think it may be time to re-examine this molecular code hypothesis using modern technology and also the knowledge that we have about epigenetic change, and even possibly genetic changes. So with that, I'll end. I want to thank the people who did the work. These are students who have done other work in my lab, and I should say this reads like a nightmare of Donald Trump's worst nightmare. And finally, I'd like to thank colleagues who have done the work, and I apologize for going over, and I'll take questions now. Thank you very much.
Thank you, David. So I have a question from Kara Weissman. We only have 4 minutes, so thanks for the talk. How specific is the RNA-mediated sensitization? Does it extend to other conditioned, unconditioned stimuli, to other body parts, etc.?
So we use the RNA from the whole nervous system, and we don't know how specific the RNA is. We don't get habituation, for example, out of that RNA. And when we apply the RNA to sensory neurons in culture, they become hyperexcitable, which is a hallmark of sensitization. I didn't have time to show this data.
Another question, this from Sander Van Bree. Are you familiar with Randy Gallistill's arguments that the synaptic framework conceptually fails to explain how memory is stored and that a molecular story is needed?
Yes,
I'm
very familiar with Randy's ideas. He was a colleague of mine at UCLA for about 15 years. So I know his work. I have a lot of respect for it. And I actually accept it. But I think one respect in which Randy and I disagree is that I don't think he's come up with a function for synaptic change. So I believe that synaptic change plays a significant role in learning memory, but I believe it's there to express the memory. So I think you can have the persistence of a memory that's silent. It's locked into the nucleus of neurons, and it needs the synapses. The synaptic change basically reflects the stored nuclear memory. So I think that's one possible area of one difference between Randy's and my ideas. But I certainly know his work, and I have a lot of respect for it.
So I'll take the last question then. So it sounds to me that your ideas become really difficult to reconcile with notions that specific synapses need to be potentiated and or weakened so that the information can be expressed through a network. However, at the same time, you can also imagine that you don't need to have specific synapses if you want a particular pattern to go through a filter. People who deal with nonlinear dynamics know this very well, and in machine learning. Do you have any comments on how to reconcile those two views?
Yeah, so I think the synaptic specificity phenomenon is the hardest to reconcile with this idea. But it may be that neurons communicate with each other. Neurons that are part of a memory network are in constant communication with molecular communication, non-synaptic communication via exchange of RNA. So Jason Shepard has these very fascinating experiments in which he showed that neurons will exchange ARC, the RNA for ARC, in vesicles. And neurons may be able to recognize other neurons that are part of a memory circuit because they're exchanging RNA. And the RNA, that exchanged RNA, may be sufficient to alter, make nuclear changes, and the nuclear changes, that interchange may allow for synaptic specificity.
Well, thank you very much. We managed to do this without being cut off by the Zoom time limit, so we have seconds left. Thank you all for attending.
Thank you very much. Thanks, Andre.
Great talk.
Andre.
Who'll be giving talks on advances in different types of neural imaging methods, specifically a lot of applications towards freely moving in mobile settings. As a reminder, this is a traditional talk, so there'll be 12 minutes of presentation, for each speaker, followed by 3 minutes of questions. And if you want to ask a question, you can use the Q&A panel, and just make sure that when you, if you see a question that you're interested in, make sure to upvote it, because we'll answer questions, or we'll ask them in the order that they have the most upvotes. You can also use the chat to share thoughts and information related to that talk. So we'll get to our first speaker then. So our first speaker is Janaka, and he is a neuroscience fellow, postdoctoral fellow at Johns Hopkins University in the lab of Arvind Pathak. And his talk is on a miniature microscope for neuro and vascular imaging in freely moving animals. So whenever you're ready.
Okay, you guys can see my screen, right? Awesome, thanks, Stephen. So as Stephen introduced, I work both with the Kavli Neuroscience Discovery Institute and the Division of Cancer Imaging at the Department of Radiology in Johns Hopkins School of Medicine. And my talk today is going to be focused on our work we've been doing the last couple of years on building miniature microscopes that can image brain activity in freely behaving animals by using many different image contrast mechanisms. So to set the stage, I'll first talk about, in general, functional imaging in awake animals. And as most of you know, we traditionally use stationary benchtop-based large optical imaging systems. While there have been many really great discoveries with these imaging systems, they do have several significant drawbacks, and I've listed some of them here. One of the major drawbacks is that they require the animal to be anesthetized, or in other cases, the animal to be head fixed, which prevents you from correlating behavior with the brain activity you're imaging. And in instances where the animals are anesthetized, of course, the effect of anesthetics can easily alter the brain activity that you're imaging. Most of these brain imaging systems are custom built. And as I mentioned before, they restrict experimentation, and they do tend to cost a lot of money. So one of the solutions to all these many problems is to try and build a very small miniaturized imaging system. Now, to give you a sense of the miniaturization I'm talking about, shown here on the left is a typical benchtop imaging system from our lab. And here's one of our miniature microscopes drawn to scale, pointed to by this arrow. So it should become evident by now that with this level of miniaturization, obviously there is very limited space to put in the many different optical components that one would need to image in really comprehensive ways. So because of this, most of the miniaturized imaging systems, microscopes that are available today, and here's a list of them. We've got Kunal Ghosh's 1211 Nature Methods paper and several open source miniature microscopes, including the UCLA Miniscope, as well as several others listed here. They can only perform a single imaging modality. For example, the first row can only perform green fluorescence imaging in general, and therefore they are used for imaging neuronal activity. And that's why they're shown with an N here. And the latter two microscopes are mainly hemodynamic imaging microscopes that use techniques such as intrinsic optical signal imaging or laser speckle contrast imaging. But these bottom two microscopes cannot perform fluorescence imaging. So there's a big divide between miniature microscopes that can perform neuronal imaging and those that can perform vascular imaging, but there aren't any microscopes that can do both simultaneously. So that's where we came in, and our microscope, for example, can image with 3 different image contrast mechanisms, as shown here: fluorescence, where you can image neuronal activity via something like GCAM, or even track cells that are fluorescently labeled. We can perform what's called intrinsic optical signal imaging, which essentially looks at the light absorption by hemoglobin. And by doing so, we can image fine microvascular structures as well as dilations and contractions in vessels. And finally, with a technique called laser spectral contrast imaging, we can assess the blood flow. Now, here is a schematic of our patented first generation microscope architecture, showing the bottom view with all the illumination sources, the blue, the green LEDs, and a red laser diode for each of the imaging schemes, as well as a side view showing the additional lens, filters, image sensor, and the focusing assembly. Now, this microscope fits onto a custom 3D printed head mount that is surgically implanted on an animal head as shown in this figure. And we can control everything via a little graphical user interface that we've developed. And here's a picture of the microscope next to a US quarter coin. We can image with the resolution of 5 micrometers over a 3 by 3 millimeter field of view, and in mice this extent of field of view is sufficient to capture an entire functional cortical region. We could go up to about 10 to 15 frames per second, and with a bit of strain relief, our weight is roughly at 3 grams, which is conveniently bearable by a mouse. So the first thing we did was we established the fact that this is as good as a benchtop imaging system. So to do this, we imaged with a microscope and with a benchtop imaging system the same field of view using each of our 3 modalities. So shown here on the left is our images with our microscope. The first one shows a fluorescent a dye injected via a tail vein of a mouse. So it shows the vessel architecture with the fluorescence channel. And the second one shows the vessel architecture with our intrinsic optical signal channel. It's an indication of blood volume in this case. And the third one shows the blood flow in the same vessel architecture. And shown here on the right are the corresponding images from a benchtop imaging system of the same field of view. And we quantified the correlation between the images you get with our microscope and with the benchtop, and we were able to observe a reasonably high degree of correlation for the 2 systems. So with that, we were able to validate the performance of our miniature microscope as being reasonably high quality. And one of the first experiments we did was we looked at the auditory cortex of a mouse that expressed a neuronal GCAMP 6S. And we gave the mouse a auditory stimulus and looked at the neuronal and vascular responses to the auditory stimulus. So each of the epochs were 10 seconds in duration, and the auditory stimulus was presented at the 3rd second for a brief 300 milliseconds. And we conducted 30 trials per channel per stimulus type. And here are videos showing the responses for a 4 kilohertz sound tone. First, let me show the calcium response. And we can see a very focal dual spot calcium response showing focused neuronal activity. And the corresponding vasodilatory footprint, if you will, is completely different from the neuronal activity. And finally, the blood flow response is even more different than the vasodilator response. So we found that this mismatch between the neuronal and hemodynamic responses in the spatial domain to be really intriguing. And what we did was we then compared the response to a 4 kilohertz with a response to a 24 kilohertz sound stimuli. So here you can see the calcium response to the 4 kilohertz and the calcium response to the 24 kilohertz, which shows clear differences. And what's more interesting is if you look at the hemodynamic responses, each column shows a different modality of summarization. First column is the average of vasodilations. Next one is the maximum intensity projections. And the third one is after fitting a general linear model and regressing out global trends, we were able to have more specific differences for the two. And you can see how distinct the 24 kilohertz and 4 kilohertz vasodilator responses are as well. And finally, the same applied to the CBF response. So in summary, with this sort of neurovascular imaging, which is now uniquely possible because of our microscope in awake and then freely behaving animals, we are in a very good position to broadly evaluate the multiple aspects of brain function when it comes to stimulations. And we then applied this to study a disease model. So here we use the fluorescence channel instead of looking at neuronal activity to look at a fluorescently tagged tumor cell line, a 9L-GFP cell line. And as shown here, we imaged throughout the tumor's life cycle, roughly 2 weeks, and you could see the tumor slowly coming into the field of view and growing to cover the entire field of view. And in our intrinsic optical signal channel, at the same time, we were able to observe how new blood vessels grew to support this tumor growth. We call this angiogenesis, and the red arrows point to the formation of new angiogenic blood vessels that crawl towards the tumor inoculation site. And then on top of that, we used our blood flow imaging channel to look at when robust blood flow got established in these newly formed blood vessels. And what you can see is that you can observe blood vessels forming around day 6 and 7, but to get robust flow establishment, you see a couple more days delay. So this was a very insightful information that we could get only because we had an independent blood vessel channel and a separate blood flow channel. And finally, we did something that's called the microvascular connectivity. So for those of you who are familiar with fMRI literature, this is something equivalent to the bold fMRI resting state connectivity maps. So we let the mouse run around for 5 minutes while capturing 5 minutes worth of vasodilation and vasoconstriction data. And then what we've done here is we put a seed pixel on top of the tumor area, and we observe what's the area that's correlated with the time trace of vasodilations of the tumor. And what we found out was that as the tumor grew, the entire tumor area showed synchronous vasodilations and vasoconstrictions that were independent from the background area. And as the tumor grew, it sort of stabilized and then eventually decayed.
Sorry to interrupt you a little bit. We're a little bit past 12 minutes.
Okay, this is my final slide.
Oh, perfect.
So, okay. Moving forward, we can quantify these so that we can arrive at summarized stats. And in summary, this was our first prototype version, and we are now building a second prototype version that is much lighter but has the same capacity. And we are looking for collaborators to fund and test our prototypes. And with that, I'd like to thank my collaborators and funding sources and open the floor for brief questions. Thank you.
Great. Yeah, thank you. That was really, really interesting. Really nice to see nice potential solution for the future. So yeah, if you have any questions, please ask them in the Q&A. I have a quick question. You mentioned sort of at the beginning that one of the motivations of this is that it could potentially lower the costs of these benchtop setups. Do you have, I guess, like an estimate in terms of how much this actually reduces costs?
Yeah, so right now, of course, the cost amount that I'm going to tell you has to exclude the effort that I'm putting in. But if the material cost, we can easily get something up and running anywhere from $4,000 to $10,000. Yeah, so, and the plan is to, if possible, try and make it really accessible to as many people as possible. So right now we are looking to sort of sign MTAs if possible, and sort of provide a prototype at cost for people who might be interested in using our prototypes.
That's great. Yeah. Also, just another quick question: what's the temporal resolution on the neural imaging?
So right now we are getting about 10 to 15 frames per second. But of course, it's a function of the resolution. Now, if I can lower the, so this frame rate is about at 500 by 500 pixels, which is at about 5 micron resolution. Now, I have another configuration where if I drop the resolution to 250 by 250, that's with about 10 micron resolution, I can get 20 frames per second. And that frame rate will grow if I make it spatially coarse. So I know, like in most cases, you might want about 30 frames per second, and that we can achieve by, let's say, about a 128 by 128 array right now.
Great, awesome. Thank you so much again. So now we'll move on to our second speaker, who is Michael McCullough. And he is a postdoctoral research fellow at the University of Queensland in the lab of Geoff Goodhill. And his talk is on large-scale calcium imaging of spontaneous activity in larval zebrafish, reveals signatures of criticality.
Hi, everyone. Today I'll be talking about some work we've been doing looking for evidence of neural criticality in whole brain single cell resolution calcium imaging data from larval zebrafish. So just to briefly outline the presentation, I'll start off by just talking about neural criticality and why it might be important in neuroscience. Then I'll talk about why the larval zebrafish is a useful model in this context, and how the process we've used to extract single cell resolution data at a whole brain scale to study criticality. And finally, I'll show our analysis of neural avalanche statistics, which provides evidence of criticality in our data. So for those of you who are new to the idea of criticality, critical dynamics are what occurs when a system is poised between states of order and disorder. It's a concept from statistical physics. Why criticality is interesting is that in models of systems at criticality, artificial neural networks and recurrent neural networks and these types of models, it's been shown that criticality maximizes information transmission, information storage, and dynamic range, and other properties that are potentially useful for computation. Now, the seminal paper by Beggs and Plens was the first evidence of criticality in neural tissue. They were using tissue cultures from cortical tissue and electrode arrays, and they showed these scale-free neuronal avalanche statistics. I'll get onto what a neuronal avalanche is very soon, but this is a key signature of critical dynamics. However, generally evidence has been sort of limited when you talk about electrode arrays on tissue cultures or in animals. You're talking quite a small number of channels, and critical phenomena, one of the key properties is that they're scale free. So they span many scales in terms of size and duration. So in terms of electrode arrays, you suffer from an undersampling problem. The other regime that's been studied in is sort of more from EEG and MRI. So sort of larger scale activity where you don't have that high resolution. So there's kind of a trade-off here. But in 2018, Ponce Alvarez and in German Sumbray's lab published this paper where they used the properties of the larval zebrafish to image whole brain activity, and they found some evidence of criticality in this data. And the benefits of the zebrafish, they could do this in vivo and without anesthetic. The limitations of their study, however, was that they were using one photon imaging, so they weren't truly imaging spontaneous activity. So what we set out to do with our work was use 2 photon imaging to extract truly spontaneous activity and see if we could find criticality in this data. So we're starting with a data set of 5 fish at 9 days post-fertilization with nuclear targeted GCAMP6S. And using a resonance scanning 2D microscope, we recorded 30 minutes of activity from approximately 10,000 neurons over 6 to 7 planes in around the optic tectum and the cerebellum, a little bit into the hindbrain and some of the forebrain as well in some of the animals. And this was imaged at 1 Hertz per volume for 30 minutes. Obviously, now we have this imaging data, we want to be able to extract the information at the single cell level. Another one of the limitations of the Ponce-Alvarez study was that they analyzed the data on a voxel basis, so they broke it down into a hexagonal grid, but we wanted to actually segment the individual neurons. So to do this, what we used was the Cayman package that was sort of the state of the art in calcium imaging segmentation at the moment. So what you're looking at on the right is a single plane from one of the fish, just showing a mean image in the green, and in the purple are the segmented neurons from the output of Cayman. Now, this will give us a set of delta F1F traces, so normalized calcium fluorescence traces. And these are just shown in the different colored traces here. Now, what I've done is I've just picked 4 random neurons, the top of each of these groups, and found the most highly correlated, the next 2 most highly correlated neurons with this neuron, just to show that the sort of diversity of neural dynamics that we're picking up across the brain. Now, one of the problems with calcium imaging data is you have a low temporal resolution. So to be able to actually infer when neural activity is occurring because of the long delay in the calcium or decay in the calcium kinetics, one needs some more sophisticated methods to deal with this. So we use the OASIS package by Johannes Frederick and colleagues, which fits the first order autoregressive model to the calcium fluorescence traces and infers the probability of spiking or neural firing in any given time bin. This doesn't enhance the resolution of the data, simply says that for in a given time step, what's the likelihood that we actually have neural activity. And so this is shown on the right in the gray vertical bars. Now, the way we do this is this package can actually estimate the time constant of the calcium kernel. So we first fit across all of the data to estimate our time constant G0, and then we take the mean of the distribution of the estimated time constants, and then we refit that with a fixed time constant across all the data to try and make it more robust to noise and outliers in the data set and artifacts in the imaging data. We find this method works quite well. The next step in the process is we want to look at neuronal avalanches. So a neuronal avalanche is a cluster of neural activity that propagates through space and time. And to define a cluster, one needs some way of defining adjacency. So 2 things can only be part of a cluster if they're nearby. And so to do this, we just take the assumption that neurons that are close together are more likely to be connected. So we take a radius of epsilon around each neuron, and define that as the space of connected neurons. And we want to do this to make our resulting network that our dynamics are evolving on to have a mean degree of 6. Now, this connectivity is dare I say, in a hand-wavy sort of way, meant to match up with the canonical random field easing model, which is one of the classic models for studying criticality in statistical physics. This ends up giving us a radius of about 8 micrometers. So we're really capturing each neuron and sort of its adjacent neuron. And this is just by way of an example, an image of the resulting network for one of the fish. Obviously, most of the avalanches that we're going to see that we're interested in are going to be evolving on this giant component here, which has a total of 8,556 neurons. All the other fish are in a similar range of neurons after this process of extracting the data and inferring activity and then defining connectivity. So one of the key properties of criticality, so I've mentioned criticality and its benefits, but one of the key properties that we can use to find criticality is looking at the statistics of neuronal avalanches. And a lot of this came out of the work of the Sethner group in the 90s, who studied this extensively in models. So what we expect to see is that the probability distribution of the duration of each avalanche and the size of each avalanche will follow a power law distribution. So what you're seeing on the left is the probability distribution for duration. The blue are the data points of the estimated PDF, and the solid line is a maximum likelihood fit to this data. And the dotted line is just for reference, is the theoretically predicted value for the random field easing model. And we do get a fairly clear power law exponent. This is just for the first fish in the data set, I should mention. Here on for size, we also see a fairly clear power law scaling relationship. I've just noticed a small typo. This should say fish 1. This is all from the same fish. And again, one of the other things that the theory predicts is that you will get a relationship between the duration of an avalanche and the average size of that avalanche. And that should also be a power law which follows this relationship T to the 1 on sigma nu Z. Don't worry too much about what these 3 things are. Just think of that as a single exponent. The next important question to ask when fitting power laws, which are notoriously hard to fit, is to validate these. So we validate them against a log normal and an exponential fit using a log likelihood test. And for all of the size exponents, we see that they are significantly more likely to be a power law than these alternative distributions. We also see that they're all higher, but relatively close to the theoretically predicted value in this dotted line here. For the case of duration, 4 of the fish are relatively close to the theoretically predicted value. Only 3 of them turn out to be significantly likely for a power law. So this shows potentially some inconsistency in the dynamics between samples, which we are yet to sort of uncover why this is the case. For the size versus duration relationship, unfortunately, this isn't a maximum likelihood fit because this isn't actually a probability distribution. So the best we can do is compare it to the theoretical value. And most of these, aside from the large error bars, the actual estimated value on the data is quite close to the theoretical prediction. I should just note here that these error bars are 2 standard deviations around bootstrap resampling of the avalanche data set for each fish.
Michael, can you wrap up?
Yep. This is my last slide. So the final key thing here is that in critical dynamics, there is this theoretical relationship between our three exponents. And what you can see is that for three of the fish, these points are quite close together. Again, for fish 3, there is a bit of a discrepancy and a little for fish 5. But generally, we are finding some evidence of criticality in this data. So in summary, we've investigated neural criticality in spontaneous activity from whole brain data with single cell resolution for the first time. And our avalanche analysis reveals signatures of criticality in some of our samples from power law scaling and relationships between exponents. But the inconsistency between some of our results may be indicative of state-dependent dynamics or potentially just a sample size that's too small at the moment. And finally, I just want to acknowledge my boss, Jeff Goodhill, and my other collaborators in the lab who've worked on this. Thank you.
Fantastic. Thank you so much. So we're running a little bit behind right now, so I think we're just going to go straight into Philip's talk. If you want to ask a question, feel free to put it in the Q&A, and I'll put it in the chat for Michael to answer it. Otherwise, you can put it directly in the chat as well. So for our final speaker is Philip, and he is a faculty fellow at the University of Arizona. And his talk will be on fully implantable wireless and battery-free neuromodulation tools.
Hey, thank you very much. It's great to do conferences again. This one's a particularly good organized one. So this talk will be a little bit of a departure from the previous two talks. What we're going to talk about is recording and modulation tools in completely freely moving subject, and there will be a distinct difference in the connectivity to external devices. So for you who are not familiar with optogenetic stimulation, I think most people are. I'm just going to quickly go over this. Basically, you can use light in order to manipulate neurons, either excite them or inhibit their function via opsins that are genetically expressed in particular neuronal population, and that allows you to do optogenetic stimulation, quite powerful technology. The main problem when you want to look at, at behavior of naturalistically behaving subjects is that you have an optical fiber that's attached to the head, which basically limits all experimental paradigms to places where you have only one animal and very simple environments with an open lid. So what my lab does is we research towards devices that are completely tether-free. The way that we do that is by, instead of using a remote light source, we bring the light source into the target area where the light is required by using micro LEDs. So here you see one in comparison to an optical fiber. You can make these probes really, really small. And what you gained now is that you can power these systems electrically. And that helps in making these devices completely wireless and battery-free as well. So most of our devices use an external antenna that casts power to an implant that's fully implanted in the subject that then can move around completely freely. So just to give you a couple of examples of how that looks like, this is relatively recent work from 2 years ago that allows us, for example, to target 4 independent brain regions at the same time in the same animal. We have various other types of designs that allow us to do optogenetic stimulation for various applications. This one can do very precise intensity control. This is multiple bilateral stimulation with multiple areas. And this here is super light with a low footprint. These devices are in the milligram range, usually somewhere like 50 milligrams. The way that they're implanted looks like this. We use off-the-shelf components. They're all non-magnetic, which allows us to record these or image those devices in a living animal in the MRI and CT. And you can see that here. This is an animal 10 days after surgery. The only reason why we see that this animal has an implant is because we have a red indicator LED that tells the operant that the device is on. You can see basically they're subdermally implanted. And the probes are protruding into the brain region of interest. Here in the CT, you can see quite nicely how that works. So most recently, and this is data from a paper that's up on arXiv, currently under revision in NatCom, where we could extend this to completely new animal models. So obviously, this tether-free mode is great for rodents because you can do a lot of ethnologically relevant behaviors and modulate at the same time. But you can now take this to freely flying animals. These are finches. And this particular device, again, another stage in terms of evolution, allows us to do millikelvin thermography to look at core body temperature, and at the same time also do stimulation. I brought a quick video while I chat about the birds. Let me quickly activate that. So here we basically have two devices that are independently programmed for the optogenetic stimulation, and at the same time we also record from both of these devices. I hope you guys can see that. So these particular systems are made for finches. The reason why you go into that animal model is you can study language acquisition really nice, because the birds, they sing. The males do it pretty regularly every day, and they learn their song from their parents. So by modulating, you can then look at the way that language is acquired. So let me move on. You just saw here one device, getting the temperature raised by just a hot air gun close by, and the other one does too. So you can see that here independently. So here's a device, again, implanted in the finch brain. And what you can do is, this here is a song. Each of these are syllables of this song. And what we look at here is syllable B and F, where we target the area X. It's really called that way, I'm not making this up, which is responsible for the formulation of the song. We could optogenetically manipulate and therefore lower the fundamental frequencies of these syllables quite nicely. So that's more of a proof of concept for this technology. So moving on to a general concept in neuromodulation. Basically, all of the techniques that we have are invasive, and they leave neural scarring no matter how good you are in terms of miniaturization. These are our probes. These are fiber probes. We reduce it a bit, but it's still there. It's just a matter of putting something in the brain will cause damage. But we came up lately with a method to completely avoid that. And with optogenetic stimulation, you already have specificity through the genetic part. And the good news is you don't actually need to penetrate the brain in order to bring light into the brain. So what we could do is create these devices that produce high intensity flashes of light that penetrates the skull all the way into the deep brain, where you can then target specific neurons, and you get the cell specificity and the local specificity with the optogenetics. And we could do that in a completely wireless and battery-free fashion. So here's one of these devices, again, similar implantable mode. This here is a Monte Carlo simulation where we set a threshold of 0.1 milliwatt per millimeter squared in terms of activation. And you can see it can go up to 5 millimeter deep with, this is red light. And the thermal impact is also absolutely minimal. This is the worst case. I'm just summarizing this real quick. Basically, you don't have more than 0.3 degree in delta for full power at the brain surface. And that's the worst case, not including perfusion. Okay, so here is a quick experiment that showcases the viability of this, where we modulate the motor cortex. We express crimson R, which is an opsin that does that works at red wavelengths, and the activation of the motor cortex is pretty intense. We measure this on the Racine score, so we can induce seizure-like behavior with these type of technologies. So works pretty nicely. This is a project that's currently in development. The other two are submitted and review. So here, if you're interested, this is in collaboration with the Matt Trash Group at Northwestern as an NIH-funded project that looks into making devices that allow us to electrically stimulate the peripheral nervous system. And here we can target the cord, but also have the fidelity to do muscular stimulation. So here you just have a couple of key metrics that I want to highlight. We can hit a compliance of plus and minus 20 volts with these completely battery-free subdermally implanted devices with pretty high fidelity, 8 channels, so pretty broad capabilities in a tiny package with off-the-shelf components. So these devices are throwaway devices. So if you implant them once, probably will be cost around $40 at most. So you can do multiple subjects. What I want to do is shift gears real quick. We talked a lot about imaging in the previous talks, and I want to highlight work that's not imaging, it's photometry. So we have a single pixel, but the good news here is that the system is highly miniaturized and completely fully implantable. So the way that we can do that is by having the light source for the stimulation of the fluorescence, the detector and optical filters all implemented in one probe that's 300 micron across and 100 micron in thickness, so much smaller than an actual fiber photometry system that operates completely wireless and battery free. So if you want to know more about the system, this is published in PNAS 2020. Again, familiar makeup, different types of electronics and similar implantation. This here is an animal 10 days after surgery, MRI, CT, all non-magnetic components. Just to look a bit at performance of this, this year's work from 2018 PNAS paper, where we done this with battery supply. This particular device that I showed you beforehand is the same, but battery free. And here you can see the performance in comparison to fiber photometry. Here we stimulate the BLA in an experiment that it delivers a foot shock. BLA is associated with anxiety. This is a known paradigm to induce that. And you can see here before, after, quite nice correlation between both of the fiber and the wireless device. Lower noise floor and roughly the same delta for our system. What I want to leave you with, this is my last slide, is the possibilities. Obviously, these devices are being used in rodents because all the genetic tools are there, but you saw with the previous paper, we can extend this to subjects that are moving in 3D, and we can also use this in completely ethnologically relevant environments for rodents. So this really expands the way that you can do your experiments. And it would be great if people make use of that. So this was, by the way, with Haijian Kai's group at University of Arizona. So here's just an example that you couldn't do with fiber beforehand. This is an enclosed box, tiny entrance, light, dark situation, and water mazes that you can now do with multiple subjects if you want to, because there's no tethers. So with that, I'd like to conclude. Thank the people that do the work. This is my lab in sunny Arizona. So if you have any kind of question, please feel free to ask now or get in contact with me via email.
Great. Thanks so much, Philip. That was really cool to see like how small you can make things and how, what that really enables. So Janaka asked a while back, and I just want to make sure he asked, can you do simulation while in an MRI?
Yes. I don't know if I have it on the slide. Let me quickly check if I have this. Should be here. Yes. So you can actually see it here. In this slide, this is running in a 7 Tesla animal MRI, and you can see the blue light from the device. So yes, we can do that. You can stimulate while you're inside an MRI.
Awesome. Great. So Elizabeth asks, can you do closed loop between photometry / recording and stimulation?
That's an excellent suggestion. I hope my grad students here listening as well. Maybe. In principle, yes, right? We have the capability to stimulate and to record, and that can be unified in one platform, but there's no paper out yet.
I guess, so in terms of this being wireless, are there any constraints in terms of distance, or is that not really too much of a concern with mice?
Yeah, that is, so we can, for most of these devices that are shown you, so actually these high intensity devices, that particular paper is introducing a new type of technology that allows us to go very long distances, actually a meter by 60 centimeters by a meter 50. So quite long and large areas, but most of the devices that you've seen beforehand, they're in a 30 by 30 or 40 by 40, kind of environment, which, most of the paradigms for rodents are this anyway. But we're making headway towards larger rooms where you could look at play cells, where you could, look at various kinds of things that you couldn't do otherwise with tethered technologies.
Great. I guess one real quick question is how finely can you control the stimulation on versus off?
Yeah, we have, they are microcontroller controlled lights. So you have microsecond resolution in terms of your stimulation patterns. So, and there's no limit in terms of speed. Like you would, the options will give up first. That's for sure.
Great. Well, yeah, thanks again for your talk. I don't think there are any other questions. So just want to thank all three of our speakers again. Really fascinating talks, really interesting to see, the cutting edge of what we can do from both a recording and a computational standpoint. I also want to thank Kevin for managing things on the back end of this, and thank you all for listening. And for managing things on the back end of this, and thank you all for listening.
All right. So this talk will be available on YouTube when all is said and done, unless you'd really rather us pull it. Up to you. All right. Shut down.
Thank you all for listening.
Let's give it another minute or so for people to come on in.
Sure.
There we go. But yeah, thanks again for agreeing to do this. It's been great getting this all set up.
Pleasure. It's a great honor.
Yeah. This time slot is pretty far off our peak in terms of audience.
I bet.
Filtering me after dinner. And I've learned to come armed with a fresh cup of coffee because I know that after just having eaten,
oh goodness,
no matter how interested I am, it's been a rough day. All right, why don't we go ahead and get started? Okay. I'm going to introduce my good friend and colleague, Paul Dux, who is going to tell us about his work on the causal neural substrates of incidental statistical learning. Paul got his PhD at Macquarie University in Australia, then moved over to the States to have a postdoc with René Marois at Vanderbilt University, and is now a professor at the University of Queensland in Australia, which I have visited, and it's a fabulous place. Paul is a wonderful host. And so without further ado, I'm going to turn the stage over to him. And I also want to remind people before I disappear here that you should please feel free to enter questions in the Q&A panel. And when this is done, I will address them to Paul. Take it away.
Thanks very much, Brad. I'd like to begin, as is custom in Australia, by welcoming you all to country and the traditional lands on which the University of Queensland is situated. I want to pay my respects to elders past and present, recognise their continued connection to these lands and the important contribution they make to Australia and global society. I also want to just send my best to all of you out there as well. Hope you and all your loved ones are keeping safe and well during these very challenging times. And all the best as we move forward through this pandemic. So like a lot of psychologists, and particularly cognitive psychologists, I've always been interested in the problem whereby we get we're presented with all this information in our visual or sensory environment, far too much for it all to be processed up to the level of awareness, and how to extract meaningful or important information from irrelevant information. And many theorists have kind of suggested there are two operations that one needs to perform in order to achieve this task. So the first is that we need to be able to individuate objects as distinct perceptual events that occur across space and time. But at the same time, it's also very important that we extract features from those stimuli as well. So we need to identify the features as well as individuate these objects as distinct stimuli. And of course, we need to put that information together because, you know, if you're on a reef just off the coast of Queensland where I live, it's important the thing moving towards you rapidly. Yes, it's a distinct perceptual object, but there's a big difference if it's a big grey shiny thing like a shark or a large, beautiful reef fish. So you need to put together spatiotemporal information and also featural information. And many people think that attention's playing the key role there. Its role is to kind of extract relevant from irrelevant in the environment. But of course, there are other operations that we undertake as well. And one of the most important things to realize about our environment is that it's not all random. Very much there's a lot of structure in the environment. And from this structure, we can also help navigate our world because that structure can be used to predict what's going to happen in the environment and we can learn from this information. And that's what we're going to be talking about today. So incidental statistical learning, how we extract regularities from the environment to influence cognition, perception, and other psychological processes. Such learning occurs very, very rapidly. We're very good at extracting structure and regularities from our environment. And this also appears to happen very much in kind of an implicit or automatic process, this type of learning. But of course, that's been a hotly debated topic and can be somewhat contentious. But definitely, this sort of statistical learning does seem to happen very much incidentally. We don't have to be engaged in learning process or consciously try to learn to undertake these operations. And across a wide range of everyday tasks, such learning is very important. So if you look at very young infants, they are very good at learning language structure. In fact, some of the earliest work on visual statistical learning is out of this infant language processing, whereby particular phonemes are likely to occur close together as opposed to others. Of course, in music, there are notes that appear much more close together and much more highly correlated in their structure, same with chords. And also in any visual scene, you're much more likely, for example, to see a dog walking on a sidewalk than to be on someone's head as they walk past you. So there's structure in scenes as well that helps influence our cognition and perception. So in terms of what I'm going to be talking about today, I'll begin by giving a little bit of background, just some brief information on different paradigms people use to examine implicit or incidental learning. I'm going to talk about the neural substrates of incidental learning and paradigms that have been used to assess this. Then I'm going to talk about not only the tasks, but also a particular brain stimulation approach we use here, transcranial direct current stimulation, which I'm sure many of you are familiar with. And I'm then going to go through 3 studies that are all kind of looking at, is there kind of a unified network involved in statistical learning across the paradigms we use to assess it. And so I'll be looking at using brain stimulation in 2 of the studies. And in the final study, I'll be talking about a large individual differences study where we look at the correlations between different statistical learning paradigms. And at the end, of course, I'll try and put it all together and try and answer that question of to what extent do these tasks and these types of learning across time and space interact and overlap in the brain. So in terms of key paradigms that are used to assess incidental statistical learning, a chief one is spatial contextual cueing. This, of course, was introduced by Marvin Chang and Yuhong Jiang back in the mid-90s. And essentially, if you're a participant in this task, it looks very much like you're undertaking a standard inofficial visual search paradigm. You're looking for a T rotated either 90 degrees to the left or right, and around it are orientated Ls. And you're trying to find this T as quickly and as accurately as possible. But generally, unbeknownst to the subject, some of those distractor arrays are repeating. So you might have 12 independent but repeating displays across a group of trials. And what one sees under these conditions is that across the blocks of trials, you start to see that people respond faster to repeat trials as opposed to non-repeat trials, suggesting that people pick up on these regularities in the distractor structure. Another key paradigm is that of the visual statistical learning approach. That was introduced by Fisher and Aslan, but it's been used by a wide variety of other labs as well. This is somewhat different to visual statistical, to contextual cueing, because here you're learning the relationships between shapes. So there's two phases. Initially, there is an exposure phase where you're just being presented with these screens filled with these shapes. You don't have to do any task, you're just watching these stimuli. Well, that's in the classic approach anyway. But again, unbeknownst to subjects, these shapes appear in pairs. So across the trials, particular stimuli are being paired together. And then after this exposure phase, that can be happening quite briefly, 10 to 15 minutes, you then have a test phase where you're presented with a base pair, so one that has been repeating, versus a foil pair, which never appeared close together in space in the task. And what one sees here is that you get people to say, which one are you more familiar with? People generally pick the base pair they've seen before, and they're above chance. So again, this shows evidence that people, despite having no task here, never being instructed to learn these stimuli, do in fact pick up on these statistical regularities in these sequences of shapes that they're presented with. In terms of the neural substrates that have been implicated in incidental statistical learning, so one approach has been using patient studies. So Marvin Chan and Elizabeth Phelps had a study where they looked at patients who had hippocampal damage, and what they found was they didn't display the standard contextual cueing effect implicating the hippocampus in this spatial implicit learning. Similarly, Barry Giesbrecht, like Nick Turk-Brown, have used functional brain imaging to examine the neural substrates associated with contextual cueing, or VSL. Particularly if you look at hippocampus, you start to see increases in pattern similarity as you're exposed to these pairs of stimuli. The representations start to overlap more. So again, that also implicates the hippocampus and other subcortical structures in this type of learning. But of course, imaging is correlational, and the patient work has generally focused on subcortical regions. And so what I'm going to be interested in the studies I talk about today is, well, is there a causal role of cortical nodes in incidental statistical learning? So in order to do that, I'm going to employ transcranial direct current stimulation. So this is an approach whereby you place electrodes on the scalp, one anode and one cathode, and you pass a weak electrical current between them. That, of course, is thought to influence the cortical processing underneath these electrodes and also in connected regions as well. There's many different approaches to transcranial electrical stimulation. In transcranial direct current stimulation, generally you have anodal stimulation. This is thought to be excitatory, inhibiting the neurotransmitter GABA, and leading to a depolarization of the resting membrane potential, so increasing the likelihood of neural activity. Conversely, the cathodal inhibits glutamate, and as a result, it's thought to lead to hyperpolarization of the resting membrane potential, and so inhibiting neural activity. Now, that's, of course, a very simplified way of thinking about how TDCS works, but of course, It's not a one-size-fits-all method. So for example, it's influenced by stimulus intensity, stimulus duration, whether a protocol is online or offline. And what I mean by that is whether you stimulate as people undertake a task versus do not undertake a task. And it's also influenced by what type of paradigm people are engaged in. You know, is it a motor paradigm or a perceptual paradigm or a working memory paradigm, for example? So it's quite a complex technique which has a lot of nuances to it. And so these classic effects of anode exciting, cathode inhibiting, it's really a simplification. That's not always what one sees in literature. Now, in terms of the first study in our approach here, so this was examining, or does brain stimulation used while using transcranial direct current stimulation of the left posterior parietal cortex and left prefrontal cortex, does that influence contextual cueing? And we used a standard contextual cueing task. We provided subjects with 0.7 milliamps of stimulation, 15 minutes. And so that was throughout the whole task. So this is a completely online paradigm. And we had 120 subjects across 6 groups. And that is whether they got parietal stimulation or prefrontal stimulation, and whether they got sham, anode, or cathode. And what we found under these conditions is that we got clear learning across the shams groups, the parietal groups, and the cathodal groups, both in the frontal groups and the parietal groups. However, particularly in cathodal stimulation, what one sees is that you actually impact learning early. So we had a significant impact here, whereby initially you have very early learning, contextual cueing in the sham condition, which is not uncommon. And that was actually inhibited by cathodal stimulation, whereby you didn't get learning as quickly where you're having cathodal stimulation versus sham stimulation. So it appears that cathodal stimulation of the parietal cortex and indeed PFC appears to causally influence these early stages of contextual cueing. And so this does implicate these cortical structures in incidental statistical learning and causally implicates them, not just correlationally. But of course, this is just one paradigm. And so in the next study, we're interested, well, does brain stimulation affect the standard VSL approach? So here we use the typical Fisher and Asline method. So again, passive viewing of these shapes. Again, base pairs are repeated throughout. And then afterwards, we presented people with the base pair again and a foil pair and examine learning. And here we had a left posterior parietal group. We focused on the parietal cortex. We had an orbital frontal control and also a sham group. And in each of these, there were 50 subjects. But we got bubkus. There was no impact of TDCS on VSL here at all. And this is very much like Bayesian evidence in favor of the null. So we didn't find that despite getting significant learning, it wasn't different across the groups. So it appears that parietal cortex doesn't influence VSL. But of course, when you're undertaking visual statistical learning, you're not accessing those early stages of processing. You have an encoding phase or exposure phase, and then you test it afterwards in this familiarization task. So what happens if we use a VSL paradigm where we can actually access these early stages of learning? And so that's what we tried to do in our next study. And so we developed a new task. So using very, very similar stimuli, but now we had a target pair. 2 stimuli that were either oriented in a different direction. And subject's task here is to just say on every trial, and each trial is 3,000 milliseconds long, do you see this task or this task? And they're responding as quickly, as accurately as possible. And then we have these distractor pairs that appear here. We actually make them appear twice to get an appropriately cluttered scene. And of course, we're repeating these pairs throughout. And in the variable target location, the identity of these distractor pairs tells you nothing about where the target is likely to appear. However, in the consistent target location, the identity of the distractor pairs predicts where the target pair will occur. It doesn't predict the orientation of the target pair, so it doesn't tell you about what judgment you have to make, just where they will occur. And again, this isn't a spatial layout predicting target location, it's the identity of the stimuli here, just like this is the case in the standard visual statistical learning paradigm. Here we adopted a pre-registered double-blind design, so we only had the parietal montage here. And the reason you have that is because in our TDCS machine, we don't want the experimenter or the subject to know which they're getting. So you input a code, and then of course you either get sham in the parietal lobe or cathode. And again, we're very much interested in early versus late processing. Of course, we stipulated that in our pre-registration. And what we find here is that in both groups, again, we do get learning here, but now we're actually able to look at learning across the blocks. But what you see here is that the cathodal group does actually show differences in learning from the sham group. However, this time you actually see an enhancement in learning earlier relative to the sham group. So learning appeared earlier in this task for those who received cathodal stimulation relative to sham stimulation. So TDCS of the PPC does appear to causally influence both contextual cueing and visual statistical learning. So one being a spatial type of learning, the other one being more an identity-based type of learning. In our last study, of course, and we can be quite confident in the results, it was very much a large sample, it was pre-registered, and we also used a double-blind design. But we got the opposite effects. While stimulating PFC both appears to influence these learning approaches, it influences in different ways. So we got impairment in the contextual cueing task and enhancement in the visual statistical learning task. So I guess that leads us to the question, well, does contextual cueing and visual statistical learning, do they draw on overlapping processes? And is there some sort of unified network involved in both these types of tasks? So in order to do this, we ran a large pre-registered individual difference study. And so here we examined different types of statistical learning and also how they relate to some common executive function and attentional tasks. We ran people, they undertook an attentional blink task, a go-no-go task, a single task, and a dual task, so a multitasking paradigm. We measured working memory with an O-span task, and then they took the standard Fisher and Naslin VSL task, the standard contextual cueing task, similar to what we've used previously, and they then undertook a learning task, but this is a motor task, and so this is the serial response time task, or SRT. And what you participants do here is every time a stimulus appears in one of these boxes, they press the finger that corresponds to that box. And gradually across training, unbeknownst to the subjects, there are patterns that are repeating. So there might be a run of 10, which are consistent, and that keeps appearing. And then we examine the reliability of the tasks and how the effects on contextual cueing and other measures correlate with one another. So first off, we got the standard sorts of effects one would expect. So we got contextual cueing and we got good statistical learning. And we also saw what one typically sees in the serial reaction time task. So initially you have slower performance, then you start to see learning as these patterns repeat. And then if you make the patterns random again, you get reaction time really increases. So we get our standard learning effects there. And we also find that all our tasks were reliable. However, interestingly, while our executive and attention tasks had very high reliability, well over 0.7 in each case, it was as low as kind of 0.37 for contextual cueing, 0.39 for visual statistical learning, and only 0.58 for the serial reaction time task. So while all the tasks are reliable, you did get decreased reliability for the learning tasks here. And if you look at how these task effects correlate with one another, we actually found no evidence of all, despite our large sample, the contextual cueing, the serial reaction time tasks, and visual statistical learning were associated. And in fact, the only measure that correlated with any of the statistical learning paradigms was operation span, and that correlated with the Fisher and Aslin paradigm, or the results we found from that paradigm. So all tasks are reliable, all our learning tasks, but there's no association between them, or we didn't find any associations between them. And the only thing that was correlated was operation span, or working memory capacity. So it does appear that incidental statistical learning does appear to tap these distinct cognitive processes. So to the question, does contextual cueing and VSL draw on the same processes, and is there kind of an overlapping causal neural network that underlies these paradigms? I guess it's kind of yes and no. We do find that the parietal cortex is implicated in both VSL and contextual cueing, but they appear to draw somewhat, at least, on distinct psychological operations. And it may be that's because if you look at contextual cueing, VSL, and our SRT, our task, is they're each tapping different types of learning. One's looking more at individuation, so looking at the temporal and spatial properties of stimuli, so contextual cueing. It's all about the spatial array of distractors predicting target location. Whereas our VSL paradigms are very much using identity to predict target location. It's not providing, or you're not learning spatial information. And finally, with the SRT, you're using motor operations there. So that may be one hypothesis why we're seeing different types of learning there. And it may be that the parietal cortex is just involved generally in a process that's important for this type of learning. I'd like to acknowledge my collaborators here, Dr. Abby Neidham, who's just finished her PhD and is trying to make her way to Canada to start a postdoc with Jay Pratt, and my colleague in the School of Psychology here, Dr. David Sewell. I'd like to thank you all for your attention. That's my lab website if you like to read any of these papers or indeed any of the other work from my group. I'm happy to take questions. Thanks again. Thanks, Brad.
As always, it's the solo clap from Zoom audiences.
Yeah.
Yeah. So first of all, you know, my sincere hats off to you for doing the hard work in running these studies in the most rigorous way possible. I mean,
well, thank you. I think that's it's important in all science, but I think particularly in brain stimulation, I think it's very important to do that. So, you know, we try to do registered reports and always pre-register our results and analyses. And by no means, you know, it's not a jail, it's a recipe, right? And just when you're cooking, you can sometimes add more salt and more capers, and it's the same also with pre-registration. Yes, I think it's very important, but it's also malleable as well. And you know, I think people need to realize that to kind of encourage more of these open science practices to be used. Just tell everyone what you do, and it makes things very easy.
Malleable in the sense of, you know, you say you're going to do X, but then you realize that you had to do Y after all.
Yeah, exactly. As long as you tell people that, that's fine, right?
Yeah, right. I mean, is there I mean, there's a point at which that essentially becomes kind of like what we had been doing before. Is there like I guess, how do you distinguish between, I guess, in effect, p-hacking afterwards, but saying, and then, you know, we tried this and this didn't work, so then we did this instead.
I think it's the documentation beforehand. I think things like, you know, the open science framework and those sorts of things. I mean, yeah, I mean, if you run a study and then change something radically it becomes exploratory, right? And means it's exploratory and, you know, it should be taken as such. Whereas in the past, I think people made changes and didn't say they were exploratory, right? And planned and exploratory make a difference, right? I think just now, I think particularly with brain stimulation and other approaches, having that documentation of each path you took, I think it makes one able to draw more definitive conclusions from the results, I think.
Yeah, and TDC
modelers have been doing this for a long time as well, right? So it's not just, you know, modelers build models and write code and, you know, say where additions have been made to models. So for computational people, it's been doing this for a long time, I think.
Well, I wouldn't say that. I mean, it's the
I hope so, anyway.
I mean, the same issues can exist in modeling.
Oh, that's true. Yeah,
where,
of all science.
Yeah. But yeah, and I also really like all of the individual differences work that you do and your efforts to make sure that things are robust and reliable within task. So I mean, I find it interesting that there's no link between, for example, the attentional blink and statistical learning, because one of the things that we've been doing is sort of thinking about the link between the attentional blink and how it might help structure our ability to form meaningful statistical relationships in the world. So I mean, my sort of prior would have been on all this, that there is some kind of link between the depth of the attentional blink and the effectiveness in creating distinct representations, with the caveat that that would entail paradigms for which there is a continuous streaming of information, where things are changing relatively rapidly over time.
Yeah, and I must also, I mean, pay kudos to you. I mean, you're one of the very early people in attentional blink research to actually say, well, what's the attentional blink for? You know, as opposed to this is the attentional blink. And of course, your theory very nicely articulates in your results also that, you know, what we're using is trying to segment perceptual events, right? And that's, you know, basically the attentional blink occurs because we have these mechanisms in place to try and do that. So yeah, I'm surprised as well. It's only one study. It's only a couple of learning tasks and one attentional blink task. You know, I'm quite confident in the results. Live sample, we pre-registered it, but yeah, I was surprised at that, given kind of what I think the attentional blink is for as well. I mean, one thing we did find that you know, working memory did correlate with our OSPAN task. Now, working memory is highly implicated in the attentional blink, and there's lots of individual differences studies that are doing that. So, you know, maybe if we took more of a latent variable approach, we had, you know, a wider range of tasks for indeed BSL and the attentional blink, you might see that learning coming through. But other than that, I can only kind of just say that, you know, we didn't find evidence for it in that study, but it's an ongoing question.
Well, so in that study, right, so the VSL is what each stimulus array is presented for 3 seconds?
Yeah, exactly. And this wasn't the one where we actually had them looking for a target. That was the passive viewing one as well. So it may be during passive viewing, you don't have the same sort of things that you do in an attentional blink, which of course is I guess it's passive as well, but it's very much, you're looking for a target and everything, and you're making a response on each trial. It'd be interesting if we used the second VSL paradigm that we introduced, the one where you're actually making a target judgment, if that then correlates with the attentional blink, or indeed other measures of temporal attention.
Right. So this was just passive viewing.
Passive viewing, yeah.
And was this the one with was there an array of many different characters, or was it a small array?
No, it was a similar sort of array with many different characters. So it wasn't like just a pair of stimuli being presented.
And eye movements were unconstrained, and you could move around. And
yeah, eye movements are unconstrained. Yeah. We've done some work actually. You can get. We've got a study we've been working on where this is the contextual cueing, where we only present the information very briefly for say 300 milliseconds to kind of constrain eye movements, and you still get learning under those conditions, even when you backward mask it. It's not as big, but humans are pretty quick at extracting this sort of information, even from like very large arrays in a visual search paradigm.
Right. Well, we've got a question here from someone in the audience. Haida Maria Sigur, let me try this one more time. Sigur Dartotir. And Haida asks, how much of a problem do you think it is that people operationally define the quote unquote same construct in different ways? Are they tapping into different mechanisms and therefore getting inconsistent results? And this person is in Iceland, and it's almost midnight there.
Well, hi, thanks for tuning in such a late hour. That's a great question, actually. And I think, you know, I've always been someone who kind of tries to not study tasks, but studies processes. And if you're studying processes and not tasks, well, generally, you want to use a wide array of tasks to try and get at a particular construct. So I think it can be very challenging if, you know, imagine you had a theory of learning and one group used one paradigm and another group used another paradigm, and we're constantly arguing, you know, back and forth with one another in terms of what gives rise to a particular learning effect. Well, that'll be very challenging, right? Because it could be that the results in the theory only generalize to each other's paradigms. So I think, you know, in terms of operationalization and indeed theory development, it's important to use a wide array of paradigms when you're trying to get at these or generalize your results. I hope that answers your question okay.
Yeah, I mean, I completely agree, by the way. I think that that's one of the biggest issues that we have in cognitive psychology is that people get married to a specific paradigm. And then we spend a lot of time sort of following the same idea. And you can have, you know, two people studying almost the same construct, but with
exactly
different versions of the paradigms, and you speak different languages, you have different manipulations, and you can, you know, exist in the same department and yet not have a very effective language for communicating across that gap.
Yeah, look, and I don't want to come across as all high and mighty. I mean, we definitely have paradigms we use quite consistently in my lab. For practical reasons, they work. We get, you know, solid replicable results, and that's very attractive. But of course, one does that. Generalizability of the results, you know, is something that's impacted by it, right?
But by using a variety of paradigms, you sort of
you get to overcome that situation.
Those things, right?
Yeah, of course. Yeah,
and that's always been one of the strengths of the research coming out of your lab. You know, is having that broad approach of looking for processes by using a variety of different well-established paradigms that everybody in the field knows and trusts.
No, well, thanks, Brad. That's very kind of you to say.
No, it's really great. So yeah, the more I think about it, I mean, when we talk about the attentional blink, for example, we sort of cast a pretty broad net in terms of what we think the implications will be for learning. But of course, there's going to be a lot of different kinds of learning, and presumably a mechanism like the attentional blink would only be involved in a sort of narrow subset of those different types.
Well, I guess it's how broad you think attentional blink characterizes something about attention, right? So, if it's looking at a small aspect of attention, then maybe its impact is small. But if it's if it taps into something fundamentally about attention, then you imagine that you might get even more generalization across learning. So, I mean, I've only talked about a very narrow aspect of learning here. There's, I do a lot of stuff on response selection learning, and there's perceptual learning as well. And I think the overlap between training effects, I think the overlap's kind of very interesting. But yeah, I mean, I've done some work on training in the attentional blink, but in terms of pure learning, like a paradigm where you combine learning with an attentional blink task, there is some stuff on, we did once where you can learn from an RSVP stream, like you can learn distractor, repeat distractors kind of predicts temporal target location. You can get learning there. But that's really kind of the only stuff. We never kind of revisited that stuff, but it's kind of interesting because it does go to what the AB is actually doing or what it reflects.
Is that what you did with that with Arena Harris? Is that right?
This was with a student of mine, Carly Mayberry, who's now a clinical neuropsychologist. So, but Arena and I have been interested in those types of questions. Yeah.
Right. So, in your experience, what are the sort of most robust locations that you found for getting replicable TDCS effects?
Well, I mean, the areas that I generally am interested in is the ones we stimulate, right? So we get quite good replicable effects. We do a lot of prefrontal cortex stimulation, and that's only because that's the area that I'm interested in. I mean, of course, a lot of brain stimulation work is generally focused on motor cortex, and there's a good reason for that, right? Is that the physiology is known very well there. It's very, very lumpy. And also you can get objective measures of stimulation, particularly things with TDC, like with TMS, for example. You stimulate motor cortex, left motor cortex, your right thumb can twitch. Right. And indeed, that's often used to kind of isolate, a particular area for stimulating and indeed the intensity that one's going to employ. And also, you can use TDCS in concert with TMS as well. So using TDCS over a motor cortex before you stimulate with TMS influences the impact of motor evoked potentials, for example. So, I think a lot of people use brain stimulation of that region because, it's easily measured. But yeah, I think it's not like you could say there's one region of the brain that's more replicable than others in brain stimulation.
Okay, right. Yeah, I take your point about the motor cortex being, and I guess also the visual side as well.
Yeah, you can do phosphenes in V1, for example, with TMS. So, but phosphenes again still require subjective report.
Right.
And it can be hard for people to generate to understand what a phosphene is.
Yeah. Do you get phosphenes with TDCS as well?
I'm not sure. I don't think so. No. I mean, again, with TDCS, you're not actually the current is not strong enough to elicit action potentials. It's really creating conditions whereby you're more likely to have activity. So that's why I don't think you see phosphenes. But I'm not an expert on visual physiology and using TDCS in vision. So I'll leave that for the experts.
So we have another question by Tijo Grutzwegers, who very sorry if I butchered the pronunciation of that name. Sorry, actually, she is. One second. Actually, we can ask her up on stage. That's one of the features of this.
Well, it's actually a he.
I guess you should have left me to figure that out.
Yeah. I just was watching you drown a little bit. It was
yeah, thank you, Paul. Thank you for throwing me a lifeline there when it was too late.
No. Hey, Tal, how are you?
Hi, Paul. Thanks for the great talk. Can you hear me again?
Thanks. Yeah, very clearly. Good morning.
Yeah, I was wondering with the individual differences studies, whether you are using just undergraduate populations or moving to more general populations, because that will give you a greater variance, right?
Yeah, so we do both. So this was with an undergraduate population. It was actually a large fourth year honors project, and so we used back in those heady times where there were heaps of students on campus, you could run undergraduates. So you did that with that group. But my group's also done stuff where we've done general population. We did a study at a museum, for example, and we ran kids, their parents, older adults. So we've done that. We've also recruited from the general population. I mean, of course, one of the challenges is, and this is true certainly of many universities in Australia, is that even when you're getting these general populations of individuals, if you're advertising through a university website, often it still does lead to it's always got a high level of education generally. So it's hard to get at the real general population. Sometimes with our older adult studies, we have challenge with that as well. But yes, short answer to your question is yes, we are using other populations as well because it does increase variability.
And as a follow-up, do you see any reliable difference there that is promising for other people doing this kind of work that would suggest that it's better to use the larger sample population? Is there anything specific to the undergraduates that you don't see in the general population?
We've never done those studies. I mean, anecdotally, I've never seen big differences there. So a lot of the tasks we use, are reliable in both groups, for example. I mean, I guess people like, the work coming out of Randy Ingles' group would be, could comment more on that. But we've never actually done a contrast where we've looked at, do you find a difference between the undergraduates at University of Queensland or just people from the general population, which of course will bring in people who live around this area. Less so for online studies. We haven't done as many online studies, but it's a good question.
Thank you.
See you, Tal.
Thanks, Jill. Right. Let's see how this works. I'm relatively new to being in webinar in this context. Can you do these studies with online studies? Does that work pretty well? I guess it's too long, right? Because that battery probably takes like.
Yeah, I mean that was a 3-hour battery. So I mean, I guess you could do it online, but you'd have to have very, very willing participants. And generally, we like to get people in. That was actually a split half reliability. I should have said that. Generally, we like to bring people in for 2 times each. Yeah. But again, we have run studies on iPads in a museum where you can have like thousands of people through a day. So you test for a couple of weeks and you might have 3,000 participants all doing cognitive tasks on iPads. But again, like anything, you lose a little bit of control and you can't run as many tasks. So, ideally for this type of work, even in this one, right? So we ran a lot of tasks. I'd love to have run more. I'd love each measure to have 3 measures so you get a latent factor for each. But again, then you're running into very, very significant challenges in terms of just recruitment. So it's always about finding this balance. So yeah, you could run it online, but you just have to make some changes, I guess. So I guess one thing about the current period is it's made us all a little bit more aware of online options for running studies.
Yeah, absolutely. We've been moving to Pavlovia and PsychoPy as well.
Yeah, we've done some of that sort of stuff as well.
Limitations. Yeah. And so you didn't find, just to correct my memory, you did not find even a correlation between contextual cueing and VSL.
Yeah, none of those 3 tasks correlated in that study. So yeah, again, I want to say it's only one study, but it's not. I mean, it's not something like I'm neither surprised or unsurprised. Like, I mean, I think there's good reason why you think that they may be tapping distinct things anyway. So I mean, think about things like the attentional blink. I mean, look at a task like repetition blindness, so similar, but are doubly dissociable, right? And I'm not sure if anyone's done a correlation between those, but yeah, they purportedly tap different things: one individuation, one identification, or bottlenecks in those processes. And so, those are very, very similar tasks, or they can be run in a very, very similar approach. So yeah, maybe that's the reason why you don't get those sort of correlations.
It's humbling, because I think that we have a tendency to dramatically oversimplify the number of different latent capabilities that people have, right? Down to like, maybe 5 or 6 different forms of whatever, and it could be 500 or 600 measures.
Oh yeah, very much so. Yeah.
So, given, and so where are you going to go next with work like this, right? Like it's difficult to find relationships between these tasks. What are you hoping to do next?
Well, I mean, I would love to do a study where we kind of combine this work as well. So this is one study on contextual cueing, one study on visual statistical learning. It would be fantastic if we had a brain stimulation approach where we touched some different regions, but a paradigm where you looked at both processes in an overlapping manner. So you could have like spatial prediction, identity prediction, and see in the same group of studies where they're both getting stimulation at exactly the same time, you know, then do you see the similar sorts of dissociations, enhancement in one, inhibition in the other. And then, yeah, running, you know, running a learning task maybe with, 3 visual statistical learning paradigms, 3 contextual cueing paradigms, and 3 modal learning tasks, and seeing, you know, what happens with the individual differences there. So I think that was kind of for this specific work. But we're always interested in kind of relating learning across different types of operations. You know, I'm very interested in training and attention and those as well. So how those sort of relate. So that's more part of the broader picture of what the lab is working on.
Well, you're doing the Lord's work, Paul, trying to put these different things together. And I know how hard it is to find relationships between these tasks. I mean, for me as a modeler, I can sort of cast off armchair theories about how X is related to Y. But you know, testing that out and finding those relationships is often a lot harder than we speculate that it'll be in our grants. So.
But it's yeah, exactly, yeah. But that's a lot of fun as well. So
yeah,
that's kind of interesting. So yeah.
Well, I have another one of these to run in about 2 minutes, so I'm gonna have to.
Okay, great, Brad. Been great seeing you.
Yeah, it's been fantastic to see you again too. Yeah, I hope that we can meet up again in the not too distant future when at least some of this craziness is beyond us.
Very much so, yes. I really hope to be able to get back to the USA soon. So we'd love to see everyone over there. So, and likewise have people visit over here.
Yeah.
So great. So. Great.
All right.
Thanks so much.
Bye,
see you, Brad.
everybody. Yep.
Bye.
Okay.
Right. Okay, Elnaz, I'm going to make you host again.
Okay. Awesome.
All right. So participants are going to start coming in.
Should I hit the broadcast now?
Yes, start the webinar.
Okay, we are going online.
And there I see people coming in. And there's going to be a split audience. Some people will be here and some people will be in YouTube. Hello, everyone. It's my great pleasure to introduce our next keynote speaker, who is Hitoshi Okamoto. And he received his PhD back in 1988 from Tokyo University and has served in a variety of positions at different places in the world and is currently the team leader in the Lab for Neural Circuit Dynamics of Decision Making at the Center for Brain Sciences in the Regan Kao Collaboration Center. And he's going to be giving us a talk on his recent findings on the habenula. And I will turn it over to him now. Go ahead, please.
Thank you very much, Brad, for inviting me to this symposium. It's actually my first time to give a talk through the webinar. So today I'm going to talk about fighting of animals. So any animal works fight with conspecifics, reindeers, bears, horse, elephants, leopards, or even the gorillas in the zoo actually fight. So today I'm going to talk about the fight of zebrafish. So if you actually isolate 2 male zebrafish for more than 24 hours and put them together back to the same tank, then these 2 fish start fighting. So the way they fight is very stereotyped. formulated, first displaying and circling around the others, and then biting the side bodies of the opponent. And eventually, one becomes a winner and the other becomes a loser. Actually, 10 minutes after the fight is over, you can actually quite easily tell which fish has become the winner and which fish has become the loser. So this is 10 minutes after the fight is over. The loser is actually staying very quietly at the bottom of the tank, and the winner is swimming freely at the center of the tank. So today, I would like to persuade that the one pathways from the habenula to the interpeduncular nucleus actually plays an important role in the regulation of this conspecific fight. The habenula is very much conserved evolutionarily from fish to mammals, and the pathway from the habenula to IPN, interpeduncular nucleus, is also very much conserved. So in case of the zebrafish, the habenula is largely subdivided into the dorsal habenula and the ventral habenula. And the dorsal habenula send out the axons to the interpeduncular nucleus. And then the dorsal habenula is further subdivided into the lateral subnucleus and the medial subnucleus. And the neurons in the lateral subnucleus project to the dorsal half of the interpeduncular nucleus. And the neurons in the medial subnucleus project to the ventral half of the interpeduncular nucleus. There is another component of the habenula called ventral habenula, which actually send the axons to the median raphe. So there are three components in the zebrafish habenula. So the lateral subnucleus of the dorsal habenula project to the dorsal IPN, and then the neurons in the dorsal IPN further send the axon to the structure called dorsal tegmental area. So this area in zebrafish actually are evolutionarily homologous to the mammalian periarchitectural gray, PAG. And as is known, that PAG is responsible for the control of the innate fear response to the threat. So the most anterior part of the PAG is, if it is stimulated electrically, then the animal takes a position as if they are trying to fight back. And if the most caudal part is stimulated, animal takes the behavior as if they are escaping or flight. And if the ventrolateral position was stimulated, animal flees. So three different areas of PAG actually regulate the different response to the threat. So considering the connection from the IPN with the equivalent of the PAG, we wondered whether this pathway from the habenula to IPN and then dorsal tegmental area may have something to do with the regulation of pathway of social conflict. So indeed, when we actually removed the brain of the winner, as soon as the fighting was over, and made the longitudinal slice, including the habenula and the IPN, and soak the slice in the solution containing the calcium indicator, Oregon green papta, then we stimulated the habenula, we can actually visualize the propagation of excitation starting from the habenula in the winner's brain. So as you see that the excitation start from the habenula and the IPN, and then it spreads to the dorsal tegmental area. In contrast, when we actually stimulate the habenula of the brain from the loser, then we actually see the spread of excitation from the habenula to IPN. Then after IPN, the excitation propagate not to the dorsal tegmental area, but to the median raphe in the posterior to IPN. So it gives us the impression that the IPN is acting as a switchboard, which downstream targets should be activated, depending on whether the animal is the winner or the loser. And this pathway of propagation in the winner's brain actually corresponds to this red pathway starting from the lateral subnucleus of the dorsal habenula. And then this pathway seen in the loser corresponds to this green pathway starting from the medial subnucleus of the dorsal habenula. So this red pathway may be putatively the winner's pathway, and the this green pathway may be the loser's pathway. So to prove that, we actually established two transgenic lines in which either the red pathway or the green pathway is specifically inactivated by the activation, by the expression of tetanus toxin in the axon termini, and see how such fish actually behave. So when we inactivated the putatively winner's pathway from the dorsolateral habenula, then essentially such fish in most cases lost. In contrast, when we actually inactivated the putatively the loser's pathway, so such fish actually almost all cases won the fight. So this manipulation of the fish actually proved our hypothesis that this red pathway starting from the lateral subnucleus of dorsal habenula is for the winner, and this green pathway starting from the medial subnucleus of the dorsal habenula is for the loser. So, so this red pathway is involved in making the winner fish to behave as winner. And then this green pathway is involved in making the loser fish to behave as loser. So then we actually wondered what actually synaptic change is responsible for this dominance of winner's pathway or the dominance of loser's pathway. So to examine that kind of things, we first examined how the loser's pathway is affected by losing experience. So we actually made the slice, including the IPN, and made the patch clamping experiment targeted at the neuron in the ventral IPN. So these are the neurons. Cell bodies are located at the intermediate zone of IPN, but their dendrites spread throughout the ventral IPN. So we actually examined. So one thing that we noticed is that when we excited the input axons from the habenula electrically, in case of the winner, almost half of the neurons did not respond to the stimulation of the habenular axons. But in the loser, almost 100% of the axon of the neurons in the IPN actually responded. So so the neurons in the IPN were kind of unsilenced by the losing experience. So to examine what is the cause for this unsilencing of the neurons, we actually analyzed the pattern of the excitation of patch clamp neurons. So these are the from winner, and these are from losers. And then we actually compared the activation time. So we noticed that in the losers neurons, so we actually observed many spikes, many currents that actually have much shorter activation time as compared to the most currents observed in the winner. So this change in the activation pattern actually suggested that different type of neurons, of receptors were expressed in the loser's neuron. And also suggested that considering that calcium permeable type AMPA receptor has shorter activation time. So the component of the AMPA receptor expressed on the neurons may have changed. Indeed, when we treated the slice with the antagonist against the calcium permeable type AMPA receptor, the neurons from the loser were, the currents were more affected. So unsilencing seen in the loser's brain, loser's IPN, appears to be caused by the expression of calcium permeable type or GLU-A2 lacking AMPA receptor. So we further asked, what is the cause for the change in the components of AMPA receptor? So the loser's pathway, the habenular's loser's pathway, express glutamate and acetylcholine. So we wondered whether the cholinergic transmission may be responsible for this change. So this is the probability of first type of the current. So as I already explained, in the loser's brain IPN, we see the drastic increase. So, but quite interestingly, when we actually treated the slice with the alpha-7 specific nicotinic acetylcholine receptor agonist, so even the slice from the winner's brain, the expression probability of the first type catch up to the level of the loser. And we actually also treated the slice with nicotine, and then the effect of nicotine was inhibited by specifically by the presence of the alpha-7 type antagonist. So these experiments actually suggested that the increase in the expression of calcium permeable AMPA receptor was due to the action by way of the alpha-7 nicotinic acetylcholine receptor expressed in these silent neurons. So that was the plasticity happening in the loser's brain in the ventral part of IPN. And the dorsal part of IPN also shows very much dynamic plasticity, especially depending on the actually internal condition of the fish. One interesting finding that we made was about the starved fish. When you actually, when we starve the fish, 1 day, 2 days, 6 days, and then 7 days, only the fish starved for more than 6 days showed the quite different fighting pattern. That is, these starved fish always won the fight. So this is the winning rate. So we actually wondered, what is the cause for this fight? And then, as you see here, if you let 2 starved fish fight, these fish fight significantly longer than the other combinations. That suggested that starved fish never gives up the fight until the opponent actually gives up. So we actually wondered whether this change in the behavior may be caused by the activation of the winner's pathway from the habenula. So we actually genetically silenced the this winner's pathway, and then we saw that in such fish, starvation did not make the fish prone to win. And then we actually examined how the excitation propagated from the habenula. And that what was interesting to us was that the propagation pattern of excitation from the habenula to IPN was very close to the winner's pattern. So that is that the excitation further propagated to the dorsal tegmental area. So this propagation pattern was observed in the starved fish even before they actually experienced fighting. So even before the fight, the excitation pattern was that of the winners. So we wondered what is actually happening in the IPN neurons in the dorsal IPN. And then we also analyzed each miniature EPSC current and compared between non-starved fish and 6-day starved fish. So we actually observed the miniature EPSC change in 6-day starved fish as shown. That is the increase, in this case, in the miniature EPSC with longer excitation time. So then we ran the literature search, and then we actually found that many AMPA receptor actually transcript has 2 types of mature messenger RNA. One type is called flip type by the inclusion of the exon 15, and the other type is called flop type. And if the AMPA receptor translated from the flip type mRNA has longer opening time as compared to flop type. So we actually wondered whether there is actual increase in the expression of flip type in the starved fish. So we treated the slice from the starved fish with the cyclocyanide, which specifically enhance the activity of the flip type. So the influence of this drug is much augmented in the 6-day starved fish. We did the opposite experiment by treating the slice with thiocyanate, which is the antagonist against the flip type. And then we could actually see the influence of thiocyanate only in the starved fish. So these two experiments support that there is an increase in the expression of flip type AMPA receptor in the starved fish. So we actually examined whether there is actual increase in the flip type messenger RNA in the starved fish. And indeed, we actually observed the significant increase in the expression level of GLA3B, which is the transcript in the starved fish. So it appears that starvation caused the biased alternative splicing, enhancing the production of the flip type AMPA receptor. So we actually further wondered which actually caused this biased alternative splicing, and wondered whether some cells which are activated by starvation may be involved. And the orexin or hypocretin neurons in the hypothalamus is known to be activated and send axons to the many places of the brain. So this is the orexin neurons in the hypothalamus in fish. And then axons from these orexin neurons actually certainly innervate the dorsal part of the interperambicular nucleus. We could luckily obtain this fish from Dr. Leo from Israel. So, so we treated the slice of the naive fish with orexin and see whether the similar change seen in the starved fish can be observed. So indeed, that the propagation pattern was actually skewed toward the dorsal IPN as compared to the naive fish, similarly to the starved fish. And indeed, we could also observe the biased alternative splicing by the treatment of the flip type F messenger RNA of GLA3B by treatment with orexin A. And then we could also express by the nitroreductase and treated the slice with treated the fish with metronidazole, which actually kills the orexin neurons. So then in that case, so effect of the starvation is actually is not seen as compared to the wild type starved fish. So this also supports that the orexin neurons is responsible for this change of the fighting pattern. So summarizing this data, hunger actually activate the orexin neurons in the hypothalamus, and then activated affected the splicing pattern of the AMPA receptor to actually enhance the alternative splicing for the flip type of GLUR3. And this actually potentiate the signaling from the habenula to IPN and eventually to dorsal tegmental area. And that caused starvation-induced weaning. So this may be part of the course for what Steve Jobs says, to win in your life, stay hungry and stay foolish. And then lastly, I would like to mention about the unexpected finding of the role of the dorsal habenula. We actually examined whether fish can learn two rules. So this is a kind of a plus maze experiment test for fish. And the fish can actually start from one arm of this plus maze and then requested to make a choice whether to turn right or left to get the food. And then we imposed two different rules. One rule is fish is asked to turn always right or maybe left or to the same direction, regardless of the color of the goal. Then in the second rule, fish is requested to turn to the same color, always red, regardless of which side of the arms the color actually is indicated. So this is an example of the directional rule behavior. So fish is requested to turn right, regardless of the goal color. Fish can behave properly. And this is an example of the behavior cued rule. So fish is requested to turn to the red goal, regardless of the direction. This time he made a mistake. But in most cases, fish can learn two rules. And then we can actually alternate the rule which we impose on fish. And then as you repeat the change of the rule, fish can more and more quickly adapt to the imposed rule. And then we actually wondered whether this connection from the habenula to IPN may have something to do with the behavior according to the directional rule. Because so this structure, as you may notice, have some left-right asymmetry. So we wondered whether they may be also involved in the behavior related to left and right. And indeed, actually, more than half of the fish which has the inactivation of the lateral subnucleus of the dorsal habenula can never learn the directional rule, while they can still learn perfectly the rule according to the presented color. So this part of the habenula, from the lateral subnucleus of the dorsal habenula to the dorsal IPN, that is the Wiener's pathway, is also involved in the decision making according based on the direction. And actually the fish inactivated with this circuit show the enhanced capacity to learn the color-based rule. So we interpret that this red circuit, the Winard circuit, is also involved in giving the increasing the attention of the fish to oneself, to one's own status, or the internal status, or left or right. And then this loser circuit may be also in controlling their attention to the outside. So in other words, the winner's fish care only themselves, but the loser's fish may care outside, including the winners. This is actually consistent with the recent data obtained in mouse. So it's shown that the dominant mouse prefrontal cortex shows the activity synchronized with its own movement, but the losers or the subordinate mouse prefrontal cortex neuron show the activity synchronized with the movement of the dominant mouse, not himself. So habenula may be acting as a switchboard to whether the attention should be directed to oneself or to outside. And it's also interesting that recently the group by Ruben Portugues reported that if the larval fish was presented with a streaming dots with a mixture of different direction, and then they actually ultimately interpret in which direction as a whole these dots are streaming, and then depending on their decision, they flip the tail. But before the fish flip the tail, the IPN is activated. Some part of IPN is activated, and then activity, for example, the right anterior IPN is activated, actually indicates there is a high probability of flipping to the right direction, maybe left, I can't remember precisely, but has a close correlation with the activity of IPN. So IPN is also involved in the coordination of the tail flip. So at this moment, we actually don't know how these things are coordinatedly regulated, but habenula IPN pathway may be acting as a simultaneous coordinator of the attention, whether to attend oneself or attend outside, and also behave winner or the losers. So we need to further study how these winners pathway and losers pathways actually interact with each other at the circuit level. And also, of course, we need to study whether this circuit is composed in the conserved both throughout the evolution. So this study was actually performed by many past and present members of my lab, as shown here, and in collaboration with the outside collaborator. And I want to express my special thanks to Dr. Leo Appelbaum in Israel for giving us the precious strain in relation with the hypocretin. Thank you very much.
Thank you. That was really fascinating. I would like to start off with a question of my own, actually. What is the evolutionary advantage of losing in a fight like this? Is there any reason to do it? To just sort of look for a balance between winning and losing?
Yeah. So losing actually, so unless somebody actually stopped fight, fish actually continue to fight. That is a waste of energy. So some type of hierarchy must be formed to make a peace. And then also the winner stopped actually attacking as long as the loser actually stopped fighting. So in fact, we actually have the data that the other part of the habenula called ventral habenula may be involved in the, so this is the fish in which the ventral habenula is inactivated. So even the fish lose, this fish continue to come back to the central part of the tank, and he keeps getting the attack by the winner. So this status is really the waste of time. So in some sense, to actually cause some kind of peaceful situation, this pathway is actually useful.
Right. So it's sort of like a co-evolutionary strategy.
Yeah.
That you need a population of people, of fish who are willing to win and lose.
Yeah.
Okay. Well, let me ask you some questions then from the attendees. Elizabeth Hansen asks, on what timescale does AMPA-R composition return to normal after starvation?
After starvation. So you mean that so after feeding?
Yeah, I think so.
Actually, we haven't checked that.
Okay.
Actually, zebrafish can withstand the starvation for quite a long time. Even if you actually starve the fish for 1 month, they can survive.
Wow. That's impressive. They don't have a lot of fat.
Yeah, I don't know what kind of metabolism makes that possible, but they can withstand very long time.
Do they transition into a low metabolism state at some point?
It's actually interesting, probably research field.
Yeah.
But I don't know.
Okay. All right, we have another question here from Sebastian Meli. Sorry if I've mispronounced that name. And it is this. Thanks for the great talk. Have you tried manipulating habenula IPN after a fight to determine whether a specific phenotype can be induced regardless of the outcome of the fight?
I see. That's also another interesting experiment. But to do that, we need to, we can do optogenetic experiment, and it's theoretically possible, but we haven't tried. Especially connecting the optic fiber and letting the fish fight is a bit difficult. Probably the connected fiber may come off. Blood, your voice is muted.
Yeah, so I guess optogenetic would work if you had only one of the fish. Then you could just shine a light in the entire tank.
Yeah.
So Takashi asks, do you have some idea how the HB-IPN pathway interacts with hypothalamic areas controlling aggression? Does the PAG integrate information from both areas?
Yeah, so as you know, that hypothalamus is related to the initiation of fight. So we actually think that this pathway from the habenula to IPN may have something more to do with the termination of fight or the giving up the fight. And we actually don't know how they mutually interact each other. That's also interesting subject, but we don't know the answer, sorry. So it may actually, PAG may have some, PAG is obviously the downstream target of hypothalamus. And I actually don't know whether there's reciprocal connection.
So do you think there's a deep causal connection between this idea of attending to others and losing a fight? Or do they perhaps just both coexist within the same circuit?
Yeah, so attending to, so the winners actually don't care what others are doing. So they actually care only by themselves. And then losers have to pay more attention to what others are doing, whether they are attacking or something. So it's actually consistent with kind of a, it's reasonable, but actually I don't know the true mechanistic explanation at this moment.
Right. So do you think it's fair to say that this would sort of be the equivalent of the sort of winning pathway would be sort of the equivalent of a sociopathy within at least the zebrafish?
Yeah.
Or narcissism, or
expand our
hugely
very optimistic interpretation.
Yeah. Right.
Yeah.
Yep. And on the on the other side, the empathy, or the losing circuit would be more associated with what we might think
actually,
of.
There are people who are too sensitive to others' emotion, too much sensitive people. And I'm actually wondering whether that has something to do with
I like your background, Abhishek. Looks very, very warm.
Yeah, it's where my parents stay.
Oh yeah,
So, yeah.
I thought it looks as pretty as stock photo.
Oh yeah, I'm quite lucky that they moved to this awesome location that I can go home and visit, and yeah.
Okay, let's go ahead and get started.
Cool.
Great.
Awesome. Okay. So I don't see anyone yet, but we'll go ahead and get started. I would just say, panelists, you're encouraged to ask questions as well. Don't fall to the regular conference trope of asking questions at the end. Feel free to ask them as they come to you. So we're going to start off with Farhan Augustine, who's currently a grad student at University of Maryland, and is going to present his work that he's been doing at Johns Hopkins under Harvey Singer. Go ahead and take it away, Farhan.
Hello, everyone. Are you able to see my screen yet?
Yeah, you're good.
Yes, okay.
So my name is Farhan Augustine. I am a clinical research assistant at Johns Hopkins School of Medicine, and I am a graduate student pursuing a PhD in biological sciences. And I will be talking today about my work with deer mice. The picture here on the right is of a deer mouse. I found this on internet under Creative Commons license. And my talk will be primarily centered around the cortical endogenous opioid findings and the link between repetitive behaviors. So to begin, let me introduce deer mouse and the type of behavior that we observe in these mice. At an early age, mice begin to display frequent repetitive behaviors, such as the ones you're seeing here: repetitive jumping, back flipping, pattern running, etc. And what we hope to do is to understand the pathobiology of these behaviors and to draw parallels between human clinical disorders such as OCD, tics, motor stereotypies, and autism, and try to understand from an animal model perspective how we can apply the information from animals to the clinical disorders. Behaviors are often talked about in terms of being goal-directed and habitual. Goal-directed behaviors are consciously performed, they are controlled, flexible, and adapt quickly to whatever the desired purpose is. The pathways are thought to originate from prefrontal cortex and the orbitofrontal cortex and project down to dorsomedial striatum. On the other side, the habitual behaviors are performed as repetitive, they're stimulus driven, they're slow to change, and often have no or little link to current or future goals. And the pathways that are considered habitual pathway originate from premotor cortex and supplementary motor area and are thought to project to dorsolateral striatum. I have included ventral striatum in this figure because recent studies are beginning to show that ventral striatum may be perhaps gating the transitions between goal and habitual behaviors. The other major pathways that are thought and talked about in literature is this direct and indirect pathway system. Motor information from the cortex is projected down to striatum. In the striatum, genotypically distinct medium spiny neurons of the striatum that either express dopamine receptor D1 or dopamine receptor D2 are thought to then segment that information into direct pathway or the indirect pathway. The direct pathway MSNs express dopamine receptor type 1. The indirect pathway MSNs express dopamine receptor type 2. And the flow of information goes from striatum to the globus pallidus interna via the direct pathway, D1 expressing MSNs. And that is thought to enhance the motor activity, whereas the indirect pathway going to the globus pallidus externa is thought to diminish or reduce the motor activity. However, in reality, the situation is a lot more complex. There are a lot more interactions between different regions and different neurotransmitter systems than we talk about when we just talk about behaviors. So here's a picture from one of my publications from this year that depicts or tries to depict how many different interactions there are between different regions of the motor circuit. The one on the right is just a simplistic representation of the one on the left. It depicts just the bidirectional or unidirectionality of these connections and interactions between different regions. So please keep in mind that interactions and connections are very complex. Researchers all over the world are trying to understand the best that they can. However, recognizing the multiple interactions between the neurotransmitter systems and the different regions, the question is, where do you start to look for the problem of repetitive behaviors? The problem could be anywhere in any of these interactive pathways. So when I joined the lab and I took the deer mouse project on, I decided to focus on the cortex, striatum, and basically the basal ganglia. Basal ganglia in the mouse kind of looks like this. And what I did was I took a neurochemical approach to try to identify if there were major neurotransmitter abnormalities that we could identify, and that if we can correlate those abnormalities to the repetitive behavior, then we could potentially have a hook that we can use to pull on the string of this repetitive behavior pathobiology. So I chose to look at 5 of these neurotransmitters: glutamate, GABA, dopamine. Dopac is actually a metabolite of dopamine. And the number 4 and number 5 are both opioids, leuenkephalin and dynorphin A. The rationale for measuring opioids is multifactorial, including their ability to regulate the release, presynaptic release of glutamate, GABA, dopamine, etc. And endogenous opioids also play a major role in the habitual and goal-directed pathway systems, as well as the gating of the striatal direct and indirect pathway systems. So from the findings of the neurotransmitters, what we found was that in the FC, in the frontal cortex, GABA neurotransmitter was reduced compared to the prefrontal region. And the leuenkephalin levels were higher in the frontal cortex compared to the prefrontal cortex. And it was statistically significant difference between the two regions when it came to opioid enkephalin levels. Luckily enough, the enkephalin and dynorphin levels both significantly correlated with the repetitive behaviors. So the lower the enkephalin level was, the greater the activity we saw. So keeping all of these things in mind, we came up with, we tried to understand what might be going on. So from doing some literature research, I found that GABA interneurons are primarily expressing enkephalin in the frontal, in the cortical regions. Therefore, assuming that enkephalin, which is represented here by these stars, is inhibiting GABA, and GABA then disinhibits the cortical pyramidal glutamatergic neurons, which will fire down into the striatum, therefore innervating the dorsolateral striatum, because that is part of the habitual pathway system. I should reiterate that habitual pathway, the frontal cortical regions innervate the dorsolateral striatum, whereas the goal-directed pathway, the prefrontal cortical regions, innervate the dorsomedial striatum. So keeping our data in mind that we have found higher levels of enkephalin and lower levels of GABA, again represented here by these stars and the broken red arrow of this GABA, what we are hypothesizing is more innervation of the dorsolateral striatum. Excuse me. Okay, then what about the direct and indirect pathway system? In literature, I found that prefrontal pyramidal neurons primarily innervate D2 expressing MSNs, which are part of the indirect pathway. So keeping that in mind, if the pyramidal neurons are disinhibited in the frontal cortex, we should expect greater innervation of the D2 expressing MSNs, or perhaps even D2 expression itself, the receptor expression itself. So what we then decided to do was let's overcome the LCMS analysis, some of the challenges that we find in the LCMS, the liquid chromatography mass spectrometry, is that we lose the spatial context. We lose the ability to see where in what region are we exactly. So therefore, we decided to go with in situ mRNA hybridization, where we can have a greater control and we can see in what regions are we spatially, and to understand the expression level of different receptor types in the frontal region and the prefrontal region and the striatum. Some of the things we are currently quantifying are the D1, D2 receptor expression, as well as mu delta kappa opioid receptor expression in the cortex and the striatum. I have some preliminary data from the current directions and the current investigations. So again, the hypothesis that we came up with from the liquid chromatography mass spectrometry was that we would observe a greater innervation of dorsolateral striatum via the disinhibition of glutamatergic pyramidal neurons in the frontal cortex. And another way to state this is to say, if cortical neurons in the FC are less inhibited and there is less production of opioids in the dorsolateral striatum down here, which suppresses inputs from the frontal cortex of mice that display high activity, then we should expect to see an inverse or negative correlation between the opioids that are released here and the activity of the deer mice. So the preliminary analysis from the RNA scope and the expression of dopamine and the opioid receptors is showing some promising results that support the hypothesis. However, there are a lot more questions that are generated from the data. What we have observed so far is that in the dorsomedial and dorsolateral striatum, the dopamine receptor expression positively correlates with the activity of the deer mice. And the same thing is happening with the ventral striatum, ventromedial and ventrolateral striatum. However, the correlations are more significant in the lateral part of the striatum, here shown up here and down here. So these are the two regions that are involved in the habitual pathway. As we had hypothesized, the dorsolateral striatum shows a significant positive correlation between the dopamine receptor expression as quantified by the in situ RNA hybridization and the repetitive activity. However, it generates more questions because D1 receptor expression is thought to lead to more motor output from the striatum. D1 MSNs are involved in direct pathway, therefore it is believed that direct pathway leads to more activity. However, what we are finding is that perhaps there are more D2 expression, there's more D2 expression in the striatum than we previously thought, and perhaps that is one of the things we should further look into. Here's an expert opinion or excerpt from a recent paper I was reading, which suggested that D2 MSNs inhibit direct behavior, and it leads to goal-directed behavior, whereas the ventrolateral striatum D2 MSN activation initiates the action, and the fact that we are seeing the ventrolateral D2 expression and positive correlation, a significant positive correlation with the activity, suggests to me that D2 MSNs, D2 expressing MSNs, are perhaps more involved with our repetitive behavior phenotype than we previously thought. The comparison I'm trying to draw here with these two graphs is a meaningful one, because in the striatum, D2 expressing MSNs are the ones that release this Lewin-Keflin opioid neurotransmitter. And as the expression of dopamine D2 receptor goes up, we are seeing the expression of opioid and Keflin going down. And what this is indicating to me is that we need to take a deeper look at the expression level as well as the activity level, and how we can do that is through electrophysiological recordings. We can identify MSNs based on the dopamine D1 expression and D2 expression and do electrophysiological recordings. We can perhaps even do pharmacological manipulations and see the behavior impact of let's say introducing a opioid antagonist and agonist in a specific location in the dorsolateral striatum. Or perhaps there are other regions that we should explore that may be introducing situations that are almost significant, such as the D2 expression in the dorsal medial striatum, that could be coming from regions like thalamus or hippocampus or other areas of the brain. So by no means do we have exact answers, but we are a lot more closer to understanding the role of endogenous opioids and their gating of the cortical output to the striatum than we were previously.
Sorry, Farhan. I was so wrapped up in what you were saying that we've run out of time.
Oh, wow, it's been 15 minutes.
I'm sorry. We have a question for you. If you want to type to that, that'd be perfect. Or we can circle back at the end if we have time. We're going to quickly switch gears. Sorry, I was so wrapped up in listening to that. To Abhishek Naik, who's going to present from University of Alberta. Go ahead and take it away, Abhishek.
All right, do you see my screen? Okay, great. All right, welcome everyone. Thank you so much for taking the time for all around the world to have a discussion about personalized brain state targeting via reinforcement learning. So this is joint work done with Amritpal Singh, Kaushani Biswas, Harini Sudha, Matthew Schlegel, and Kyle Matheson. All right. All right, so the motivation for this research project was that to do something to help achieve desired states of the mind without active effort by an individual. So let me make this motivate this with an example. So, so there's something from my personal life that I have been trying to meditate recently, as many of us might have tried. And so I haven't been particularly successful with it in the sense that, so there's this voice. So there are many ways to do that, right? And one of there are some apps, and so there's this voice that tells you that, okay, close your eyes, take some deep breaths, like hold it for a few seconds, release it, count your breaths, or focus on some part of your body and relax it and things like that. And so I keep getting distracted by other things that are going on in my mind. And so I was wondering that, hey, so when I watch some sports on TV, I'm instantly excited, right? So like something is going on, I'm super enthusiastic, focused on that. Wouldn't it be nice if there was something like that? So let's say look at a screen, and in like 2 minutes, it takes me to a meditative state, right? Wouldn't that be nice? And so that was the motivation behind this research project. And I was wondering how we could do that if possible, right? So maybe meditative state is one example, but you can stretch it to think that you could, go to, for example, a deep sleep state, right? So imagine that you just had a surgery of some kind, you're in an accident or something, and now the surgery was successful, and now you're in the recovery room. But unfortunately, you're drugged, and you have a lot of physical discomfort, some pain, and all of these things can throw off a person's sleep schedule, right? And all the noise and lights in the recovery room don't really help. So it would be nice if there was something that you could just look at that would instantly, take you to the deep sleep state. And so maybe it happens over a period of 2 minutes or 5 minutes, but it would be nice if it was something you didn't have to actively try to do, right? And So yeah, so some other people have also thought about how they could potentially make this happen. And so there are these, so if we take sleep as an example, there are these sleep assistant systems. So the simplest one could be something open loop, which is like you play lullabies or some soothing music and hope that it puts you to sleep. And I call it open loop because there is no feedback mechanism here. So maybe you are not falling asleep, and yeah, it keeps on playing and doesn't really help you, or you've fallen asleep and it's actually creating some noise that might wake you up, right? So there's no feedback mechanism here. And so that's why I call it open loop. In contrast, there are these closed loop approaches. So for example, Zhang et al. In 2015 came up with something called iSleep, in which it monitors your EEG data in real time, and based on some hard-coded thresholds, it decides when a person would have fallen asleep. And again, based on some other thresholds, tries to decide which sounds to play in order to put you to sleep, right? And now there are some more proprietary companies that are trying to do something in this space: Muse, Mind Alive, Snozic. I'll get into some of the issues there which we are trying to solve. But all of this relies on the existence of a goal state that the system can take you to, right? So for example, in sleep, there is this deep sleep state that the objective of the system is to take you to, right? And so fortunately, at least in sleep, there is some evidence that that your alpha levels go down and your theta levels go up, and that is an indication of an onset of sleep. So maybe there is something that we could work with there. And now, if there are these goal states, we also need to identify them, right? And so you can imagine there's a system which takes your EEG data in real time, for instance, and is able to give a score to at every second, for say that, okay, you're this state, you are like 0.6 or 60% close to a deep sleep state, right? Versus like a state which is like 98% close. It would be nice if this was measurable, quantifiable, so that you can actually mediate the audio or visual cues that you're using to get to this particular goal state, right? And so fortunately, there is some recent research which uses some recent deep machine learning techniques trained on data sets collected from a large number of people all over the world to categorize these sleep stages. Okay, that is something maybe we can use as well. And then finally, we also need to actually be able to affect the brain state, right? That if I play some sound, that should actually help you get from state A to state B without you doing something about it. And again, there is some research which says that in case of sleep, there are some audio cues like binaural beats or some kind of pulses that they can give of certain frequencies that induce a certain type of response in the brain. Okay, so these are all the ingredients that you could use. And the closed loop systems that I talked about earlier try to use some or more of these techniques, but they are limited in a sense because they're trained offline from data collected from a population, which is not tailored to any specific individual, right? And if any offline adaptation per individual, if at all, is very slow. And if you're using some kind of this brain-computer interface, it is critical to have the speed of adaptation to be quite high, because poor initial performance or long training periods are barriers to adaptation of such technologies, right? So if I put on the system and it is making these weird noises and for 20 days it is not really helping me sleep, I'm not going to use it. So we need something which acts in real time, is able to learn from the data it collects from you online and react to it and learn very quickly, right? And so that's where I think reinforcement learning comes in. So what is reinforcement learning? So it's a subset of machine learning in which the system learns via interaction with the world. So for example, this is the agent which is acting, and it is interacting with the world. It observes, it gets this set of observations from the world, and in response to those observations, it performs some actions, right? And for that action, it will receive another observation from the world and a corresponding reward, right? And so to make this a concrete, so imagine that you're learning to ride a bike, right? So, so you are trying out different things. So you're on the bike, you're trying to balance, you're trying to pedal ahead, and there is no one like your parents are there to like help you, but they can't really tell you that, you know, apply 40 pounds of pressure on your right foot while leaning 3 degrees to the right? Like they can't give you such instructive feedback, which is common in the supervised learning setting, right? That for every instance, you have some particular label associated or a correct action associated with it. You don't have that. But what you do is you try something and maybe you fall and you're like, okay, maybe this was not a good thing to do. You get like a negative reward. And so now you try a second thing and maybe that works and you're able to cycle ahead and your parents are clapping, and that's the positive reward that you're using to reinforce that action, right? And so that's basically what the reinforcement learning paradigm is. And so what we're trying to do is, let's say the world is this person who's using this particular system. So you get some real-time EEG data from the world, and then you provide some audio-visual cues to this person, and then the reward is your distance to the goal state, which you can measure in some particular ways. And so we developed a proof of concept system in which we made a lot of simplifying assumptions, and so basically modeled the entire brain state as this scalar which lies between 0 to 1, and the current state is like one particular real value between this thing, and the goal state is, let's say, somewhere here, right? And your actions is to these three, that the effect of this action is that this current state moves towards the right, or it increases, move towards the left, or stays there. And we made some a model of how the brain state, the simplified brain state, could change. So it's drawn from a Gaussian and has some effect of the action. And so this keeps changing, and you take some actions, it changes, and you get some reward proportional to that. And so based on a recently proposed algorithm, so when you start training, this is the goal state. This is how the state of the brain is evolving. And you're trying out different actions. They don't really seem to work. And this action says that, okay, the arrow means that it takes the brain state towards the right or the left. And towards the end of training, you'll see that it has learned that wherever the brain state is, if it's on the left of the goal state, take it to the right by taking this right action, or take it to the left using this left action if it's here, right? And so after a few interactions with the real world, this system learns this policy very quickly. And that is something very encouraging that we have. And so now, to moving on from this prototype to product, there are multiple things that we are doing. So like the obvious one is take the full multidimensional, let's say 30-dimensional vector of EEG data as input, along with certain features that can help out, like time of day, age of the individual. And then you categorize the input that you see, this entire thing, based on how close it is to the goal state that you have, which is the sleep state. And then actually use full-fledged audio and visual cues to affect the brain state instead of this simplified actions which deterministically affect the brain state. And so the goal is to develop an active closed loop system that learns in real time the set of audiovisual cues best suited for every individual to lead them to a physiologically objective goal state from any state. And so that is kind of it. And if you have any questions, feel free to post them in the chat here, or thank you so much for listening.
Thank you so much, Abhishek. So just to stay on schedule so that people coming in and out know what talks they're in, we're going to move on to Kelly, but please, people in the audience, put your questions in the Q&A box, because at the end we can circle back to the previous speakers and and get their inputs. So now we're going to move on to Kelly Garner, who's a postdoctoral Marie Curie research fellow working with far too many institutions and people for me to list. So Kelly, I'm just going to have you take it away.
Sure, thank you. Just give me a moment and I will share my screen. Okay. One moment. I should be able to share just the presentation slides with you, right? Just the browser. I feel Zoom is not behaving. Here we go. This is what I want to do. Okay, so you guys can all see that, correct?
Yeah, looks great.
Beautiful. Okay. So this is the human basal ganglia. This collection of nuclei have been strongly implicated in the influence of associative learning on action selection. So we're interested in mapping the functional changes in the basal ganglia when associative learning instead influences visual selection, as this is currently largely underspecified. However, these regions are tough to image. Their higher iron content relative to the cortex means that signals detectable by MRI decay sooner than other regions. Therefore, to accurately map functional changes during visual selection, we need to identify the best protocol to image the basal ganglia. So that's our aim, which is to compare 70 MRI protocols for signal quality when participants complete a task that taps the influence of learned associations on visual selection. So how we did this, we recruited 5 participants who completed 4 sessions. The first was a task learning session where they performed a variant of the Posner queuing task. Participants responded whether a Gabor was oriented clockwise or counterclockwise. The target and distractor were preceded by probabilistic spatial and reward cues, and I can go into those during questions if desired. So then participants performed this task over 3 sessions in the 70 MRI scanner, where we compared 3 imaging protocols counterbalanced across sessions. The details of the protocols are listed here, and the protocols were each optimized to different spatial and temporal resolutions. So for now, just note that they differ in their TR, and I shall refer to them by their TRs from now on. So then we assess the signal quality by using the contrast noise ratio. So the way we calculate this is we fit a GLM to our data and we ask what is the ratio between our estimate of the amplitude of a contrast of interest. So this could be the response hand that participants are using. And what is the ratio of that estimate to the standard deviation of the residuals of the model. Thus, we have a measure of the size of our effect of interest relative to a measure of the noise in our data. Okay, so this is example CNR data from 1 participant over our 3 sequences. The brain is intersected, so you can see across a few of the ROIs. Because for my question, I care about the magnitude rather than the direction of the difference, I'm going to extract the voxels from each ROI and compute the average root mean squared CNR for that region. And that's what I'll take you through now. So just to recap, on the left, we have our regions of interest, which comprise the subnuclei of the basal ganglia and a few cortical nodes we identified using a neurosynth meta-analysis. And here we're looking at the RMS CNR measure for the response hand contrast. So remember, we know that the basal ganglia is well implicated in action selection, and this is a typical contrast to use when assessing signal quality in the basal ganglia. So here we have our CNR measure plotted by the TR on the x-axis, or the protocol on the x-axis, with each subject plotted as a single line, and this is for the global pallidus external region. And so now I show you for all our ROIs, and as you can hopefully see, we don't see any particular pattern in the cortex, which is that top row of plots there. But we do see a statistically significant trend in the basal ganglia ROIs, for the protocol with the shortest TR offering the best signal. So now the question is, do we get a similar result when we look at a contrast that is not typically used to assess signal quality in the basal ganglia, such as on which side the target was presented? And that's what I'm showing you on the left here. So again, we see a non-consistent pattern for our cortical ROIs. But hopefully you can see we actually see the opposite pattern in the basal ganglia, where now the protocol with the longest TR offers the best signal. Therefore, it appears that if you're optimizing your protocol, this can be dependent on the contrast that you're using and the regions that you want to look at. So in conclusion, when mapping the functional activity of the basal ganglia, the optimal protocol appears to differ dependent upon what you seek to map. Protocols that are good for mapping motor activity are not the same as those which are best for mapping shifts in visual spatial attention. And we need to disentangle whether this is due to the changed temporal or spatial resolutions, or if there's an interaction between the timing of mental events and the hemodynamic response function. And so I'd just like to say thanks to my collaborators and for your attention.
Awesome. Thank you so much, Kelly. So please feel free to put your questions in for Kelly or for our previous two speakers. I will abuse my position as MC and take the first question. So what, I know you mentioned sort of some next steps, like what's your sort of future directions on this project, Kelly?
So our future directions are to now run a larger study using our best protocol, so we can start teasing apart how learning about probabilities and reward values influence visual selection. So the story of what we know about visual selection so far is very cortical based, and we're a bit underspecified, but we know the basal ganglia should be contributing to learned association, so that's really what we're looking to pin out next.
Awesome, very cool.
I have a question for Abhishek, if that's okay.
Oh yeah.
Yeah, for
cool.
sure.
That was a super cool talk. I really enjoyed it. I was just thinking about, so I've been super interested in doing similar things. And I was wondering what so with the brain state, which is a configuration of activity across EEG electrodes, for example, as you can achieve multiple, you could possibly achieve that brain state through multiple mental states. And so I was wondering, have you guys thought about that at all? Are you like seeking to teach people particular strategies, or what's your thought on the mapping between the EEG brain state and the cognition that's going on underneath?
Right. So yeah, that's a great question. So as you might have seen, one of the things that we were focusing on was that there should exist a physiologically objective goal state. So for example, the deep sleep state is something that across people, if you take the EEG data of that person during the deep sleep state, you can instantly tell that, that person is in the deep sleep state, right? And you would hope that that is the case, as opposed to, let's say, if I take a recording of a person jumping around, it shouldn't be the same as the deep sleep state, right? So, okay, so now that we have these physiologically objective goal states, now the idea is to be able to get to this from any starting state. So let's say, I had a nice relaxing day and I go to bed. From that state, I should be able to get to this goal state, versus I'm super stressed about a presentation. I just had a bunch of coffee. Hopefully I can still get to that goal state from this position, right? And so the starting state shouldn't matter, but the point is that it should be able to take you to the goal state. And the thing about these reinforcement learning systems is that they learn from whatever data they encounter, right? So for example, if maybe the first time I start from a caffeinated state, it might not be able to do as well, but it is trying out different actions. seeing which work, reinforcing the ones which do work, and maybe the next time or the third time it will be able to directly take me there, right? And so the good part about this is like you can start from any initial state, any starting state, and with some training, it will learn a policy to take you to this goal state. So yeah, that's the end goal. Obviously, it's harder to execute than to talk about it. But yeah, that's what we're hoping to achieve.
That would be a dream. Please make that product. I need that in my life.
Yeah, as do we all, yeah.
Could you imagine would it work a similar system where you change the goal state to be like a state of arousal? Like I run EEG studies and apparently have quite boring paradigms, and like fighting alpha waves in my zoning out participants is a constant battle that I just try to use my personality to defeat. But I'm wondering if you could shift the goal state to awake rather than asleep and still use a reinforcement learning system.
Yeah, so the good thing about this is that as long as the goal state is something you can uniquely identify, it would work out. So like it shouldn't happen that it's aliased with some other state which is, completely opposite from what you wanted, right? So for example, if you wanted an aroused state, the brain state at that time should not be the same as when you're sleeping, right? Otherwise, like, according to the system, it took it to the state you wanted, but it happened to be something completely different, which it doesn't know, right? Because it just can see the brain state, it doesn't see the behavior. So that's why maybe you can help the system out by giving it some other cues. Let's say, in case of the sleep system, we are thinking maybe time of day and age might help. For example, tell that, if it's nighttime and the state is like this, the person is probably trying to sleep, right? And as opposed to like 3 p.m. or something. So yeah, so it could be any goal state, it could be any starting state, as long as things can be identified and given a score based on some function that you have trained.
Very cool. Farhan, I was wondering during your talk, I know you started your talk relating like the importance of this work because of how it relates to human models with disorders that have repeated behaviors. I was wondering, is there a need to understand why the mice are having repeated behaviors? Like, does it matter what's driving that in order to relate it back to humans? Like if it's anxiety or stress or is that just not part of this? My field is not very far away.
That's a great question. So there is a possibility, or I mean, there's always a possibility that the animal model has evolutionarily evolved to do these behaviors, and the circuitry that's involved in the motor behaviors has evolved to be like this. If we can understand the normal physiological function of the motor circuits in deer mice, we can perhaps take that information and identify how it's different from other animal models, from another mouse strain. And we often use mice as a good model. There are lots of different models, but we often use animal model research to try to understand human symptoms, human clinical symptoms. And that has historically been a good way to do research. But on the other side, we also run human studies to do MRIs, to do fMRIs, and do spectroscopy to identify the circuits of human brain and see how well they are distinguishable from the animal research. The cool part is that we're identifying links that are showing us that perhaps the GABA neurotransmitter system in general is altered for example, what I am involved in is the research of motor stereotypies, human motor stereotypies. So in those children, we have identified linkage of GABA in the frontal and a little bit anterior of the frontal cortical region. And the interesting part that how it relates to the deer mouse is that we also see reduced GABA in the frontal cortex of the deer mice. However, we still need to be able to explore a little bit more before we can draw a parallel between what's going on in humans versus deer mouse.
Awesome. Thank you so much. Well, guys, we are to the end of our session and we'll shortly be transported out of this room against our will. So I just wanted to say thank you so much to our panelists. You guys did an awesome job and I really appreciate you sharing your work with us.
Thank you, Chloe.
Thank you, everyone.
Yeah, thank you for moderating the session. Bye-bye.